and gentlemen, as they say, there isn't a moment that doesn't arrive, be it a minute or a second. My name is Chipiwa Mandiwana Neudhani, and I'm standing here on my capacity as the convener of the Faculty Higher Degrees Committee in the Faculty of Science and Agriculture, and also as the chairperson of the local organizing committee of the 15th Faculty of Science and Agriculture Postgraduate Research Day. First of all, let me take a moment to extend my sincere greetings. I'll wait for Prof. Siwe to sit down. Thank you very much. Let me extend my sincere greetings to all of you in attendance, and also those who might be watching us from the University of Limpopo YouTube channel. So here we are today. It is indeed the 15th of September, 2025, and for the next 3 days, until the 18th of September, 2025, this is where we are going to be gathering like this and engaging one another as the scientific community. Ladies and gentlemen, before I could go on, please join me as I begin with the acknowledgements as I've lined them up. So to start with, I'm going to acknowledge the management of the University of Limpopo under the leadership of the Vice Chancellor and Principal, Professor Mahlo Mukhalong. And in this case, it's in absentia. Along the same vein, I'm now going to move on to acknowledge the Vice Chancellor and Principal Designate, who is in our midst, Dr. Jeffrey Mabilibili. Then I'm going now to move to the host of this conference. And in this case, we have the Executive Dean of the Faculty of Science and Agriculture, Professor Liseilan Mampuru. And so Prof. Mampuru, at this point in time, also allow me to say on behalf of the Faculty of Science and Agriculture, we really are thankful for the support that you have given us as we were preparing for this conference. So we really, really thank you so much. Can we please give him a round of applause for that? The Faculty of Science and Agriculture Management, and in this case, I'm mentioning the directors of the school, the heads of departments, and different units, and we also acknowledge you in this case. Clap for them, please. Thank you. So coming back now to the objective of this year's conference. So this time around, the Faculty Higher Degrees Committee organized this conference in the manner that we did. But remember, we couldn't do it alone. We have some stakeholders that came on board, and starting with, I'll mention all the sponsors. These are the people that also made it possible for us to be able to have this event in the manner that you can see. Also, I would like to move on and acknowledge the invited guests, because without them, we really cannot have the event as we wish to have. For them to put away everything and decide to join us. We really, really appreciate. So in this case, of course, that includes the keynote speakers that we have lined up for the next 3 days, the plenary speakers also lined up for the next 3 days. The other important stakeholders are the presenters. Without them, we could not be gathering like this. And here, I would like to specifically mention all the students who are going to be presenting, the staff, and we also don't forget the supervisors who are behind all the work that the students do. I'm also going to say my acknowledgement to the University of Limpopo Marketing and Communication, and also the University of Limpopo Information and Communication Technology, ICT, as we always will know them like that. The last group that I want to acknowledge is the Faculty High Degrees Committee, also including in total the local organizing committee for this conference. So ladies and gentlemen, we are here, the 15th edition of the Faculty of Science and Agriculture Postgraduate Research Day. So this year, the Faculty High Degrees Committee decided to organize this conference under the theme, Advancing Science Through Research, From Lab to Market, Integrating Science, Innovation, and Entrepreneurship in University Research. This is the theme that the Faculty High Degrees Committee felt is very relevant and also suiting the times that we are in as the Faculty of Science and Agriculture, and also as members of the University of Limpopo and also the society. So without any waste of time, we're now going to move to the next item, which is it's welcome. And this item is going to be taken off by the Vice Chancellor and Principal Designate, Dr. Jeffrey Mabiliville. Thank you. Let's give him a round of applause as he comes to the stage.
Dumelang. Abusheni. Good morning. Hoyemore. Machiluni. What else? Sawubona. Osanbonani. Thank you very much, Program Director and Convener of the conference, Professor Mandiwana Neudane. I should also join you in acknowledging in our midst important people, given the fluidity of my standing at the university currently. If I don't do that, I might not get the support that I need going forward. Firstly, in absentia, Professor Matlo Mkhalong, who is not able to join us today due to his commitments at the university that he could not wrestle himself out of. But secondly, also the Deputy Vice Chancellor for Research, Innovation, and Partnership, Professor Mutiba. I'm informed that she will be joining us shortly. Executive Dean of the Faculty, in acting capacity, Professor Mampuru, who has already been acknowledged. And I should indicate also that it will be remiss of me not to acknowledge Professor Siweya as the former Executive Dean, who played a very instrumental role, I understand, in conceptualizing this Postgraduate Research Day 15 years ago. Professor Siweya, welcome, and you are acknowledged. Your efforts are really acknowledged. The directors of our 4 schools, Professor Van Staden, Professor Chauke, Professor Mafeo, and Dr. Takalane, as I understand it, from Mineral and Physical Sciences. Our distinguished guests and partners and researchers from other universities and research institutions, particularly our keynote speaker, Professor Mutalipule G. Matsavisa from University of Free State. I understand he will be joining us virtually during the course of the deliberations, and he will then give his keynote address virtually. Dr. Nomusa Dlamini from the CSIR, Dr. Darren Past from the University of Alberta in Canada, Dr. Samkelo Malchas from the University of Pretoria, Dr. Kulufelo Malemela from SMU, a former colleague of mine in the Department of Microbiology, microbiological pathology, as they call it. And also Dr. Simon Lashma from Agriculture Research Council, Dr. Emmanuel Mtongo from University of Venda, Professor Michael Mueti from Tshwane University of Technology, and one of our own also, Dr. Yvonne Maila, Agro-Food Technology Station Coordinator. I should also, in absentia, welcome or acknowledge Dr. Jacob Mashilu from the University of Limpopo Department of Agriculture and Rural Development. I'm informed that he will also be joining us later during the course of the day. It will also be remiss of me not to acknowledge Professor Masuku, who was also, together with Professor Ware at the time and the directors of the schools at the time, played a very critical role in conceptualizing and framing this postgraduate research day of the university. All academics from our university and other universities present here today, members of the organizing committee led by Professor Mandiwana Neudani, the faculty higher degrees committee, including also the academics, the support staff from the office of the dean that were instrumental in organizing this event for us. I should also acknowledge postgraduate students and researchers present here in this hall for the kind of work that you are doing. And also the presenters who will be coming here to present exciting papers for us to build on. As I've already indicated, I've been asked by the Vice Chancellor, Professor Mkhalong, to represent him at this conference as he's caught up in other matters at the university. Please accept his sincerest apology. He would have wished to be here with you, but unfortunately, exigencies of the moment have prevented him from coming to join us today. He wishes you all the best as you grapple with the theme of the research day, which is From Lab to Market, Integrating Science, Innovation, Entrepreneurship in University Research. It is a great honor and joy for me to welcome you all to the 15th Faculty of Science and Agriculture Postgraduate Research Day, which I'm told will run from today until the 18th of September 2025. The university has a total of 24,762 students, and over 986 students are students in the Faculty of Science and Agriculture. Of that number, that is 24,762 students, approximately 808 are postgraduate students. The faculty's proportion of that postgraduate students stands at around 27.9%. That is 1,062 students are, postgraduate students are from the Faculty of Science and Agriculture. Now, given the faculty's preeminent role in postgraduate education, the research day event you have organized and the thematic areas you have identified are critical to strengthening our postgraduate education efforts on the one hand and research innovation on the other hand. The university is indeed humbled to be joined by an extraordinary community of scholars from various fields, a community that has kept faith with a transformational promise made in 1959 when the university was first established. The university remains committed to contributing to the greater good of South Africa through teaching, through research, through community engagement. Therefore, the many papers to be presented and deliberated upon today are consistent with the existential mission of this university to critically examine and interrogate challenges of the moment and the future and provide solutions to humanity through teaching, research, scholarship, and community engagement. I'm advised that about 350 papers will be presented today, and this is a testimony to the kind of contribution that your faculty is making to the advancement of knowledge in the country and the world. I'd also like to welcome all our academics present here today, both within UL and outside the university. We are proud of your scholarly and curiosity and accomplishments, and your unwavering commitment to the knowledge project of the university. You have devoted your time and talent to teaching, to mentoring, to research and scholarship in ways that continue to advance the University of Limpopo's promise, epitomized in our motto, finding solutions for Africa. You inspire, you spark and advance the curiosity amongst our students in their journey of becoming. Also to all our guests and partners from other universities and sister institutions in South Africa and other parts of the world, please feel welcome. Your scholarly and industry-specific insights and perspectives and contributions are highly valued, and we look forward to a strengthened relationship with your academic peers, researchers and students at the University of Limpopo. Please make use of this event as a great opportunity for you to forge mutually beneficial ties. I'd also like to welcome all our postgraduate students in attendance today, without whom this conference would not have been possible. The future of South Africa is in your hands. The university is immensely proud to be part of your life and academic journey. Please make use of every opportunity in the classroom, every opportunity in the laboratory, every opportunity that is presented to you through face-to-face or virtual meetings with your supervisors, conference, seminar, workshop, to enrich your research, talent, and ability. You all worked hard and made wise choices that enabled you to reach this point. And we are happy that you could move mountains in terms of the papers that we're presenting here today. And we hope and believe that you'll be able to build scholarship on the basis of what you're going to present in today, and be able to find your own voice as a student, as a researcher in the coming years. Program Director and Mr. Executive Dean, we hope and believe that the event will finally crack the proverbial code of integrating science and innovation and entrepreneurship in the university research. The themes comes at the right time, as all our universities across the world are grappling with this problem statement. As most of you would know, South Africa is a well-established national system of innovation that the government seeks to advance, which is meant to focus on leveraging public research and advance private sector capabilities for national benefit. There are a number of policy directives in this regard which you must be able to interrogate. The decadal plan from science and technology, which guide government research, development, and innovation efforts. There are agencies such as TIA, National Research Foundation, and others, which are also funding research in a particular way. It will be important for you to interrogate those policy directives and how those platforms could be used to support our students and our research broadly. Please use those opportunities, as many others, to close the chasm between basic research, innovation, and entrepreneurship, which is at the heart of your thematic statement today. Again, welcome to you all to the 15th Postgraduate Research Day of the University of Limpopo's Faculty of Science and Agriculture. We wish you the very best in your deliberations and hope that the many papers you'll be interrogating over a period of 4 days will translate into a body of knowledge that could only reaffirm the correctness of a view that science can advance both the human condition and development. In that way, ladies and gentlemen, science can truly and practically be of service to humanity and can also advance human civilization. I thank you, and I wish you everything of the best as you deliberate for the next 4 days on issues that are on your agenda. Thank you very much, and best wishes.
Thank you very much for that. Okay, thank you, Vice Chancellor and Principal Designate. I hope all of you are now feeling welcome. we now have to move to the next item, which is the item where we are going to listen to our very first keynote speaker lined up for today. And the way I'm going to do it, I'm going to introduce him by way of reading out his short biography. And ladies and gentlemen, today we have in our midst, though visually, we have Professor Mutlalipula B. Matsavisa from the University of the Free State. Professor Matsavisa, welcome. We can already see you. So I'm going to take this moment to read out your short bio. Professor Matsavisa holds a PhD in pharmacology. He is the director of the Indigenous Knowledge Systems Lead Program at the University of the Free State, where he also heads the African Medicines Innovation and Technology Development Platform. He is a guest professor at Beijing University of Chinese Medicine and a member of the University of the Free State Senate. Professor Matsavisa serves on the South African Health Product Regulation Authority's Complementary Medicines Committee as the vice chair and also leads its expert group. Prof. Matsavisa also chairs the WHO Afro-Regional Expert Committee on Traditional Medicines for COVID-19 and advises the Administerial Advisory Committee on Cannabis. He contributes to SADC, the South African Bureau of Standards, ASO, which is the African Organization for Standards, on herbal medicine standards, including a joint long COVID-19 project with Beijing University. Prof. Matsavisa has already made a huge mark as far as supervision of postgraduate students goes, and here we're talking about 40 honors, 30 MScs, and 10 PhDs. He has already supervised 18 postdoctoral fellows and published widely in natural herbal products and has secured more than 150 million rands in research funding, including the 58 million rands for a CGMP herbal medicine factory. The herbal medicine factory that he is developing. He is also developing an integrated health facility combining traditional and modern medicine. Prof. Matsavisa is internationally recognized. He has delivered invited lectures worldwide and continues to advance research, policy, and innovation in African traditional medicine. So the title of his presentation goes From Grassroots Knowledge to Application Through Science, Traditional Medicine Product Development. Let's give him a round of applause as I provide him with the platform so that he can present.
To be with you, but now I had a conflicting, aspect because as the deputy president of the South African, Society for Basic and Clinical Pharmacology, I had to come, I think, you know, to the conference in Durban. But I will be with you, I think, in spirit, but I also promise that I think one day I will come and visit. I also want just to really, I think, you know, my gratitude, I think, you know, to the university management, you know, the VC, you know, say the same thing, I think, you know, to the Dean of, I think, you know, the Faculty of Agriculture, Sciences, and also I think all the directors and senior management of the university. And then two, I think my I think, you know, speakers that would be also, I think, taking through the program, and I think the students and everybody in the room. Thank you so much. What I want to do today is just in the next, I think, you know, 35 minutes, 40 minutes, take you through a journey on how can we, I think, you know, as a country, as a continent, take our own initiatives, you know, to do two things. One, research that is applied, research that does have an impact, I think, to society, as well as, I think, research that could contribute to the formal economies. And I will do it through the title of looking at grassroots. But I took also, I think, the opportunity to begin to look at traditional medicines, because I strongly believe that traditional medicines spans across all the disciplines, whether it is science, chemistry, physics, pharmacology, medicine. You know, you would then, I think, have an expertise, you know, to work through drug discovery, drug development. So without any wasting more time, I would like also, I think, to share with you how I would want to do that. One, I think, you know, just to talk about, I think, you know, one or two slides, you know, where I come from. And then secondly, give you a justification. Why have I chosen traditional medicines? And then I want them to take 2 or 3 examples of the work that I think is being done, I think, in the program that is applied research that does then, I think, create impact, and it goes into industrialization. I lead, as I was saying, I think a platform. This is a platform supported by the Technology Innovation Agency, African Medicines Innovations and Technologies Development. And we are within the School of Medicine in the Faculty of Health Sciences. And really, to be with you, I think, on this event, I would have loved to be there in person and spend time and interact with the students. That's really my passion. But I just want to then say, who are we? And then I'll talk about the importance of traditional medicines, and then give those 4 examples, and then say, why is science research important in policy making? And then I'll then conclude and give you some take-home messages. I hope to do that, I think, in the next 35 minutes. The picture you see there, that's the picture in our laboratories, I think, with our honors students. I come from that, I think, you know, unit. That's our building, the Indigenous Knowledge Systems, you know, Center, where we do pharmacology of traditional medicines. And those are our labs. We can do anything, I think, from cell culture, 3D cell culture, isolations, formulation work, and then eventually, I think, product development that gets into clinical trials. And that's, I think, the journey that I would want you to take through. It's exciting, it's beautiful. And then I think, you know, on the top, you know, right, I think we host a lot of people. I think that picture, it was one of our pharmacists hosting people from Africa Centers for Disease Control. At the bottom, I think those are our cell culture. We can do 3D cell culture and create up some organelles. I think that's fair enough, good enough for marketing, but what then do we do? In research, we always want to strive to be different. We strive to innovate. We strive to lead. But nationally, I think we address research priorities, and these are disease research priorities for the nation, and also how we contribute to the region of Africa. But we also address community research needs and aspirations, and that's what we're talking about, these grassroots innovations. And then we look at how do we respond to industry research questions. So we do a lot of contract research. I'll give you some examples, and then contribution to the development of an African traditional medicines industry. We are sort of heading this team that, Professor Mandiwa was talking about. How do we begin to review our Medicines Act, and how then do we propose a regulatory framework for products based on traditional medicines to be commercialized? So we do responsive research. We are looking at old diseases like malaria. We're looking at new diseases, COVID, or the transformed streptococcal diseases. These are the diseases that you'll find that the bacteria eats up your face, and there's no cure for that. We look at neglected diseases, your Chagas disease, your sleeping sickness, and then we also look at the pandemics. We have been involved in the COVID-19 and also running some clinical trials on traditional medicines for COVID-19. So we are here also to say we are here to establish, I think, smart and strategic partnerships, but also I think these partnerships must be functional. They must not be sitting somewhere in the drawers. We look at community grassroots, innovations. What do communities do? So we work based on that, and then we look at basic research, trying to understand the safety, efficacy, and improved quality of these medicines, as well as your drug interaction studies. Then we will do quality controls, because we always strive that we need to produce quality medicines that are best available at any given time. And then we also do formulation studies, because we have pharmacists, I think, in the group, and then we can do our own product development and piloting to clinical trials, and then ultimately commercialization. So we have postgraduate students. I think every year we get applications of about 300, honors students that wish to join our program. We can't do that. And one of the things that we are thinking that probably we could have joint, I think, degrees, where we could have partnerships, I think, with universities, especially we're more looking at some universities where there are no medical schools. Then we can actually have these students joining jointly, I think, with our degrees, and then we can have joint lectures. But we do work with traditional health practitioners and knowledge holders and communities, and these are our living laboratories. These are our partners, as well as working with industry. So what we do, we need to actually do research that is in compliance with the regulations. And I will not go through that, I'll give examples, but I think, we sign collaboration agreements with communities. We have developed research principles and guidelines of how to work with communities, and those guidelines have now been adopted by the World Health Organization. And then we also do tailor-made trainings for communities and traditional healers, because these are our, I think, living laboratories. And then we have developed biocultural protocols. What should students do when they approach communities? So why traditional medicine? And I'm saying this simply because the whole world, is now moving towards traditional medicines. Whether we think about the United Nations, it does talk about traditional medicines. The World Intellectual Property is very strong, as a supporter of indigenous knowledge systems on intellectual property. The World Health, is... Yes, ma'am.
I'm sorry for the interruption. Could you please share the slides?
Sorry, my apologies if I have not done so.
No
I thought
problem.
I did. Okay, my sincerest apologies. I really apologize for that. I think I started, I won't be long. I started literally just sharing with you where I come from. This is, I think, what I wanted to talk to you about. And then these are the facilities. These are our facilities. I mentioned that we can do a whole lot of other work. Our facilities that in Indigenous Knowledge Systems, I think, Innovation Center. And then we can do anything from cell culture, 3D cell culture, formulation, and product development. And then we talked about, the pharmacists, who are doing formulation work, visit by the Africa Center for Disease Control. And this laboratory specifically does work on the pharmacology of traditional medicines. And I really apologize. And then I talked about how we want, to innovate what we do, community research, addressing national research priorities, and then also
Okay, just put it on presentation mode, slideshow. So the little tab down there.
So it's is it now on the presentation mode?
Okay, thank you.
Okay. So we train postgraduate students, and then we start, I think, our program from honors, up to masters and postdoctoral fellows. And then I talked about regulatory compliance, that when we do what we do, there needs to be regulatory.
O kana ke usofo ukhathi into sana. Tutsonto ikela sina khona go tla ba re ke itumela ka moso. Ke lo fodela yona tse.
we ready? Are we ready? Okay. Prof, can you hear me? Okay, we are ready now. We will continue. No. Okay, thank you very much for your patience and apologies for this interruption. So we are continuing. Thank you. We cannot hear you, Prof.
continue. Sandy, can you hear me now? Okay, I don't know what's happening. Let me share again because it gets stuck. I don't know. I can't seem to present. The whole thing is stuck. Is there no way of doing this either on Teams instead of Google Meet? I don't know because there are other people that are trying to join, but it's stuck.
Please just
And
make
yeah, because I don't know what's happening. It's it's stuck. Let me try and email the slides. Yeah, Prof, I don't know. It's stuck. It's stuck on my side, but I'm trying to email you the slides. Yeah. Okay. Chairman Prof.
come and control this here because I don't want to be. So it's fine.
Talk to you.
Okay, we are ready now.
Presentation.
Here.
Okay, too sharp.
Yeah.
You can tell him to speak.
Eh?
You can tell him to speak. Then after you close the mic.
Okay, I will have to close the mic as he speaks.
When he's speaking.
Okay. Okay. Thank you for your patience once more, and we are ready to go. Prof. Matsavisa, you can then begin with the presentation. Thank you.
Hey, thank you so much, Prof. And I really apologize for that hiccup. But can they put it into a slideshow? It's not possible. That's fine. I can proceed. Can you hear me? So, can you hear me? I don't know, Prof, what's happening because it looks like you can't hear me. Can you hear me?
We can hear you.
That is fine. I was just asking to put it in a slideshow format.
It is already.
Oh. Thank you so much once again, and I also apologize for the hiccup. But where I come from, it's from the African Medicines Innovations and Technologies Development, which is a platform of TIA, and we are specifically working on African traditional medicines and the pharmacology of that, doing applied research on the medicines development. Can I have the next, I think, slide, please? So I'm not so sure if it's moving, but I think what I wanted then to share with you, it's not moving on my side. It's literally who we are, and then I wanted to justify why it is important. I think, for us to do traditional medicines, important, because traditional medicines research spans all the different disciplines, whether you are in chemistry, agriculture, pharmacology, or health sciences. But also, I think the world is now moving into recognizing traditional medicines. We are now late talking about One Health, where we speak about patients having the choice, of their, healthcare services. Can I move into the next slide? And that's a slide on the facilities. And if that's on that slide, I think I just wanted to show that our facilities development through to clinical trials, phase 1, phase 2, phase 3. So this is I think, what I would want then to share with you. How then within that, I think, scope, do we work with communities to do applied research? The next slide just shows our cell culture facilities where we can do 2D cell culture, 3D cell cultures. We can grow little organelles. If you want to grow, say, for instance, a pancreatic, cancer, you can actually grow it as a 3D little model. It's easier that way because it also helps us, I think, do away with all the animal ethics. Can I go into this next slide? They are not moving on my side, so I'm literally not so sure where I am. Is there a way of moving the slides? Can we move the slides, please? they've been stuck, they've not moved at all. Okay, so thank you so much for that. So ideally, I think what we do, we look at national research priorities in terms of diseases, cancer, we look at, I think, diabetes, hypertension, Alzheimer's disease, but also we look at community research needs and aspirations. What do our traditional healers, I think, what do our communities require in terms of, the support that we can give them on their innovations and their ideas? And we work with industry to resolve industry challenges, whether it is in terms of product development, quality control, or formulation studies. But also we are now moving towards the development of a formal traditional medicines industry. We do responsive research. We address old diseases like malaria, as I said. We look at new diseases, your COVID-19, your, transformed streptococcal diseases, but as well as your neglected diseases. And then we look at pandemics. We establish, I think, partnerships. These should be smart, strategic, partnerships. And we really believe in functional, partnerships. And I'll talk about that later on. Can we go to the next slide? This has not moved.
Let's wait.
Okay. I think I sent that as a PDF, so you have to change it manually. Yes, hello.
Are you able to see the slides when they move
No, nothing is being shared now.
Okay, just wait.
Okay. Or is there a way of doing it in Teams, not in Google, in Google Meet? Yeah. Yes. Okay. Okay. All right. Now let's do that.
We can continue.
Thank you so much. Let me continue. And once again, I think I really apologize, but it has not changed anything. So if I were to quickly, I think, run through and start from the beginning. We are at UFS, School of Medicine, and we mainly do, I think, pharmacology of traditional medicines. We have, I think, the facilities to do that, which is, I think, the slides on the facilities. We have the facilities to do that. And those facilities, we can do anything from research into, I think, the mechanism, looking at safety, efficacy, as well as, our formulation studies, to running clinical trials and making up pilot products. The facility is a GLP facility. We are also now getting into the ISO 17095, accreditation with SANAS, because we do a lot of research for the pharmaceutical industry. On slide number 6, this is where I am. I think we work with communities, we work with the pharmaceutical industry, and we address national research priorities, and in terms of diseases, those being diabetes, hypertension, we look at cancer, we look at Alzheimer's disease, and malaria. In slide number 7, I think we're talking about, our capabilities and our competencies. In terms of competency, we establish and we maintain those functional, partnerships, and these partnerships should be strategic, and they should be smart partnerships. But we do what we do because we have a strong collaborations and partnerships with communities at grassroots level. We put value into their ideas and their innovations. We work with, I think, entrepreneurs to do basic research in terms of safety and toxicology, in terms of the efficacy, I think, and the claims that they put to us, and we understand the mechanism. How do these traditional medicines work? We do quality control studies, stability studies. This we also do for industry, and then we do formulation research and development studies into prototypes, and we do pilot production. We have the equipment to make up tablets, capsules, tea bags, and we can also do, a number of other creams and formulations. And then we run clinical trials, through pharmacies next to our building. So that's, I think, our capabilities in terms of traditional medicines research. And then slide number 8, if we move to slide number 8, that's, I think, where we look at training postgraduate students from honors level. Undergraduate, in terms of pharmacology, we do it for nurses and medical doctors. But anyone that has not done, I think, pharmacology and wants to join pharmacology with a B degree, then starts their honors degree with us. We have postdoctoral fellows, and then we work and teach traditional health practitioners in a number of, I think, activities, including research collaborations, aspect of IP and hygiene. And then we work with communities. I mentioned that I think one of the things when we work with traditional medicines, I think there are rules and regulations that one needs to apply. And we have been leading in terms of, establishing up research collaboration agreements. Whatever we do with communities, it's based on the research agreement that we entered into. But also we have developed the research principles and guidelines that helps us to work with communities. And these guidelines are the guidelines that we actually cannot, I think, deviate from. Any partner that wants to work with us needs then to be bound by those research guidelines. We have training programs that we offer for traditional healers and communities. And these workshops are run through entrepreneurship. They run through, I think, agriculture, because we will need to create the biomass for the products that we want to do. Biocultural protocols, these really relate also in terms of how students should actually behave, how we should actually behave when we get to communities, what are the taboos, what are the things that we need to look at. And then we have a documentation and a record keeping, I think, program where we train traditional healers how to document, their work. And then we said, why traditional medicines? That's the next slide. Why traditional medicines? And I said, it is simply because the world is now recognizing the value and the importance of traditional medicines. From as, high as the United Nations, through its, I think, organs, such as your World Intellectual Property, the World Health Organizations, and the World Trade Organizations. All of them are now pushing for traditional medicines. If you look at, I think, the World Trade Organizations in terms of the discussions around, I think, trade, what is important is also that traditional medicines is part, I think, of those discussions in terms of, I think, your technical barriers to trade. There's a committee that's looking on that, but also the trade-related intellectual property agreement that also governs international trade, in products includes traditional medicines. World Health Organization, in fact, has just issued out a new strategy, the WHO strategy on traditional medicines from 2025 to 2034 that talks about, I think, how then do we begin as countries to implement our integrated, I think, health, where traditional medicines is central. It talks about access and benefit sharing to communities. The World Intellectual Property is very also strong in terms of what it does, in traditional, I think, medicines. But also, I think what we know is, if you look at, I think, in terms of, I think, caregiving, health provision, you see that many countries, including South Africa, there are more traditional healers than, I think, medical doctors. And what that slide shows, it just shows, say, for instance, if you are in Swaziland and you want to go and see a medical doctor, you have a 1 in 10,000, chance that you would be, I think, seeing that doctor. So there's one medical doctor to about 10,000 patients, while in fact there would be one traditional healer to about 100 patients. So that means we can actually utilize the power and the numbers of traditional healers into our health system. This is not just about Africa. You look at the United States, you look at, I think, France, in the US, about 40% of the people do use traditional medicines. And then France, you are looking at around 70% of the people, while Germany, it will be around 80% of the people that are using traditional medicines. So this is not just an African issue. It is a global, I think, interest in the use of traditional medicines. The next slide, where we talk about the value and the knowledge of traditional medicines, we would want to say today to all of you and the students that, for instance, 30% of all the prescription medicines, whether you can go to any clinic or any hospital or any pharmacy, 3 out of 10 of the medicines that you'll find in there, they actually derived from plants and natural resources. But the second thing that we also know that we need to share with everybody is that medicines that are derived from plants, they treat 90% of all known human diseases. think about it, if you have diabetes, we give you that little pump. It's called salbutamol. Salbutamol is made out of a plant, and that plant contains a molecule called theophylline. It is from the theobroma tree. Whether you look at cancer, vincristine, vinblastine, Pacific U, that gives you Taxol, which is Plaxil for breast cancer. All those come from plants, as well as your heart failure drugs or the drugs that we use for, your organ transplantation. They all come from plants. So 90% of all cancer medicines are derived from plants. That then shows why it is important that we continue our research in traditional medicines. At the bottom, if you look at, I think, the market value, the MRFR analysis has now predicted that up, say, to 2034, the global market size of herbal medicine, about, I think in 2024, it was 100, million US dollars. And that is now projected to be 350 billion US dollars by 2034, which is a growth of about 14%. And this could actually change the lives, I think, of people. So I'll give you a quick snippet. I think, the next slide, that's some commercial prescription medicines. I don't know if you are on the same slide. Here, I just wanted to show you some of the medicines that whether you go and you look at Disprin, you send someone to go and buy Disprin. Disprin comes from the wild willow. I don't know what you call it. Kimuduani kasisutu. Metformin for your diabetes, it comes from a plant. And then we have Tamiflu. Tamiflu was used for, I think, the avian, influenza H1N1, and it comes from the Chinese herb spice. What else? You look at, digoxin for your heart failure. That's the next slide. That comes, I think, from a plant. And then those people that have got asthma, the inhaler that people use has a molecule called theophylline. It comes from a tree called the theobroma tree. And then we talk about, I think, cyclosporin. This is for your heart transplant, your kidney transplant. So people that go and have those transplants, you remove your heart so that your body does not, I think, reject it. We give you cyclosporin so that then your body doesn't reject. It is an immune suppressant. A lot of you normally talk about immune suppressant, but there are many, examples that I would want to share with you. And then we have artemisinin, we know about it. And then also, I think we have a whole lot of cancer drugs. 90% of all the cancer drugs that we use today, they do come from plants. And then luckily, and back at home, if you go now and you go to either Pick and Pay or you go to Clicks or you go to Mediclinic, you go and find a drug called Lintagon. If you know what Lintagon is, that's for your flus. That actually comes from a South African plant that's being collected, I think, in the Eastern Cape. So that's what we are saying, that we then need to have, I think, our traditional medicines taken seriously. We need to move into, the research into traditional medicines. And then the next slide, I think now that also then justifies why do we need to work with communities. This is a slide on the Convention on Biological Diversity. Many countries, signed, I think that, CBD. It talks about the indigenous people's rights. It talks about the conservation of our natural resources, but also more importantly, Article 8J of that CBD then talks about the sovereignty. And it talks about the benefits that are derived out of the knowledge, of indigenous peoples, and which therefore brings about the aspect of the benefit sharing. And then I think the next slide, it talks about the CBD protocols. I hope you are still together with me. So the CBD has got two protocols. One, it is the Nagoya Protocol. The second one, it is the Cartagena Protocol. The Nagoya protocols, it talks mainly about fair and equitable sharing of benefits arising from the utilization of natural resources, including plants. And this is a WIPO, product. And then the Cartagena, it's more about the biosafety. So many countries, if we go to the next slide, in terms of, I think they are talked about 8J, we can move. If we go to the next slide about parties to the CBD, I hope you can see that slide. I just wanted to share with you that there are only 2 countries in the whole world that have not ratified or that have not signed, the CBD. It is the United States of America, and I think one country, it is Sudan in Africa. The reason why the United States has not signed that, CBD agreement, it's simply because of Article 8J on the fair sharing of the benefits that are derived from the utilization of natural resources. That's the reason why United States has not signed that. We do not know why Sudan has not signed it. Probably it is because they do not have time. It is because of the conflict that is actually happening there. But all the countries in the world are signatories to the CBD. What is happening back at home? So if we go to the local situation, back at home, we have the National Environment Management Biodiversity Act. We call it the NEMBA Act. Chapter 6 of that act also then tries to then fulfills the mandate of the CBD. It fulfills the mandate of the Nagoya Protocol by saying, if you go into a village in Limpopo or somewhere, or you speak to a traditional healer, there must be free prior informed consent. And also you must then have a benefit sharing model in place, for the utilization of that, I think, knowledge. And that comes from chapter 6 of the NEMBA Act. I will not go through that. And then I think if we can then move a few slides, because I'm just cognizant of the time. And then there is a slide that shows South Africa. And what I want to share with you in terms of South Africa, remember I talked about other countries where there are more traditional healers than Western trained doctors. We have exactly the same, I think, situation. Even in the most modern health facility, you still see people using traditional healers, simply because for one traditional healer, there are 500 people in the population. For one medical doctor in South Africa, there are about 40,000 people waiting in a queue, to see that one person. And that explains the status of our public health facilities. So we believe that we have the science, I think we have the plans in this country, and we have the political will. That science then should add value to that knowledge. And we should then move away from talking about many plants, talking about many traditional healers, but we should talk about now, how do we begin to move into a knowledge generation, where we can then use science, to stimulate our economy. The next slide is about the stakeholders, that not even one single person, not even one single institution can develop a drug on their own. Drug discovery and drug development, it is a collaborative effect. It is a multi-institutional effect. You need researchers from different expertise, you need marketing people, you need policy making people, you need the regulators to do that. And therefore that is important. And what then I'm moving at then is, we must then begin, I think, we see how the geopolitics are moving into globally, begin to do needs-driven research. So the research that we do must have an impact. And I normally put it up and say, we should have what we call a grip. GRIP, in my explanation, which is something that I have coined, is getting research results translated into products, patents, policy, and public health. The research that we do should produce products. The research that we do should produce patents. The research that we do should influence policy. And the research that we do should be put into public health. And that research must then also change the quality of life of people. It must be needs driven. That's what this country and this continent must do. That type of doing the pie in the sky research is gone because many countries and many multinationals are hogging up their innovations. So how then do we approach the next slide in drug development? How then do we approach making drugs, from plants and the knowledge from communities and traditional healers? One, we could do what chemistry people like doing. You find a molecule, which we call it a new chemical entity, the NCE in that slide. It's a new chemical entity, but that is expensive, that takes long, and we may not have the funds to do that. But the next slide then would be, let's just standardize the herbal medicines and we make a herbal formulation, and that's easier for us to do. We have neglected looking at developing vaccines from plants. Many plants, can be used to develop vaccines. The tobacco mosaic, vaccines, that have been used in a number of, viral infections. They come from plants. So we should begin to think about that. But also more importantly, there is what we call adjuvants, a combination of plant extracts with prescription treatments, make the prescription treatment work better. Let me give a simple example that sometimes when people go to a doctor and the doctor know that when we give patients this drug, this is how they should respond. But you'll see there are certain people that respond miraculously. They recover faster. And they do that simply because they do not tell the doctors that they have been taking their prescription medicine together with traditional medicine. If you take aloe, aloe has got a polysaccharide called ace-mannan. And then ace-mannan, if it's combined with AZT for HIV, it actually makes AZT works 3 times better. And that's, I think, what we could save our governments in terms of, combination therapy. And that's where the world is moving in terms of what we do. The next slide is just a pipeline of drug discovery. I will not go through it, but what is important for us is the research starts with traditional healers right at the bottom, where the knowledge, the ethnomedical, and ethnopharmacological activities starts there. Because we know that if we randomly go and select plants, the chance of us getting a hit are very low. And therefore we rely on traditional, healers and communities that have experimented on the use of these plants for many, many years. So we just need a science, whether we are in chemistry, whether we are in pharmacology, to begin to add value to that knowledge. And then the other slide, I'll skip it because I've taken up this slide. I've borrowed this slide, I think, from our presentations in SAPRA, where we say, what is the pipeline? That SAPRA follows, and that's the same pipeline to say you need to do preclinical research, work in the laboratory, give us some evidence, whether it is in vitro, whether it is in an animal model, and then from there you go into clinical trials, phase 1, phase 2, or phase 3, and then we get approval. And then we go and get the drug onto the market. That's a process that scientists needs to help SAPRA, in terms of generating the information and generating the knowledge to get our medicines into our shops, formally. Again, the next slide, I think about the herbal medicine commercialization value chains. I'll not talk about that because all I was saying here is there are many steps that needs to be, I think, put together in commercializing our traditional medicines. One, we need to look at the raw material procurement, grow it. People in agriculture come help us so that then we can grow, I think, these plants and standardize them and make sure that the quality, is good. But also more importantly, that there will be constant supply, of your raw materials. Other than that, don't put anything onto the market if it is there today and tomorrow people don't find it, because customers are quick to go and find substitutes. If I go to a shop, I buy an Albany bread. Tomorrow I go, there's no Albany bread. The next day I go, there's no Albany bread. I will not go and starve. I'll go and find a substitute, and move on in my life. And once you find a substitute, the risk is the people have, I think, that now trust and faith in that, branding, and they will continue with it. So it's very important that we have the right raw materials. So this is what we do. This is our research. I hope you are still with me. These are the products, excuse me. These are the products that we have developed, ourselves, and some of these are now onto the market. Now you look at, we develop medicines. These are all plant-based medicines. They are not, chemically based. They are plant-based. We have done research, safety studies, clinical trials, and then formulation. So if you look at, I think, the Pela plant, this is a mixture of 4 plants. We need agriculture right now. In fact, that's where we are. We have just signed up with Sanbi to begin to help us grow these plants. So people who have got the agricultural expertise, I'm in pharmacology, I can just grow things for me, but we need massive agricultural pilots to begin to commercially grow these plants so that we can then supply South Africa and also supply the world. So we created up that, medicine, and it was for HIV. We used it for COVID. We had a clinical trial for COVID, but we have now formulated the same medicine as a throat spray for people that cannot swallow. So it is exactly the same medicine, but now it's in a throat spray. If you have a sore throat, we can then quickly give you that. Health beverages, those teas, if you go to Super Spa, you will find those teas. This is a joint work that we have done with communities in the Northwest, with communities in the Eastern Cape. I know that, for instance, there is now a community in Limpopo. Probably I'll come to University of Limpopo and say, let's work together to help this community within your vicinity that have also said to us, we have our indigenous tea. Can you help us, to develop those teas? And together with, I think, one company that then came to us and said, can you take these teas and make them into iced teas? And that's what we have now done. In fact, this week, we are exhibiting the same products at the G20, in Pretoria at the CSIR to showcase what we can do with our indigenous knowledge and with our medicinal plants. I'm not so sure whether this will show because probably you have a PDF, but also we have been working and assisting industry. We have been assisting Tiger Brands, one, to reformulate umuti wenyoni. That is our product. It's now being sold. You can find it, I think, in the shops. Oh, mama, they know that when you have little babies, you'd go and buy umuti wenyoni. That came from our hands. We know about gripe water. Again, I think Telement gripe water, we reformulated that. It's being sold as a pediatric gripe water by Tiger Brands. That is a product that we had reformulated. And we are now busy with another product, on peaceful sleep. And that's also an industry product where we're helping the industry to reformulate and make those products better. And then the next slide is really about compliance. I will not talk about it, that we then need to comply with the regulations, the rules. I will not talk about it. Let's move to the next slide. The next slide is about, the slide about people sitting around a tree. The message here, we were in the Northwest talking to the Tribal Council, planning inclusively how we're going to start the project on the teas. And this is what we really refer to as inclusive research. We need to work with communities. We need to take them all away along with us so that they understand and we can build trust. That is important. The next slides, I think we were in the Eastern Cape where we're putting infrastructure, at the community level. We're discussing where to put in the infrastructure, what needs to be put in place. And you could see those were different, I think, communities. I hope you are still with me. And then if we go to the next slide, I think, I saw Professor Numalo could be around. These are what the chemistry likes. We have been working on this plant. It is a South African plant, but we have isolated novel molecules. And active pharmaceutical ingredients, and most of those would come, I think, from plants. But also there is a need for us to start seriously considering directed research, research that has got an impact, and research that would actually have, an impact on the social activities, of people. Where an ordinary person somewhere in Limpopo could walk into the street and say, I now appreciate what the University of Limpopo has done, on my quality of life. That's what we need to be doing, I think, as South Africans. But also, lastly, I think, is how then do we help the regulators? How do we help SAPRA? We can help SAPRA through generating a lot of research, a lot of research that would then influence policy. We are working on cannabis at the moment, and we are now beginning to influence policy that cannabis is not as bad, as I think everybody thinks. We have now developed, I think, hydrogels for pain. We have developed anti-inflammatory products from cannabis. We've developed diabetes products from cannabis. So it is now to say, let us not bedevil the plant, but let us look at the users of those plants. But I think with those are few words. Thank you so much. I think despite, all these challenges, Professor Mandiwa, I want to make a commitment that I would come to Limpopo at my own expense. I would come and talk and give this talk one day, so that then we can have a greater discussion and a longer discussion. Despite all this, I still, am optimistic that we could find a way to resolve it, so that we don't have all these challenges. I take the blame because I should have been there. But as I said, I really appreciate the time that I was, with the team. I could not see all of you, but one day I'll come and see you. And then I also want to make just one, announcement that next year, we are hosting the International, Pharmacology Society. There's going to be a very strong slant, on applied research, health, and traditional medicines. So you'd see that in my last slides, I've talked about it. So I would want already to extend an invitation that please join us, at the University of the Free State when we do our international, 60th, 59th, South African Society of Basic and Clinical Pharmacology conference. Thank you so much, Prof. And thank you so much to everybody. And it was really great to be here. Thank you so much.
Thank you, Prof. Matsavisa. We also would like to thank you for your patience, and also on behalf of the faculty to apologize for these interruptions. But we are happy. I think it's not just me. I would imagine that the message that you tried to relay with the presentation has actually gone through to the audience. And we will, of course, open a session just for a few minutes so that we can have some engagements with you before you leave the platform. But I just want to say that from my side, I have the take-home message. And from what I could get was the fact that the research that we do should actually give us products. And the research that we do should allow us to patent. And we should also be able to assist the society to address its challenges and also solve its problems. So that is the take-home message I got. So we will then open this session for the question and answers, comments, and all of that. So we do have the microphones that are roaming, so please, you raise your hand if you want to engage Prof. Matsavisa. Thank you.
Oh, thank you.
Okay, just wait. Can you hear him, Prof. Matsavisa?
Yes, I can hear him.
Okay.
Okay. So, can it be possible for me to be co-supervised by him? I mean,
Did you hear that, Prof. Matsavisa?
I heard that. I thought maybe we'll take a few questions, but yeah, definitely. We can do that. I have a lot of master's students that are graduating that come from Limpopo. They joined me, when they were doing their honors. So definitely, I would be keen, to help you co-supervise. I think one of the things I hope she said she was going to be in the meeting, Professor Rangwana Agnes. That was my student. So I actually supervised her. So that would be great for me to work with someone at the University of Limpopo.
Thank you so much, Prof. I just wanted to ask if you could comment on how medicinal plant studies can be standardized in universities, because most of us, with the research that we do, especially after saying that most medicinal plants or herbal traditional medicines are taken concurrently with prescription drugs. So most of the studies that we do that I've seen don't usually focus on herb or herb drug interactions. So do you comment on how maybe we can standardize research in universities so that it includes this, and then that way it could possibly be easier to translate into clinical trials and so forth? Thank you.
Did you hear the question? We can't hear you.
Can you rephrase it?
Okay, Prof. In short,
There's an echo.
okay, Prof. My question. Okay, Prof. My question in short was just to say that, can you comment on how we can standardize research in universities that works on herbal traditional medicines, especially when you said that traditional medicines are usually taken concurrently with prescription drugs, and most of the studies that happen don't usually focus on herb or herb drug interactions. So can you kindly comment on possibility of standardization of these studies?
Let me just catch it up there. I mean, there are two words that I had: drug interactions and then standardization. Indeed, it is important that we do drug interactions. So one of the things that we do to address that, we look at the cytochrome P450 work. We both do in vitro and in vivo, where we look at the effect of these extracts on 2 or 3 major drug metabolizing enzyme. I think it's a CYP3A4 and 2D6. Those are the major drug metabolizing enzymes, and we look and see if those herbs would interfere. Many of the drugs that gets taken off the market, they get taken off the market not because the drugs themselves are toxic. They get taken off the market because they interfere with the other drugs, and that's why toxicity comes. So it is important. So when we do toxicology, we do the cytochrome P450 work. a must that we should do. The second thing is how then do we standardize, I think, the extracts. You can literally start by saying the very first extract that you work off, the one that the healer gave you and the one that works. You create up, whether it is an HPLC, chromatogram, you look at, I think, the UV spectrum of representative, peaks, and then you look at the peak areas, and then you can set that one up as your base, I think, chromatogram, with that subsequent ones would follow that. Or you could also use basic things like, TLC. We do have a high performance TLC, fortunately, so we can do that. Or secondly, we use, I think, our high resolution, LCMS, where we can begin to get, I think, accurate masses. So once we have those accurate masses, from the very first extract, then we use that as our sort of baseline for subsequent, studies that will be following up. This is very important, I think, what you are asking in terms of your stability, of your product. That's another thing. We can do accelerated stability or we can do real-time stability. Fortunately, we have the equipments to do that, but we are open to assist anyone that wants, to do research, with us. We can avail our facilities for that. So we can do HPLC, we can do, LCMSs, we can do TLCs, as well as stability testing, of those extracts. Once then you have created up that chromatogram, you use it as your base, and everything else then must follow that one. And SAPRA, which is the regulator, does accept that, for standardization. I do not talk about, identifying chemical markers. Remember, when you talk about chemical markers, we don't even know which of those chemicals work. So therefore, chemical markers mean nothing when you have a total extract, because they may not be your biological or your pharmacological markers. Thank you so much.
Should just take one more question and then we close it.
All right. Good morning, Prof. I hope I'm audible. Yes. So my question is kind of like in line with the previous question, and I was quite fascinated on the moment you talked about adjuvant therapy, especially with herbal medicinal plants. So my question was, have you worked on any adjuvant interventions, especially in breast cancer? And if so, how is the future of adjuvant therapy in treating breast cancer? Thank you.
Thank you so much. We have an ongoing project where we are looking at a combination of one of our extracts with your Taxol. Taxol, I think it gets sold as a Paclitaxel. It gets sold as that trade name, and we're looking at those combinations, and we can see that, in fact, you can see potentiation. Now, what does that mean? You can see that at low concentrations, that the extract plus the drug, increases, the activity. But what we have conclusively done was on diabetes, and that's something that we have published, that a combination of some of the plants that are being used, Lipia javanica, with your metformin, we have now shown that drinking those teas could actually be beneficial to people that are having diabetes and they are on metformin, because they make metformin work better. They manage to stabilize the sugar level, and they keep the sugar level stable for a long time, while in fact the patient does not even get hungry. The same thing we have done, I think, on hypertension. We have shown that Buddleja saligna, again, it's being drank in the Eastern Cape as a tea, does actually help, people that have hypertension. We are now starting up an animal study that has just been approved by the Medical Research Council. We'll do it with them, where we now combine Lipia javanica, I think Kimusukujani, Lipia javanica with Budleja, and we are now going to do two things, on the rats. One, induce them, with type 2 diabetes, using high-fat diet. They become very obese, and then we give them lots of salt, and then they become hypertensive. And we are now going to treat them using a combination of these plants together with the treatment combination and see comparatively with the extracts alone and the extracts in combination. So we have done a lot of these combination studies, synergy studies. We just got one of, I think, the PhD students that also did that for breast cancer. I think that was what you were asking. And we have published that work where we also look at network pharmacology. How does this thing work at a molecular level? So we're beginning to understand, and this is a policy thing that once we are satisfied, we have published it, we can go back to government and say probably a combination of people drinking this traditional medicine would be beneficial to, I think, those patients. Remember the same thing I said, we have 600 patients that we are now going to be looking at. These 600 patients had COVID-19, but now they are having TB. And we are now taking a combination of TB treatment with our Pela traditional medicines and seeing what would then happen if you ask these people that are taking TB medication with Pela, will that, I think, improve the improvement rate, because we have shown in our experiments that Pela is an immune modulator. We've shown that it is antiviral for COVID-19. That's what we got approval for to do the clinical trial. But also we've shown that Pela does disrupt the biofilm formation. Your bacteria clumps up, they make up these colonies, and those colonies are the ones that are very virulent. One single bacteria is not toxic, but those bacteria, they need to clump up and make up colonies to become virulent. But if you have got a product that then disrupts the formation of those colonies, it could be a very good general bacterial agent. So these studies are very important for us because they help us understand why sometimes when you are in the lab, you are extracting, you are doing all these extracts and your results come up negative. While actually a traditional healer will be saying, but my patients are recovering. So we're beginning to apply appropriate research method to address, I think, what the traditional healers are saying. Thank you so much.
Let's give him another round of applause, please. Thank you very much, Prof. Matsavisa. We've come to the end of this session, but I also would want to let the audience know that you had to shift your next engagement without us asking you. So we really, really appreciate for that kindness. Thank you very much. The last thing is that this faculty of Science and Agriculture has actually prepared something in the form of a token of appreciation that we will send through to your place. So thank you very much, and this is really coming from the hearts of each and every one of us. Thank you. Okay, thank you. Do you have anything you would like to say, maybe?
No, I really want to say thank you so much from the bottom of my heart, and I wish you the best for the next, I think, 2 days. Thank you.
Thank you very much. It's a goodbye from us.
Okay, thank you, colleagues. Let us now move to the last item before we could split into our parallel sessions. This is the announcements that I would like to make. The first one is got to do with the halls. Okay, so the first one, it's got to do with the conference halls that we'll be using for our presentations. The School of Agricultural and Environmental Sciences, they are going to remain in this hall, which is Limpopo Hall. It's not Ivory Hall as indicated in the program. And then the School of Molecular and Life Sciences, they will go to Litava Hall, which is, you go out and you go towards that direction. And then we have the School of Mathematical and Computer Sciences. They are going to be in the same venue with the School of Physical and Mineral Sciences, and this is going to be Capricorn Hall, which is closer to Litava Hall. So these halls will be used by those who will be giving standard talks. Now coming to the honors presentations, the posters, the honors students from the School of Molecular and Life Sciences, you are going to be in Litava Hall, where the standard talks will be taking place as well. Then honor students from the School of Physical and Mineral Sciences, School of Mathematical and Computer Sciences, and the School of the School of Agricultural and Environmental Sciences, you will go to Mujaji Hall. That is where the poster presentations will be taking place. You go out here, Mujaji Hall is at the tail end of this hall. So you just go out and around. Then the next announcement is the fact that in the foyer of this hall, which is outside here, you may have seen that we do have stations that are set up by the companies that are here who are exhibiting their products and services. So please do visit these stations during breaks, engage them. The last one from me is the session chairs and adjudicators. As we move to the different halls, if you could quickly just assemble in this foyer out here, and then Prof. Masuku will be with you. Thank you very much. This is the end of these sessions, and let's all enjoy the balance of the day. Thank you.
presentation. Can we chair Dr. Simon Lashma as he comes to the stage? Over to you, Dr. Lashma.
Good morning, everyone. Can you see my slides? I can't. Is it showing? Okay, so good morning. As I was introduced, I'm Simon Lashma. I'm a researcher in animal breeding and genetics at the Agricultural Research Council's Animal Production campus in Irene, close to Pretoria. So just a bit of a background about me. I'm an animal scientist in training. So I did my undergrad and my honors in animal science and then moved on to do a master's degree in animal breeding and genetics. And then my PhD in animal science, which I did while I was doing the professional development program of the ARC as well. And then I graduated in 2020, which is not too long ago. So I was where some of you guys are now, not so long ago. So I'm still quite like early career, I'm not going to say young, but early career researcher. So the title of my talk is A Timeline of Genomics Research on Indigenous South African Ruminants. So we can't really speak about genomics if we don't first introduce animal breeding and genetics. So animal breeding and genetics is one of the pillars of animal science or livestock development. So alongside animal health, animal nutrition, and animal production and physiology, this is one of the key disciplines in animal science. It's a broad field ranging from characterization to conservation to genetic improvement. And it mainly has two branches, which include quantitative genetics and molecular genetics or genomics. So the distinction between these two branches is mainly on the information used to study the genetics, and for quantitative that would be phenotype or pedigree-based data, and for genomics that would be DNA level information. So early efforts in animal breeding and genetics research solely focused on phenotypic observations and selection of a desired trait. So animal breeding programs are based on the principle that phenotypic observations on related individuals can provide us with information about their underlying genotypic values. So there were some major advances in animal breeding technologies in the form of BLUP, which is a statistical model developed in the 1950s. And this is used to estimate breeding values and the genetic merit of animals. And this basically reflects the individual's genetic capacity in comparison with all other individuals within its population. So quantitative animal breeding and genetic methodologies relies on two main information sources, and this includes the pedigree and performance data. So we all know the popular formula, P equals G plus E. So with quantitative animal breeding and genetics, they sort of provided us with the first insight into the G, or the additive genetic component of this formula, through the use of, as I said, pedigrees and relatedness and actual per animal performance. So using quantitative data, we can perform several types of studies, which include characterization, that might include assessment of livestock populations and their production environments. So this would include population structure, generation interval of the animal, of the population, the variance components, etc. Also conservation, in terms of inbreeding levels and the effective population sizes of the breeds, and then also for improvement, which would include genetic evaluation and selecting genetically superior animals to be the next generation's parents. So we have several service providers in our country that is in charge of the genetic evaluation and animal recording, And this includes SA Studbook through its logic database, LRF through BreedPlan, and the ARC with their Intergist database. And then also this is obviously in collaboration with all of the breeder societies within the different ruminant species. So in quantitative genetics, obviously the breeders would set a breeding goal based upon certain desirable production traits for a specific breed, and then in each breeding generation, sires and dams are phenotypically scored, and then given a breeding value that is used to rank the animals in order of their production. So sires and dams with the most desirable physical characteristics, in other words, the highest breeding values, are then selected for breeding. Offspring from these matings are then evaluated according to the breeding goal, and those with highest breeding value scores are used as breeding stock for the next generation. However, there are certain limitations in terms of quantitative or phenotypic data, because even the best phenotypic assessment cannot ensure that the mating of two high breeding value animals will result in high breeding value offspring. And why is this? It's because genetics is a lot more complicated than just that we were taught Mendelian inheritance. There's a lot of randomness in genetics as well. There's also decreased selection efficiency for traits that are difficult to measure. There's a restriction to traits that can be reproducibly and inexpensively measured. Also, the genetic progress is sometimes slow, and high selection pressure is on key traits that may be harmful or negatively affect other important traits. Also, not to forget that many farmers, especially in the commercial and small-scale sectors, do not always record as they should, whether that be in quantity or quality. So there's a lot of incomplete data when we look at pedigrees and performance as well. So how can we bypass some of these limitations? Well, one way is to look at the animal's actual DNA, so at the genome level. So DNA was sequenced for the first time in 1977. So the genome of an animal refers to the entire set of DNA instructions found in a cell. So that would be approximately 3 billion base pairs. So different combinations of A, T, C, and G nucleotides. And genome sequencing basically provides us with smaller little pieces or fragments that we can build the genome with. So in terms of sequencing or capturing the genomes of animals, we started off with Sanger sequencing technologies, and then have moved on to next generation, which is more high throughput, and third generation, which is long read sequences. So obviously sequencing technologies has allowed us to assemble genomes, and then by reassembling all the reads to reconstruct the original genome sequence. And in 2001, the first draft human genome was published, and followed by 2006, the first bovine genome. So I'm going to go through this very quickly. So I just mentioned the first type of sequencing we had was Sanger sequencing. So that was limited to shorter reads that we could sequence. And in 2003 only, with the introduction of next generation sequencing, we could sequence up to 25 million base pairs in a single 4-hour run at 99% accuracy at less than 1/6 of the cost. So obviously, sequencing technologies and methodologies have become a lot more inexpensive and efficient and automated. So this allowed us to set up or sequence reference genomes for our different species. So currently there are reference genomes available for pigs, goats, cattle, chicken, and all of the major livestock species. And basically what this reference genome is it's a representative example of the set of genes in one idealized individual. and it's used as a guide on which new genomes can be built or compared to. So just there in the upper right-hand corner, just to give you an idea of how difficult it was to sequence the first genome. If you take 3.2 billion letters, that equates to about 6,680 PhD theses that needed to be actually captured or sequenced. So the technology has really come very far. So how do we use these reference genomes to develop other genomic platforms or genomic tools? So first of all would be through the discovery or identification of single nucleotide polymorphism markers. So these are basically single base pair changes across the genome. There are millions of them across the genome that can point us towards the differences between animals. So once these SNP markers are identified, they then have to be validated, obviously, in some of the world's most popular or global breeds. So usually when they validate these SNPs for inclusion on SNP genotyping panels, they would validate them in the most popular breeds globally. So then, after they've been validated, we then go on to commercially developing and releasing SNP genotyping panels or SNP chips for the different species. So how do we use them? So obviously we do on-farm sampling, whether that be tail, hair, or tissues from the ear or blood. We then do a DNA extraction of the biological sample, and then once the DNA is extracted, we load them onto the SNP chip, and then depending on what the density of the chip is, the animal is genotyped for, let's say, 50,000 SNP markers. So how we can use this is sort of similar to the applications of the quantitative data. It's also characterization type studies in terms of diversity parameters, genomic composition, within and between breed relationships, and also parentage verification. In terms of conservation, again, we can look at inbreeding and effective population size just more accurately. You might have heard of runs of homozygosity before. We can look at traits in terms of genome-wide association studies and selection signature identification, and then also look at genetic evaluation or animal improvement through genomic selection. So just for time, I want to skip this one. So in terms of South Africa's history or timeline with genomics, in the past, this included the validation of genotyping panels for locally important non-discovery breeds. So as I said, when they develop the SNP chips, they validate these chips in globally popular animals. But what about some of the more country-specific indigenous breeds, such as those that we have? So in terms of cattle, we have Sangha cattle, we have our indigenous goats, we have our fat-tailed sheep. So what about those animals? They aren't used for the validation of those genotyping panels, and it's not to say then that the genotyping panels themselves that are commercially available do actually have utility in our breeds. So our breeds, obviously, they have local importance, they display unique adaptability, and also some of their population sizes are decreasing, but they are the most important genetic resources for sustainability. Also, certain breeds that have other production purposes, for example, in the first goat genotyping panel, there were no fiber-producing goat breeds included in the validation, so we weren't certain whether there was utility of the goat genotyping panel, for example, the South African Angora goat, which we are the biggest exporter globally of, mohair. So this introduced what we call an ascertainment bias. So this just basically means that the most commercially available genotyping panels weren't necessarily as useful or optimally useful as they should have been for our animals as well. So one solution would be whole genome sequencing, which is more on a per animal basis, but we'll get to that later. So in terms of South African achievements, in 2013, the first genomic study was published on cattle by Olga Kwabe. She was also at the Agricultural Research Council's animal production. In 2015, myself and two of my profs at the University of Pretoria did a validation of the goat 50K SNP chip in South African Angoras. And in 2016, the 50K sheep panel was validated as well by a group at Stellenbosch. And with these validations, we also saw that the animals that weren't included in the validation process or the discovery process did have lower information or polymorphicity with these chips. That was followed by various South African genetic diversity studies on cattle, goats, and sheep. Some of the initial diversity studies, however, had the limitation of having very low sample sizes. So relatively recently, starting about 10 years ago, there were major improvements in the amount of genomic data. So in 2015, the Beef Genomics Project or Beef Genomics Program was started, and this was basically a multi-stakeholder collaboration, including universities, breeder societies, research councils, etc., to genotype more animals in the cattle or beef cattle industry to be able to eventually employ genomic selection. This was also followed by a dairy genomics project in 2016. So the first phase of the beef genomics project included about 14 breeder society covering taurine, Indocin, and Sangha cattle, and then the DGP included about 3 breeder societies, including the 3 major dairy breeds, Holstein, Jersey, and Ayrshire. So the aim, as I said, was obviously to establish reference populations for the major SA beef and dairy breeds, and the added bonus was increased numbers of genotyped animals that we could use for diversity studies, GWAS studies, etc. So more recently, the sheep industry also made an effort on increasing the number of genotyped animals, and currently the SA Merino is leading with about 1,400 plus animals. I think it's a lot more now. So this obviously allowed us to do larger scale genomic diversity studies, such as the one that we published in Frontiers, where we did a genetic diversity study of 8 cattle breeds and 8 sheep breeds, which also produced quite interesting results. In addition, as I said, this allowed for the application of genomic selection. So the first single-step GBLUP GEBVs were released, genomic EBVs were released for South African Bonsmara by SA Studbook, and this was followed by the Drakensberger, Beefmaster, Huguenot, and I think now more than 10 of the breeds actually are regularly or routinely receiving genomic estimated breeding values. So also dairy, in terms of Holstein, Jersey, and Ayrshire, they are also partaking in genomic selection. And the first sheep breed to receive GEBVs was the South African Merino. There's also been ARC-specific projects on genomic selection where GEBV accuracy has been tested in Afrikaner cattle and Brahman cattle as well. So just in terms of some of the ARC-based genomics project, we have the LEAP Agri or OptiBov project, which is a multi-country international collaboration aiming to characterize some of the local breeds per country. We also have a SADC-specific country, including Lesotho, Zimbabwe, and Botswana, where we're looking at certain cattle breeds. And then we have various heat stress focused projects, methane emission. We have a traceability study going on that aims to sort of help with stock theft. And then we also have some more welfare-focused genomics projects, including one on baldness or hornedness. So in terms of what the future will look like, so we're already employing a lot of short read sequencing technologies, such as Illumina, where we're doing SNP discovery of our local breeds, and more towards from 2020s onwards and towards the future, there will be an increase in long read sequencing technology applications such as PacBio and Oxford Nanopore. So long read sequencing as opposed to short read sequencing will just be longer fragments, which will ensure better assemblies and structural variant detection. So larger genomic variants than SNP markers. They are at the moment, however, more expensive and lower throughput, but they are improving. So in terms of what's next for South Africa, as I said, whole genome sequencing projects. So this will allow millions of SNPs instead of thousands that are included on SNP genotyping panels. And this will obviously allow us to more extensively characterize the genomes of our local breeds. This will allow high resolution analysis of diversity and finer mapping of genes, for example, that are related to adaptation, which will be important. The long read sequencing technologies may allow pangenomics and structural variation identification. Also, as I said, with genomic selection, we expect further reductions in genotyping costs, which will allow more data to be available and which will allow the implementation of genomic selection more routinely for more breeds, including some of the smaller composite breeds as well that are very important. Just important to note that phenotype is still king, so just because we have genomics doesn't mean we don't need phenotypes or animal recording anymore. It's now actually more important than ever, and it's still one of the most important data source that we have. Together with that, there has been a rise in phenomics and high throughput phenotyping as well, so that's a good thing. So just lastly, I wanted to say we need to bridge, also we've started, but we need to start bridging the gap between genomic technologies and the emerging and smallholder farm section. So the ARC does have a KYD program for cattle and the Pela Village program for goats, whereby they are helping the farmers to improve on their animal recording, etc. There are, as I mentioned as well, several DALROD projects focusing on stock theft and the genomic composition of nondescript cattle. So I do think we are making an effort in including the other sectors as well. Thank you, that's all.
Thank you very much, Dr. Lashma, for such a wonderful presentation. Can we give him a round of applause again? Dr. Lashma is our plenary speaker. I am going to give 10 minutes of interactions or questions with regard to what he was presenting or any other related question that relates to genomics and quantitative studies in animals. Can I get the hands? Can I get questions? I see a question here. Any other hand? we will encourage students to participate. Eh? Don't hold your question. Can we have you say that? Yes.
Thank you very much. Dr. Lashma, what a great presentation on quantitative genetics and molecular breeding in animals. Such good work being done by yourselves and other scientists at the ARC. Please enlighten me. Some achievements to date in terms of the breeds that could have been developed integrating phenotypic and genotypic data.
Some of the
achievements at the ARC.
Yeah.
Integrating both phenotypic and genotypic data that you have assembled. And secondly, with regards to GWAS, what are some of the traits that you have identified, and how will you use them to, or how are you using them to inform the breeding program?
Okay. So in terms of ARC, we have an Afrikaner herd on the farm. So at the moment they have what they call a grow safe system and a green feed system, which records methane and all of the other greenhouse gases. So the grow safe system as well does quite advanced feed intake recording and all of that. So that's a way of where we're busy generating more phenotypes. In terms of genotypes, we have a lot of collaborations with, for example, Sweden and the Netherlands, where we've sequenced Bonsmara, Afrikaner, and Hereford cattle. So we're going to use that data to look at whether the genomic composition of the composite is still on the DNA level as it was intended when they actually developed the composite. GWAS, we've identified a lot of genes. The problem is, and I know probably why you're asking that question, is because there's a lot of GWAS and selection signature studies being done at the moment, but they're not really being implemented. So we're identifying a lot of genes that are under selection or genes that are associated with phenotypes. The next step now for anyone in South Africa working on cattle or goats or sheep is to actually start implementing or developing chips or something where we can select for those SNPs. Yeah. Thanks. I think that's it.
Is there another hand? Yeah, Dr. Matapo. Maybe just before you ask your questions, let me find out from the next presenters if you have loaded your slides. If not, you can quickly come and see me. Dr. Matapo.
Okay, thanks, Chair, and also thank you, Dr. Lashma, for a good presentation. If you can check now, we do have the smaller holder farmers that are trying, and they are still on phenotypic direction of improving their production. So I want to check with you as the researchers, what is the plan for future in bridging that? Because this method, if you can check of genomics, it's very expensive towards the smaller holder farmers. So what are the future plans in assisting those farmers so that they can adopt this method of genomics to improve their production, since you indicated one of it, the accuracy as compared to the phenotypes, which is the quantitative genetics? Thank you.
Yeah. So I'm not that involved with that at the moment, but I know the focus at the moment is more on animal recording and phenotyping. And the animals that are being genotyped in the smallholder and communal sections, we are using those genotypes more to characterize them, so to see what the genomic composition is, what the level of inbreeding is, and all of that stuff. But we will first need to establish a very fixed and routine phenotyping system before we can actually go anywhere close to genomic selection. I think that's the priority at the moment. It's just helping them with the recording and breeding programs. I don't know if I'm answering your question. Okay.
Is there any other question? If no question, the last one will come from me. Dr. Lashma, I want you to share with the House, how is your research benefiting smallholder farmers, especially goat farmers, knowingly that overwhelming percentage is in the hands of these guys, and also taking into account the changing climate? How does that actually impair or increases the abnormality in animals? Thank you.
So in terms of how we are trying to help them is, as I said, we are doing more genomic composition studies at the moment, so to see whether those animals are more, let's say, taurine type or indocin type, and then to sort of enhance the way in which we advise them to select or include the animals in their breeding programs. What was the second part? I can't remember.
Can we give him another round of applause? Thank you very much for your presentation. Yeah, yeah. Our next presentation will be coming from our students. I could see some of the students were pointing at me. I think they were timing Dr. Lashma's presentation. I am the chair here, and please don't co-chair with me. We are going to continue with our presentation, and the next So our next presentation will be coming from Pasha Tobejane, and the presentation is titled "The Effect of Bechemia discolor on gastrointestinal nematode infection of South African nondescript goats." Can we chair Ms. Tobejane as she comes to deliver a presentation?
Good morning. My name is Tobejane Haipashia. I'll be presenting the slides titled "The Effect of Bechemia discolor live male monitoring on gastrointestinal nematode infection of South African nondescript goats," under the supervision of Prof. Bigunya My co-supervisor is Prof. T.G. Manyelo. Here is my outline. Okay. Introduction. Parasite restrict goat production, which raises mortality rate, lower fertility, blood sucking, and skin irritation. Main health problems in small ruminants are gastrointestinal parasites such as Haemophilus continuus and Fasciola hepatica. The use of the medicinal plant is the alternative measure to control the parasitic infections adopted by farmers. Becamia discala is a plant that is beneficial for the health of the goats. Researchers have done commendable work on discovering Becamia discala, which help with the use of medications for a variety of conditions, including lowering the wooden beam in goats. Hence, the study aimed at investigating the effect of Becamia discala live meal on gastrointestinal infections. The aim of the study was to investigate the effect of Becamia discala live meal on gastrointestinal nematode infection of South African nondescript goats. The objective was to determine the effect of inclusion level of Becamia discala live meal on gastrointestinal nematode infection of South African nondescript goats. The study was conducted at University of Limpopo in Malfarm, and a total of 16 goats was used and assigned to 4 dietary treatments. The table below shows how feed was formulated. Fecal samples were collected at the end of the feeding trial, and they were freshly collected directly from the rectum. After they were collected, they were transported and promptly analyzed them for fecal egg count and fecal oxide count. The results show that supplementing nondescript goats with Becamia discala influenced strongyloids, oxide, and trichuris. And trichuris was only found in all of the dietary treatments, and Becamia discala had an influence on oxide among the treatments. Strongyloids was found on the fourth dietary treatment. A positive relationship was observed on strongyloids, and negative relationship was observed on trichuris and oxide. In this study, goats were fed Becamia discala had 5 FEC to ascertain that goats in BFL were not given Becamia discala. Becamia discala influenced the FSC and significantly reduced their wooden home. In this study, Becamia discala live meal did not reduce the oxide value. I conclude by the study indicated that the Becamia discala supplementation significantly influenced gastrointestinal nematode of South African nondescript goats. The findings highlighted the differential effects of Becamia discala supplementation on gastroparasites, suggesting its potential managing specific infection based on the supplementation level. I recommend that several studies can be made to improve the performance of goats. Promote the use of Becamia discala as part of an integrated parasite management strategy. I would like to acknowledge University of Limpopo, Department of Agricultural Economics and Animal Production. Here are the references used. Thank you.
Thank you very much, Ms. Tovejane. Can we give her another round of applause? The second round of applause was just opening the session for questions. Can we have questions? I think she completed her presentation in 8 minutes. We have some time for questions. Can I see hands? I see a hand here. There is a revolving mic, yeah. Can I ask students that are going to present in the next session to take your PowerPoint to the guys over there so they can load them for you? Go ahead.
Thank you very much. Your aim and objectives are the same. Can you comment on that?
Yes.
Why?
They're the same. They're the same.
Maybe just to help you, the aim is more like your vision for the study.
Okay.
After you've done all the science in the lab, what do you aim to achieve with your study? So take that going forward. Thank you.
Thank you.
Okay. I see another hand, Dr. Matapo.
Hello. Thank you. I really want to understand, Ms. Tovejani, on how did you ensure safety security measures as the extraction took place? Thank you.
Okay, thank you. The goats were held in individual cage so that if one can get an infection, it doesn't contaminate the other.
I think I saw another hand. Thank you for that question. I hope you are answered, Dr. Matapo, and then we will come over there. Yeah.
Okay, thank you. Mine is on the sample size. I am interested on the sample size, so I want to know how did the presenter arrive to 16 goats, because I find it being small sample size for the study. So I just want to know any scientific method that she applied to arrive to that 16. Thank you.
Okay, thank you. We had 16 goals and 4 treatment. Per treatment, there were 4 goals. And then with the symbols, we collected symbols from each treatment.
Mr. Vejan, are you done? Okay. I hope you are answered. She was trying to explain how she arrived at that 24. Can we have the last one before we get the next speaker or next presenter?
Oh, okay. Thank you, Hypatia. I wanted to find out what's the difference between an FEC and FOC.
FOC stands for fecal oxide count and FEC stands for fecal egg count. Fecal egg count it measures number of parasitic egg per gram of feces, and then FOC measures the parasitic oxide per gram of feces.
Thank you very much, Ms. Tubejane. Can we give her another round of applause? You have exhausted your time. Thank you very much. The next speaker is Mamufelo Mashinye. Mamufelo Mashinye is going to give a talk on the integrated water resource management for deficit irrigated potatoes, the influence of cultivar selection, fertilizer application rates, and mulch application of water productivity. Can we give a round of applause?
Good day, I am Mashinye Mamufelo. I am a second-year master's student and I'll be presenting on my current study titled Integrated Water Resource Management for Deficit Irrigated Potatoes, Influence of Cultivar Selection, Fertilizer Application Rates, and Multiplication on Water Productivity, by myself, along with Professor N.Z. Ngobese, Professor P.E. Dlamini, and Dr. S.T. Hadebe. Potatoes are a major staple crop and an economic asset amongst other crops produced in South Africa, particularly in the Limpopo province, which accounts for over 21% of total potato production in the region. However, the production of potatoes is limited by water scarcity as a result of low erratic rainfall, along with increasing evaporation due to high temperatures. The resulting water stress not only limits yields, but it also reduces overall water productivity, which is defined as the total agricultural output or economic output that is produced per unit water taken up by the crop. And despite efforts made to increase irrigation, the issue of food insecurity persists, which calls for sustainable water management strategies to curb these challenges. At the intersection of science, innovation, and entrepreneurship was an integrated water resource management strategy whose goal was basically to maximize tuber yields using less water, where potatoes were established under deficit irrigation under an integration of fertilizer application rates, mulch application, and cultivar selection. Optimizing fertilizer application rates based on specific crop requirements helps to not only increase crop productivity, but it also reduces overall fertilizer wastage. And this is because while fertilizers are generally known to enhance crop yields, excessive or inadequate fertilizers can potentially limit both the agricultural and the economic constituents of water productivity. Mulch application, moreover, was an essential method for ensuring that water productivity is maximized under deficit irrigation. And this is because mulch has generally been shown to increase soil water retention whilst reducing overall soil water losses through evaporation. Cultivar selection based on agroecological adaptability and cultivar maturity cycles, moreover, was an essential method for ensuring that high-yielding and resilient cultivars are selected for respective production regions, which can help to maximize water productivity despite water limitations. So this study focused on determining the influence of integrated water resource management, particularly centered on cultivar selection, fertilizer application rates, and multiplication on the water productivity of deficit irrigated potatoes. The study was established at the University of Limpopo Experimental Farm. The trial was laid out in a split-split plot in a randomized complete block design with 3 replications, under which fertilizer application rate was the main plot factor, consisting of 100% fertilizer, 50% fertilizer, and 0% fertilizer. Mulch application was the subplot factor, consisting of mulch and no mulch, and cultivar selection was the sub-subplot factor, consisting of Sababa, Mondial, Tyson, and Panamera. Sababa is early maturing, Panamera is late maturing, and Mondial and Tyson are medium maturing cultivars. Moreover, Tyson has especially been bred for agroecological adaptability to hot, dry conditions such as those of the Limpopo region. Agronomic management consisted of inserting access tubes in each subplot for weekly soil water content determinations, along with sprinkler irrigation up to 80% of field capacity and deficit irrigation employed through the restriction of irrigation at the late tuber bulking stage of potatoes. 100% fertilizer rates were represented by 240 kilograms per hectare of nitrogen, 135 kilograms per hectare of phosphorus, and 108 kilograms per hectare of potassium. 50% fertilizer was half of these rates, and 0% fertilizer was simply an omission of fertilizer. Mulch, particularly lucerne mulch, was applied at 6,000 kilograms per hectare. Data collection consisted primarily of phenology, along with crop growth parameters such as canopy cover, chlorophyll content index, and biomass. Yield data, collected after physiological maturity, primarily consisted of tuber fresh mass, along with tuber grading in the listed measures. A moisture probe and moisture meter were used to determine weekly soil water content, where the sum of each week's irrigation then constituted total irrigation of the production season, and which was then inputted into calculations for evapotranspiration, otherwise known as water use. And ultimately, in this study, water productivity was calculated by dividing tuber fresh mass by evapotranspiration. Data were subjected to analysis of variance using GenStat version 21 software, where multiple mean comparison was performed using Bonferroni test, and mean separation determined at a 5% probability level. The study found that water productivity was maximized by integrating 100% fertilizer with cultivar selection. And here we see that this was primarily because under 100% fertilizer rates, critical growth parameters such as canopy cover, shoot dry mass, and chlorophyll content index were maximized, which basically maximized the overall photosynthetic area. The result of this was the highest yields being observed under 100% fertilizer at 35 tons per hectare, along with the greatest proportion of large tubers also being found under 100% fertilizer. So what does this mean? It basically means that by applying complete fertilization, we're able to generate higher yields using the same amount of water, which not only shows an overall enhanced water use efficiency, but it also generates a means for farmers to actually increase their profit margins. And this is because by applying 100% fertilizer rates, this complete fertilization actually offers an economic advantage in the sense that farmers will be able to enhance their yields without having to incur the additional costs that come with applying more water. And this is especially true if you integrate 100% fertilizer rates with cultivars such as Panamera and Mondial, which in this study were found to have prolonged physiological maturity period, which further enhances their overall yield potential. We also found that water productivity was enhanced by integrating mulch application with cultivar selection, reaching a water productivity value of 3.9 kilograms per meters cubed. Primarily here, we saw that mulch indeed proved to act as a soil barrier that enhances soil moisture retention whilst reducing overall evaporative water losses. And although the results in this case are lower than those observed on 100% fertilizer, the results still show that there is a potential for mulch in the sense that it acts as an affordable alternative, particularly for those farmers who may not be able to afford 100% fertilizer rates or even the soil tests that often come with applying more fertilizer. So in this way, they'll still be able to enhance their yields under the available soil moisture content. We also found that under mulch conditions, Panamera and Sababa showed distinct yet complementary characteristics in the sense that Panamera, the late maturing cultivar, benefited from the extended water availability that was provided over a longer growth period, while Sababa, the early maturing cultivar, capitalized on the immediate water resources that were facilitated by mulch. So this shows a potential for these cultivars to actually be established simultaneously, which would not only further enhance water productivity, but it would also present farmers with the opportunity to actually spread their financial risks and generate overall more stable incomes because they'll be able to harvest more potatoes over different periods. So in summation, we're able basically to show the potential of integrated water resource management to transform agricultural practices, particularly under deficit irrigation. We found that water productivity can be maximized by integrating 100% fertilizer with either Panamera or Mondial, or integrating loosened mulch with Sababa and Panamera. And in this way, we're able to demonstrate how science-driven innovation can actually lead to practical solutions that align with economic objectives. And hopefully, this research can serve as a guide for farmers and other agricultural entrepreneurs who are aiming to maximize their agricultural and economic outputs with limited water resources. In closing, I'd like to thank NRF DAD, who funded the study, along with West Grow, who provided the potato seed tubers for the study. I'd also like to thank Northwest University and the broader CRISP project under which the study falls. Thank you.
Thank you very much, Ms. Mashinye, for such a wonderful presentation. Can we give her another round of applause? I will open this time for questions. She is smiling. She wants to respond to your concerns. Can I get questions, please? Dr. Slepe over there. Yeah, I recognize you as well. Let's start with Dr. Slepe there.
Good day. How are you?
Fine, and you, sir?
I'm okay. Thanks for your presentation. You couple water resource, isn't it, with the productivity, less upscaling. From my understanding, you check how productive if you use fertilizers and then you water those plants. I just want to know from my side, what is your intake? When you were checking these different plants, is there any genetic modification?
So can you please clarify on the question?
Excuse me?
Can you please clarify? Genetic modification in what sense?
In the content of your product. You are using the fertilizers. You are using water at the same time. And then did you check any genetic modification of your plants or of your potatoes you have produced?
No, we did not take any genetic modifications.
Maybe I'll recommend in future you need to do that, because when we talk about food security, we eat so many things which are genetically modified. It can be maybe through the type of fertilizers we are using. And then another question which I just want to hear from you. I'm not familiar with this study, but I was just doing, thinking. With water resource, you water these potatoes using sufficient water. What is your take on the groundwater? Something to do with pollution. Do you think you are polluting the groundwater? What is your take on that? Because fertilizers themselves, they are detrimental. That's why if you look at the farmers, there are some restriction like using the aerial fertilization. Because at the end of the day, they do have impact. What is your take on that?
Yes, I do understand that fertilizer can have a polluting impact on the environment, definitely, particularly your chemical ones, especially if you just apply and there's no actual threshold or standard that you're using to just apply. And basically, I'm applying with the assumption that you'll just make your plants grow. But in this case, we conducted soil tests before to find out how much fertilizer we needed to add in accordance with a set target yield of 50 tons per hectare. So basically the amount of fertilizer that we had in the soil then, you could say, was sufficient for the plant to actually take up and then produce and grow optimally and then produce maximum yields. Thank you.
Thank you very much. I can see Dr. Slepe want to make a follow-up, but you can ask later on. Guys, let's try to be short and straight to the point so that we save time. We are only going to take the last one now because we no longer have time. Can we have the guy there behind, next to Neptaru? Yeah.
Thank you, my sister, for your presentation. My question is, did you do any soil analysis to prior application of your fertilizer?
Yes, so as I mentioned, we did do soil analysis. We basically sent our soil samples to the soil analytics laboratory near Sadara, and then they did the soil test for us to basically see how much fertilizer we had, and how much nutrients we had in the soil, and then how much fertilizer we needed to add in accordance with a set targeted yield of 50 tons per hectare.
Okay, thank you. The last one is, did you check for the allelopathic effects of your organic mulch, which is lucerne?
Excuse me?
Did you assess or check if your organic mulch, lucerne, has any allelopathic effects?
No, basically here we just aimed at maximizing tuber yields with the amount of water that we had. But I think it's a good suggestion, definitely.
Thank you very much, Ms. Mashia. Can we give her another round of applause? I know the guy still wanted to make a follow-up, and thanks for your understanding. Can we move on to the next presenter? The next presenter is Lerato Koloti. Lerato Koloti. Can we chair her up as she's moving to the fore? So Lerato is going to present to us assessing the effectiveness of community-based early warning systems in the Nelson Mandela Bay. Over to you, Lerato.
Greetings, everyone. I'm Koloti Lerato, a first-year master's student from the Department of Geography and Environmental Studies. I'm here to present my research proposal titled Assessing the Effectiveness of Community-Based Early Warning System in Reducing Coastal Flooding Risk in Informal Settlement, a case study of Nelson Mandela Bay, under the supervision of Dr. N. Matebula and Ms. I. Botha. Climate change is expected to become more pronounced in this century and in the upcoming years. It is already causing severe weather occurrences, such as heavy rainstorms that lead to a serious flooding across the globe. Nelson Mandela Bay is one of the municipalities that have been facing challenges posed by climate change, such as flooding, which particularly a coastal flooding, which is a major hazard to communities around the world, especially in informal settlement, where people lack communication, infrastructure, and necessary facilities to respond to such disasters. Lapland is one of the informal settlements in Nelson Mandela Bay that has been experiencing flooding that has destroyed homes and causing the displacement of the resident, because it's situated in a floodplain area. Therefore, there is a need to evaluate how community-based early warning system can enhance community resilience, particularly in informal settlements. Picture 1 of Figure 1 depicts the living conditions in Nelson Mandela Bay, which are characterized by poor infrastructure and poor housing materials. Picture 2 depicts the dam that is situated near Lapland, which during heavy rainfall, it does contribute to the coastal flooding. Picture 3, depicts the example of early warning system. Community-based early warning system offer timely information to communities, potentially for saving life and reducing property loss. The effectiveness of community-based early warning system and formal settlement is not well understood, since the majority of literature review focuses on urban formal settlement. Therefore, there is a limited empirical data exists on the functionality of community-based early warning system and informal settlement. Nelson Mandela Bay is a significant case study area due to the frequent coastal flooding events which have affected informal settlement, particularly Lapland and Karianga. Evaluating the effectiveness of community-based early warning system can help to improve disaster risk management strategies and align with sustainable development goal level, which advocate for making cities inclusive, safe, resilient, and sustainable. The aim of the study is to evaluate the effectiveness of community-based early warning system in reducing coastal flooding risk and informal settlement of Lapland, and the objectives are as follows: to investigate how well community members of Lapland understand floods and their vulnerabilities, to analyze the current state of community-based early warning system and informal settlement of Lapland, to assess the level of effectiveness of community-based early warning system in facilitating timely evacuations during coastal flooding events, to explore the perceived challenges faced in community-based early warning system and recommend strategies to improve their effectiveness. Here is the study area of Lapland. It's situated in South Africa, Eastern Cape Province, Nelson Mandela Bay Municipality. Lapland is an informal settlement in an urban area of Karianga and Eastern Cape Province within Nelson Mandela Bay Municipalities. Here are the coordinates, the exact coordinates of Lapland, and is situated approximately 35 kilometers from the Indian Ocean's coastline, and the total estimated area of Lapland is also here on the slide. Lapland is located in a floodplain area between Trivel Dam and Rosedale Recreational Wall, and it has approximately 125 number of households. This study will employ mixed method approach, which will incorporate both qualitative and quantitative methodologies, and the primary data will be acquired through tools like a questionnaire and key informant interviews, and the secondary data will be acquired through reviewing existing reports, government documents, and research studies. The study population of the study is the residents of Lapland that are residing in informal settlement, and they must be aged 18 years plus that are vulnerable to coastal flooding. And a sampling frame of the study is all 125 households in Lapland informal settlement. And this study will adopt complete enumeration approach, which will include all of the 120 number of households. And the sampling method that will be used is a purposive sampling method. And purposive sampling method will be used to select the officials from the government, ward councillors, officials that are involved in disaster risk management and evacuation processes during coastal flooding. For objective 1, the data will be acquired through questionnaire, which will include Likert scale question that will provide ordinal scale data and will be analyzed through descriptive analysis and Mann-Windley U-test, which will test the significant difference maybe in awareness score between males and females, and will be presented through bar graph, pie charts, and tables. For objective 2, the data will be acquired through questionnaire and will be analyzed through Spearman p-test, and will be presented through bar graph, pie charts, table, and p-value, which will show the strength of relationship between two variables. For objective 3, the data will be acquired through secondary data. We will be reviewing municipal disaster risk management records and will be analyzed through Kendall test and will be presented through a graph to analyze the trends and summary of key themes. For objective 4, the data will be acquired through questionnaire and key informants and will be analyzed through descriptive analysis and thematic method and presented through bar graph, pie charts, table, and summary of key themes. The scientific contributions of this study will enhance the understanding of early warning system and informal settlement, will also contribute to local policy and disaster planning, and will also support the alignment with national climate and disaster framework and promotes the sustainable development goal based on resilience strategies. The University of Limpopo offers researchers a number of necessary resources such as library and ICT. For the collection of this data, BRICS project will offer transport and accommodation during the data collection process. Audio recording equipment will be used to capture interview data. Ethical clearance will be requested from the University of Limpopo, which is CHIREC. Informed consent in English and isiXhosa will be provided. Compliance with POPIA Act 2020 for data privacy. I would like to pass my gratitude to the University of Limpopo, Department of Geography and Environmental Studies, appreciation of guardians from Dr. Matebula and Ms. Aibotha, and also like to acknowledge Mr. Sekome for helping me with the study area map. Here are the references that are used in the slides. Thank you so much.
Thank you very much, Ms. Koloti. Can we give her another round of applause? Community-based early warning system in reducing coastal flood. Can we have questions for her, please? Dr. Adebede, you can start.
Thank you, Ms. Koloti. Just a quick question. Is this a proposal or are you actually, do you have any results which you are supposed to present to us?
Okay, thank you for the question. So no, it's a proposal. I'm a first year master's student.
Okay.
I don't have a result yet.
Fair and fine. Maybe I didn't catch this then from your work. Which community-based early warning systems are you going to focus on?
Early warning systems.
Like, give me examples of typical ones that you will find in the Nelson Mandela Bay, within the communities that you're looking at.
Maybe according to the literature review that I've gone through, the examples, it will include maybe sirens or through sending SMSs. Thank you.
Can we have another question? I think we still have 2 more minutes for her. Yes, you can go ahead, yeah.
The p-value does not measure the strength of the relationship between 2 variables.
Sorry?
The p-value does not measure the strength of a relationship or between 2 variables.
It shows the, what's that? It shows the significant difference between 2 variables. Yes.
But not the strength, okay.
Not the strength, yes.
Yes, correlation analysis and regression analysis would measure the strength of the relationship between 2 variables.
Okay, thank you for that, sir.
Oh, okay.
Thank you very much, Koloti. Can we give another round of applause? Let me take this opportunity to thank you for, giving me an ear to chair the session and to the presenters as well. I think there was not even a single question that was aimed at, embarrassing anyone. I think it was just, a question that seeks to understand your study and some, clarity-seeking questions. And I think you did exceptionally very well. Thank you very much.
Good afternoon, everyone. I hope the afternoon is treating you very well. I'll be taking over from Dr. Mugwabana as the chair. There is one thing that I need to highlight. There was a message that if there are those who are having proposals, please, we're not supposed to present proposals. We're here to present the work that is already conducted. So if there are those, please bear with us. We are going to follow the approach that was applied by Dr. Mugwabana in terms of time. Our allocation for presentation is 10 minutes, then we've got 5 questions. As we are asking questions, let's try to be straight to the point and let's ask that question that you think should be the one that should be answered to avoid wasting time. Our first presenter is going to be Patience Ntibani. Is the presenter here? Let's welcome here. Thank you very much. And the topic is accumulation of pathogenic bacteria in agricultural soil irrigated with treated wastewater and their relationship with selected soil fertility variables. We can give a welcoming hand again as she takes over.
Good afternoon, everyone. My name is Ntibani Patience, and I'm currently pursuing PhD in soil sciences. Today I'll be presenting to you my MSc study, which is titled Accumulation of Pathogenic Bacteria in Agricultural Soil Irrigated with Treated Wastewater and Their Relationship with Soil Fertility Variables. The study was supervised and co-authored by Dr. P.M. Hopa and Dr. Al Mujonji. Now, water scarcity is one of the greatest environmental challenges that is being faced on a global level. And as a mitigation strategy, there's been a huge shift towards the use of treated wastewater in the agricultural sector to help alleviate the pressure of water scarcity. Now, the water contains a high concentration of macronutrients such as nitrogen, phosphorus, and potassium. And when this water is used for irrigation, it acts as a fertilizing agent, reducing the need for chemical fertilizer use to supplement any shortages in the soil. Now, despite the aforementioned benefit, a study that was conducted by Hopa et al. 2021 indicated that treated wastewater from the University of Limpopo Experimental Farm contains traces of human bacterial pathogens such as E. coli and enteric bacteria. Now, when such water is used for irrigation, according to Farad Khani et al. 2018, it actually introduces the pathogens to the soil. But then a study that was conducted in 2015 highlighted that we cannot actually predict the level of contamination that will occur in the soil when we use the water by just looking at the bacterial loads that we have in the water. Now, this then brought us to our research questions, which we developed looking at the study that was conducted by Hopa et al. 2021. The first question was, what is the level of bacterial pathogenic contamination in agricultural soil irrigated with treated wastewater from the University of Limpopo Experimental Farm? And then we also wanted to know what then could be the link of the accumulated pathogenic bacteria with soil fertility variables. Now, in order for us to answer the questions that I've already mentioned, we started by collecting treated wastewater and a composite soil sample from the University of Limpopo Experimental Farm. Now, the soil was taken to the University of Limpopo, where it was actually air-dried, and then we used the water and soil sample to actually establish a 2x5 factorial experiment. So the first factor consisted of controlled temperature, which was kept at 37 Celsius degrees, and secondly, uncontrolled temperature. Now, the second factor consisted of 5 irrigation treatments. We had 3 levels of dilutions, which we created using treated wastewater and tap water, and 2 levels of irrigation treatments that were actually not diluted, and that included treated wastewater and tap water. Now, from the water, pre- and post-soil samples that we collected from the University of Limpopo Experimental Farm, we actually isolated 4 bacterial pathogens, and those included Staphylococcus, enteric bacteria, E. coli, and Pseudomonas aeruginosa. Now, these were isolated using the spread plate method, and the selective agars that we used are actually shown on the presentation. Now, we then did some analysis for some physical-chemical properties, and the parameters that we actually did check in the soil were soil texture, phosphorus, soil pH, soil electrical conductivity, potentially mineralizable nitrogen, and nitrate. Now, all the data that we collected after we have contaminated our soil with treated wastewater, we actually subjected it to a 2-way analysis of variance in order for us to actually see the impact of the 2 factors that I've mentioned, and also to see their impact when they are actually interacted. Now, we then separated significant means using Duncan's multiple range test, and then lastly we did Pearson's correlation to assess the relationship between accumulated pathogenic bacteria and selected soil fertility variables. Now, on the data that we got from the soil just before we irrigated it with treated wastewater, the pH actually indicated that our soil was slightly alkaline, and the electrical conductivity of the soil indicated that our soil was non-saline, but our soil actually had low concentration of potentially mineralizable nitrogen and nitrate. When we looked at the selected pathogenic bacteria, we actually observed that the soil before irrigation, it already had traces of all the 4 selected bacterial pathogens, but it had the lowest counts of enteric bacteria. Now, we then looked at our irrigation treatments that we developed using treated wastewater and tap water. And from the results, we did see that tap water had no traces of Staphylococcus and E. coli, but had the lowest counts of enteric bacteria and Pseudomonas aeruginosa. But the other thing that we realized is that each time we increased the concentration of treated wastewater, it also increased the bacterial loads of the selected bacterial pathogens, indicating that treated wastewater did actually have the higher loads of these pathogens, and each time we actually used tap water to dilute it, it actually decreased. Now, when we looked at the data that we obtained after we've irrigated the soil with treated wastewater, we did observe a significant increase in terms of the bacterial loads in the soil, and treated wastewater contributed more to the accumulation compared to tap water. And the one thing that we were able to pick up from this is that our data pretty much correlated with the information that we have on our water. I would like to indicate that the black dotted line actually indicates the state of the soil just before we irrigated it with treated wastewater, and one would have expected that tap water where we irrigated with tap water, he or she won't find Staphylococcus and E. coli. But I would like you to keep in mind that the soil already had these counts in the soil. And us providing the soil with the water, we just simply created environments that were conducive enough for the proliferation of these organisms. Now, when we looked at the fertility variables, we did observe a significant increase in terms of the electrical conductivity, but not above the threshold where it indicates that the soil is actually non-saline. And then when we looked at the pH, we did also observe a significant increase, but not beyond a state where it wouldn't favor the availability of soil nutrients to the microorganisms. And then in case of the soil organic carbon and phosphorus, there was a significant increase, but there were a few decreases here and there. And if you can look at the graph for soil organic carbon, where we irrigated the soil with 100% treated wastewater, we observed a significant decrease. And this is because pathogenic microorganisms use carbon for their own metabolism. And then over time, this actually leads to it going down, especially when it is actually not being supplemented. And when we looked at the nitrate and potentially mineralizable nitrogen, we did observe some increases, similar to what we observed in, for carbon and phosphorus. But however, there were some decreases. For example, where we irrigated the soil with 50% treated wastewater combined with 50% TP, we observed a decrease and potentially mineralizable nitrogen. And this again was linked back to the accumulation of the pathogens in the soil, which were actually using these nutrients for their own metabolism. On the correlation results, we observed a positive correlation between our pathogenic microbes, and those pathogenic microbes included enteric bacteria, E. coli, and Pseudomonas aeruginosa. They had a positive relationship with nitrate and phosphorus, and this is because treated wastewater is actually associated with a higher concentration of macronutrients, and when these are delivered to the soil, they actually favor the survival and proliferation of these microorganisms. And surprisingly, we observed a negative relationship between the bacterial loads of Staphylococcus and soil organic carbon, and this can be attributed to the fact that counts of Staphylococcus under treated wastewater, they were observed to be much higher, and they then used the carbon in the soil for their own metabolism, thereby decreasing it. In conclusion, what we observed is that treated wastewater led to an increase in the accumulation of the selected pathogenic microbes together with the nutrients, which included nitrogen, phosphorus, and carbon. But however, each time we diluted it, it actually reduced the level of accumulation. The other thing that we were able to pick up from our study is that controlled temperature pretty much favored the survival of the pathogens, whereas the uncontrolled temperature did not favor it up to the level that controlled temperature did. And this can be because under uncontrolled temperature, the temperature would sometimes fluctuate up to levels that were not suitable for the survival of the microbes. And when it reached such levels, they got denatured and their counts got reduced. This study wasn't able to answer all questions, and so we would like to say with the study, we think other studies can actually try and answer the following questions, which what is the long-term effects of using treated wastewater on the accumulation of soil pathogens, pathogenic pathogens and soil health, and also they could try and unravel possible interactions between various crops and treated wastewater that may influence the accumulation of bacterial pathogens. Now, these are the references that are used in compiling these slides. I would like to acknowledge the National Research Foundation for funding and the University of Limpopo for infrastructure. If there's anyone who wishes to read further on the work, I would like to let you know that the work is actually available on the internet. I would like to thank you all for your attention. Over to you, Chair.
Thank you very much for the wonderful presentation. Can we now take hands for questions? Only one question per person, please. Can you have questions from the floor? One, there's one hand there.
Thank you so much for your presentation. I'm not too much of a soil scientist, but you know what I was listening out for? If you look at the theme of the FSA Research Day, it says that we are trying to merge research that happens in the field, in the lab, together with entrepreneurship and human impact. What would you comment is the contribution of your work now? I know it's a bit challenging to that theme. Thank you.
Can you please repeat it?
I'm just asking you to relate your work, which is much more on the lab and field side,
yes,
to the human impact which is basically the theme of the research day that we're conducting. Thanks.
Okay, with regards to the human impact, there is some limitations which I did highlight, but I would like to say, actually, our study actually highlights that we shouldn't use treated wastewater as it is, especially the one that we have at the Mangweni treatment plant for irrigating our crops. I think you do know that Limpopo, some areas of Limpopo are classified as semi-arid, and we think that we can use actually that water to help supplement the water shortages that we have so that we can obtain optimum growth, and then we can actually help with food security. But provided we don't actually use the undiluted form, because it can actually be quite harmful, as you saw, accumulation of pathogens taking place in the soil, and this can actually be transported from the soil to the crops. So we would advise that one actually uses the diluted form.
Thank you very much for the wonderful presentation. I don't see any last hand. May we give her the round of applause, please? Okay, because of time, we're going to now go for lunch. The announcement as far as the lunch is concerned is that we're only allocated 40 minutes. And then when we go to lunch that side, let's occupy one area so that the others can be able to use the other side. We can use the left side of the tables. Thank you very much.
Hello. Hello.
And the South African orange subsector, it faces complex challenges of balancing social, environmental, and economic sustainability. As the orange cultivation expands, it has raised concerns regarding its impact on local communities and ecosystems. And then the research gap that I've analyzed is that there is a lack of understanding of selected sustainability factors in relation to the subsector within the citrus industry. In terms of the importance of the study, the orange industry is economically important to South Africa, contributing significantly to exports and local livelihood. However, sustainability concerns, including environmental impacts, social equity, and long-term economic viability, they pose a challenge to the sector's future. And then the study aims to examine the relationship between these selected environmental, social, and economic sustainability factors in the South African orange industry. The objectives are to determine the stationarity, where to determine the stationarity of the variables, and the key one was to examine the short and long run relationship between these selected aspects of environmental, social, and economic sustainability factors. And then the study hypothesizes that there is no short or long-term relationship between selected aspects of environmental, social, and economic sustainability in the South African orange industry when analyzed using the RDL model. Methodology and analytical procedure that was used. The study was conducted within South Africa, and then the data sources were collected from 2000 to 2020, of which that amounts to 21 observations from the World Bank, European Commission, and the Greenhouse Gas Inventory for South Africa and Statista. And then the methodology, we used the eView software to conduct the ADF test to test for stationarity. And then we also used the autoregressive distributed lag model to examine the short-term and long-term relationship. And then my variables are land, of which it measures the area allocated to orange farming, and a proxy variable for environmental sustainability, and then employment, of which is a proxy for social sustainability, and then export, which measures the volume of oranges exported, which is a proxy variable for economic sustainability, and then CO2, which is a proxy variable for environmental sustainability. In terms of the result, the ADF test result revealed that all variables were stationary at first difference with their p-value less than 0.05. The VAR leg order selection test result, they revealed that they showed that leg 4 is the best model because it has the most asterisks as portrayed in the table, showing it performs best across the key criteria. And then the RDL short run result, they revealed that amongst all the variables that were tested or analyzed, only CO2 was statistically significant, of which it posed a negative impact on sustainability, and then the other variables were not statistically significant, meaning they do not influence sustainability. And then the RDL long run result revealed that export and land area are statistically significant, of which they influence negatively on sustainability of the orange industry. And then employment also it showed a slight positive contribution, since the p-value is much greater than 0.05, but it's closer to it. In conclusion, data stationarity was conducted using the ADF test, and it confirmed that export employment, CO2 emission, land area are stationary at first different. And then the modeling framework, we opted, it revealed that we must use the optimal leg length of 4. And then the long run findings, they revealed that exports and land area showed significant negative effects on sustainability, and then employment slightly positive contribution. And then the short run findings, they revealed that CO2 emission have a strong negative impact, highlighting immediate environmental challenges. And then employment and export, they showed a limited term influence. The study recommends that the orange sector must invest in sustainable farming, of which it includes precision farming, organic method, pest management to reduce the CO2 and enhance environmental valuation, and to protect the land. And also invest in renewable energy, which is in solar, wind, and alternative to reduce fossil fuels reliance and load shedding risk. And also trade relations, of which it has to negotiate fair terms, comply with regulation, and secure market access, especially with the European Union. And then in terms of employment, it recommends employment policies to improve wages, conditions, and training for workers and ensure social equity. I would like to pass my sincere thanks to Dr. Elsie Muchopa and the Almighty and also for her invaluable guidance throughout the study. And these are the references that I've used to compile this presentation. And thank you.
Thank you very much, sir, for the presentation. You can come this side. It's okay. Can we give him a second round of applause as we go to the questions? Can we now take questions from the floor? Questions? There is one there.
Okay. Thank you for the nice presentation. I just wanted to ask you that, looking at those years, didn't you have a particular year of which maybe the orange production was low?
Looking at what? You cut it from the beginning.
I mean, you looked at the years, so I wanted to understand if you didn't encounter, using that data, the year whereby the orange production was low.
Not really, because, okay, my study is secondary data. I use secondary data, and then it is collected from credible sources, of which I analyzed using the RDL model, of which it doesn't really account for a specific year, but it gives the overall result. Yes.
Any more questions? Is it the only one? Okay, mine would be, what is the implications of your forecasting in relation to future production based on your study?
Oh, okay. In terms of future research. My results imply that we used future research must use panel data analysis in order to capture like the accurate result in terms of, since my study it had like lower years, which is it had less observations. I mean, yes.
Thank you very much. We can give him a round of applause as he's going down. And can we have Maluleke? Do you have answer? Can you also give the last round of applause before she starts?
Good day, everyone. My name is Success Malulega. I am currently in my first year of Masters in Agricultural Management, specializing in plant production. Today, I will be presenting my honors research study under the topic that says the effect of treated wastewater on the mineral content of 2 tomato cultivars under the supervision of Dr. Hupa PM and Dr. Masewe MR. Treated wastewater is water that has undergone the reclamation process to remove pollutants. These processes include the removal of solids and the breaking down of organic matter until the water is released into a water body. Tomatoes are edible fruits belonging to the Els Lycopersicum species, and in this study we focused on 2 cultivars, which is Roma and Rhodate. Water is an important resource for all life forms, but it is becoming more difficult to have access to it, which proves the issue of water scarcity in agriculture, especially in South Africa. And farmers need alternative water sources like treated wastewater in order to sustain their production. The use of treated wastewater in some studies has resulted in an increase in tomato yield compared to the use of tap water. This is because treated wastewater is found to have beneficial micronutrients and macronutrients which promote plant growth. However, the water still contains harmful elements like cadmium, lead, and nickel, which proves us that there is a lack of information on the potential effects of treated wastewater on the mineral content of Rhodate and Roma tomato cultivars, making this study even more important. The limited availability of freshwater resources is affected by several factors. Factors like population growth, climate change, and water pollution. The increasing demand for nutritious food has made tomatoes' nutritional status of great importance, and less has been discovered about how treated wastewater affect tomatoes' mineral content. Tomatoes are an excellent source of minerals like phosphorus, potassium, and nitrogen. According to studies, tomatoes cultivated in treated wastewater may be less antioxidant-rich than tomatoes produced in fresh water. This is because there are some pollutants found in treated wastewater that may impair the enzyme activities that create antioxidants in tomatoes, which is another reason why this study was done in order to see the effectiveness and safety of treated wastewater use in tomato cultivation. This study aimed to investigate the effect of treated wastewater on the mineral content of 2 tomato cultivars. The objective was to determine the effect of treated wastewater dilutions with tap water on the mineral content of Rhodate and Roma tomato cultivars. It was hypothesized that treated wastewater dilutions will not affect the mineral content of these tomato cultivars. The study was conducted at the University of Limpopo in a plastic tunnel next to the soil science laboratory. The soil used in the study was collected from a fallowed field in the University of Limpopo experimental farm, and the treated wastewater was collected from the treatment plant in Mankweng. The study was laid out in a randomized complete block design. It was a 5x2 factorial with the first factor presenting the 5 levels of irrigation treatments, or we can say the 5 levels of water dilutions, number 1 being 100% tap water, 25% tap water plus 75% treated wastewater, 50% tap water plus 50% treated wastewater, 75% tap water plus 25% treated wastewater, and lastly 100% treated wastewater. Whereas the second factor represents the 2 tomato cultivars, which is Rhodate and Roma. The treatments were replicated 4 times. Under pre-trial soil analysis, soil texture was determined using the hydrometer method, soil pH using pH meter, electrical conductivity using the EC meter. And under tomato analysis, the minerals between the brackets were measured from a red ripe tomato using the ICPOS methods. The samples were taken out to ARC. The data was subjected to analysis of variance through GenStat 18th version. Duncan multiple range test was used to differentiate means at 5% level of significance, and Pearson correlation was used to identify the relationship between the minerals. These are the results under the pre-trial soil analysis. The soil had 8% of clay, 18% of sand, 74% of silt. The textural class was silt loam. The pH was 5.8, which is slightly acidic, and the EC was 2100, which is quite high salinity. Looking at figure 4.1, we can see that the interaction between the irrigation treatments and the tomato cultivars was significant, especially in rodent, as treatments with increasing treated wastewater percentages have high levels of iron. And going to figure 4.2, we can clearly see that there were no statistical differences in copper between the irrigation treatments and the tomato cultivars. Now going to figure 4.3, we also see that there were some changes in potassium levels, but looking at the 50% treated wastewater plus 50% tap water, raw date was the highest. And going to figure 4.4, treated wastewater increased magnesium levels in both cultivars, but raw date was the highest. And this is because magnesium is highly available to plants in acidic soils. Now going to figure 4.5. Treated wastewater is generally known to have high salt levels. That's why the sodium levels are so high in all the treatments in all cultivars. But again, Roma tried to be high, but then rhodate was the highest. And going to figure 4.6, we can clearly see that zinc had no statistical, there were no statistical differences in zinc between the irrigation treatments and the tomato cultivars. Going to figure 4.7, we can see that calcium levels between rhodate and Roma were fluctuating. This is greatly because calcium is greatly influenced by sodium and magnesium levels in the soil, which tells us that there was some sort of competition between these minerals. And now going to figure 4.8, which shows the effect of treated wastewater on manganese. We can see that manganese was also high, but then it was, Roma tried to cover rhodate by being higher than it. Going to figure 4.9, looking at both cultivars, in Roma and rhodate, we can see that nitrogen levels were almost the same. And going to figure 4.10, we can see that phosphorus levels are increasing in all cultivars, but the increase is more in rhodate than it is in Roma. The overall reason why it seems like rhodate has high levels of these minerals than Roma, it is because of their genetic traits and their physiological behaviors. Rhodate has a vigorous root system which can cover up a large volume of soil and absorb nutrients effectively. On the other hand, Roma has a slightly on the high salinity soil. We can see that rhodate showed that it is a South African bred cultivar by being able to adapt to whatever conditions that were in the soil and that were brought out by the water and managed to absorb all these minerals and have high levels of them, whereas Roma couldn't do that because it is naturally a light feeder. The study findings show that tomatoes' mineral value is significantly impacted by the irrigation of treated wastewater, especially when it comes to the rhodate cultivar. High treated wastewater ratio increases the mineral content in the tomatoes. However, treated wastewater had very little effect on the amount of copper and zinc, suggesting that not all minerals are similarly impacted by water quality. Therefore, it is recommended that the government should build more treatment plants to solve the issue of water scarcity that highly affects the agricultural sector. I would like to acknowledge my supervisors, Dr. Hopa and Dr. Maseve, Ms. Pintimbane for their contributions in the study, and the University of Limpopo for the resources, and also the National Research Foundation for the funds. These are some of the references that were used to compile these slides, and this marks the end of my presentation. Thank you.
Thank you very much for the wonderful presentation. Now we're opening for questions from the floor. Can we take questions, please? There's one there.
Thank you, Ms. Maluleka. I'll just quick fire 2 questions to you, and then there are no follow-ups, Chair. Yeah. The first one is you gave us generic chemical profile for your wastewater treatment. Like you didn't tell us exactly what was in the wastewater. Do you have those results? How do those results mirror what you are seeing in your plant? The second one is that you said the reason why differences between cultivars was that the other one was a light feeder, the other one was a heavy feeder because of the root system. The question then becomes, in a pot, and I saw your pots, they're not that big, does that really translate to an uptake advantage? Even if you have got a small root system there, does it not cover easily the pot that you are irrigating? Thanks.
Okay, thank you for the questions. No, I do not have results on the water because I did not do any analysis on them, but I did some analysis on the soil before transplanting the tomato seedlings, and we are sure that the results that we found on the tomatoes are really coming from the effect of the treated wastewater, because on the treated wastewater, it is where we try to do some manipulations with the dilutions. And now going to your second question, yes, the size of the pot did have some effect because during irrigation we saw that sometimes when we go there to irrigate, we found some of the pots very dry, meaning that rhodate is a heavy feeder when it comes to minerals. That's why sometimes when we irrigated in the morning and then we go there late, we found the soil dry. But then in Roma, you can irrigate today and then go back tomorrow and find that the soil is still wet.
Thank you. Do you have any last one? Thank you very much. We can give it a round of applause.
Thank you.
And the next speaker after Maluleke, Make. Can we have Make? Make, please. Great. You must be ready. People have been presenting. Don't worry. These people, they're just like you and me. Next time we must submit a PDF format. Huh?
Let's give the PDF.
For consistency, it also helps that people don't tamper with the things. Because when we handle them, then Gashutisha Fidewa. introduce yourself and the topic again. Let's cheer up again.
Good afternoon to you all. I go by the name of Mike Matlo, and I'll be presenting my study titled Effect of Feed Restriction on Growth Performance of Ross 308 broiler chickens, under the supervision of Prof. B. Gunya and under the co-supervision of Prof. T.G. Manielo and Mr. M. Kashega. This will be my outline. On the introduction, feed restriction is widely used management practice in broiler production, aimed at controlling growth rate and improving feed efficiency. Broiler chickens are characterized by fast growth rate and their maturity at day 42. Faster growth in broiler is associated with negative effects such as metabolic stress, increased body fat, higher mortality, more metabolic disease, and skeletal disorders, especially in those that are fed ad libitum. However, feed restriction have been found to be an effective management strategy for reducing fat in broiler. In addition, et al. Discovered that feed restriction reduces waste and production cost. The impact of feed restriction on growth has been inconclusive. The aim of this study is to evaluate the effect of feed restriction in broiler chickens. And the objective of the study is to evaluate the effect of feed restriction on initial and final weight of Ross 308 broiler chickens, and to evaluate feed restriction on intake, body weight gain, and feed conversion ratio of Ross 308 broiler chickens. This experiment was conducted at the University of Limpopo Experimental Farm in Sefilgale. A total of 270-day-old broiler chicks were used for this study. The broiler chickens were assigned to 3 different treatments in a complete randomized design, replicated 6 times with 42 chicks per house. The chicks were provided with light and water ad libitum. SPSS general linear model of statistics were used to analyze the data. The average gain weights were determined using McDonald's formula. And on result and discussion, feed restriction had influence on both initial and final weight of Ross 308 broiler chickens. As we can look at figure 1, we can see that broiler chickens that were assigned to treatment 1 had higher final weight compared to those that were subjected to treatment 2 and treatment 3. This can indicate that broiler chickens increases with an increasing weight. Broiler chickens that were assigned to treatment 1 demonstrated lower initial weight, and those that were subject to treatment 2 and treatment 3 demonstrated lower final weight. This indicates that broiler chickens that were subjected to treatment 2 and treatment 3 did not compensate for their growth. In fact, they used the feed that were fed to them to make up to the nutrients that were deprived during feed restrictions. And on the result of average daily feed intake, feed restriction influenced feed intake. The reduced feed intake and lowered gain weight and increased feed conversion ratio. This can be due to the altered nutrient intake. And Prof. et al. Also reported that feed restriction did not always result in growth or increased feed conversion ratio. This result shows that body weight increases with an increased level of feed restriction. And feed restriction also had an effect with feed conversion ratio. Broiler chickens that were assigned to treatment 2 and treatment 3 demonstrated higher feed conversion ratio, suggesting that broiler chickens' nutrient promotes weight, leading to compensating growth. Feed restriction did not always result in growth or did not always impact feed restriction. This can be concluded that feed restriction affected growth
performance. Broiler chickens that were subject to treatment 2 and 3 had lower performance in feed intake upon refeeding and did not contribute any significant improvement in growth performances. Applying that psychological demand of broiler growth initiate constant nutrient availability. However, feed restriction such in treatment 2 appear to be beneficial and could be practiced, and therefore it can be recommended to farmers. This study gives a virtual insight. However, there is no significant lack of research in this feed restriction. Thank you. And I would like to acknowledge the experimental farm staff for allowing us to conduct the study on their farm, and also acknowledging Department of Agricultural Economics and Animal Production for funding it.
Thank you very much for the wonderful presentation. The floor is now open for questions. Can we take questions? There is one there already.
Thank you very much for a very insightful presentation. You mentioned something about the stress indicators on your introduction, originally, if I'm not mistaken. So, since you mentioned that, I wanted to find if, did you perhaps maybe try to check about the stress indicators to show that, let's say, the feed restriction had an impact on the stress indicators such as glucose or cortisol or hematology?
No, we didn't go for that one.
Okay, thank you.
Thank you.
Okay, thank you. I want to check with you about the levels. Did you follow any literature? Because we know that some of the disadvantage of feed restriction, they come with the stress, which chicken might have the feather picking, and also some of the other disadvantages. So did you, the levels that you considered, can they let chicken to be stressed to a point that they do the feather picking and also other actions after being stressed? Thank you.
Can you repeat that question again?
We know that as much as we restrict feed to the chickens, they become stressed. Yes. Then they can do the feather picking and the other things. So I want to check the levels that you apply of restriction. Are they suitable enough to a point that they won't let the chickens to be stressed and have start to do feather picking and stuff?
Oh yes, we did check with other literatures, and we did experience some sort of stress, but that was one of the consequences of using the levels that we chose to use in this study. And thank you.
Thank you very much. Do you still have the last one? She still have enough time. Okay, on that note, it means that was the last one. Thank you very much, ma'am. And our next presenter is Koti. Can we welcome Koti on the stage? And while she's coming, Munisi and Mutapo, are they in? Okay, that means they are not around.
It's not my
Wait.
Okay.
Let's just wait for some few seconds to get the correct document. Let us also motivate her with hands.
Yes, please. Thank you. Greetings, ladies and gentlemen. My name is Sina Okoti, and I will be presenting my current study on. Okay. Greetings, ladies and gentlemen. My name is Sina Okoti, and I will be presenting my current study on assessing herbaceous vegetation composition after cropland abandonment in semi-arid savanna rangelands in Limpopo, supervised by Dr. Kusha, Mr. Klashek, and Dr. Mutseh. Cropland abandonment is the long-term cessation of cultivation without conversion to the other intensive land use, and it is driven by socio-economic, environmental, and institutional pressures. As it promotes woody species proliferation, reduces herbaceous cover, and alters disturbance regimes through fire suppression and reduced grazing. It is common in former homelands, which is Limpopo, Eastern Cape, and KZN, as it occurs at 0.08% to 0.28% per year. And it is linked to soil fertility loss, input shortage, and insecure tenure and rainfall variability. Abandonment initiates the secondary succession. Biodiversity recovery, it is often incomplete, creating novel ecosystems dominated by disturbance tolerant woody species, and this suppresses grasses and reduces regional productivity. Cropland abandonment, it is the key driver of passive restoration. Passive restoration, it is the natural recovery of ecosystem without human disturbance. The objective of this study is to assess the effect of cropland abandonment on herbaceous vegetation composition, diversity, and biomass. And the study was conducted in Sanctuary Village, Limpopo Province. The climate is semi-arid, and the rainfall ranges from 400 to 600 millimeters, and the temperature ranges from 4 degrees in winter and to 38 degrees in summer. The vegetation type is Polokwane plantive bushveld. The herbaceous vegetation sampling, and it was collected in abandoned cropland and natural rangeland site within the Centenary Village. 10 time-stamp plots were randomly established across each site. The herbaceous vegetation within the plot, 0.5 meters squared quadrant were laid, and all herbaceous species were identified and harvested using the grass-cutting shears. And the harvested herbaceous samples were oven-dried at 60 degrees Celsius for 48 hours to determine the dry herbaceous biomass production. And the data was analyzed to estimate the diversity using the Mechanics Index, which was used to estimate the richness, the Sonowina Index for diversity, and the Paulus evenness for the evenness. And then it was analyzed using the independent t-test, correlation, NMTS ordination, and the software that was used. It was R statistical software version 4.51. And this table shows the species composition, the life form, and the grazing value of the species found in the abandoned cropland site and the natural rangeland site. As we can see that the abandoned cropland site was dominated by Aristida congestia and Aristida ranochlora, whereas the natural rangeland site was dominated by the Chloris virgata and the Erograssus planum. And these species are low forage species. And these species are known as low forage quality species, and they are adaptable to harsh conditions. That's why they are dominated on the abandoned cropland site. moving on to the species diversity, the comparison, this figure shows the comparison between species diversity, the richness and the evenness. So as on figure 1A, we can see that the species richness was high compared to natural rangeland, meaning that the abundant cropland favored the more species coexistence because of the succession that was going there, that was happening there. And then from the Shonen winner index and the Pilo winner index, there is no significant. Figure 2A shows that the figure 2 showing that the frequency of ecological status on abundant cropland and the natural rangeland. As we can see that on figure 2A, the ecological status, the abandonment cropland had increased two species that were high compared to natural rangeland. As I said that the abandoned cropland, the abandoned cropland was dominated by species that are not, that can grow or are adaptable to harsh conditions. Compared, and the plant succession on abandoned cropland was dominated by the pioneer species, whereby the pioneer species are species that can grow in disturbed areas. And then figure 3 shows the non-metric multi-dimensional scaling of herbaceous composition, which it confirms what I've said on figure 2, that these two sites were compositionally dissimilar. Abandoned cropland and the natural rangeland, they had dissimilar species. In conclusion, despite the increased grass species richness in abundant cropland, the herbaceous layer is dominated by the stress-tolerant, low palatability species, indicative of the degraded condition and high grazing pressure. So this suggests that the richness alone does not equate to ecological recovery, as the functional quality remains compromised. So these findings underscore the complex interplay of land use history, disturbance regimes, and biotic pressures in shaping the post-abandonment vegetation dynamics. So the passive restoration may be insufficient in this context, highlighting the need for targeted management intervention to facilitate woody regeneration and improve herbaceous community composition for sustainable rangeland restoration. Thank you.
Thank you very much. Let's give you another hand before we go to questions. Can we now take hands for questions, please? Questions from the floor?
Thank you, ma'am, for a wonderful presentation. I'm interested in finding out what are did you go to the extent of determining the drivers of dissimilar species under the two land uses? And what were the findings?
Yes, I did find the drivers of the dissimilarities between the two sites. I think the dissimilarities between the species composition is because of on abandoned cropland, because of the fertilization history due to cultivation.
Any last question? The first presenters have given us enough time to ask a question. Okay? That was the last one, ma'am. Can we give a round of applause as she had assembled? In the absence of the two students who did not submit, I think that was our last student, and thank you very much for being a good audience. And I am Lesa Jalendowa, as I did not introduce myself in the beginning. Thank you.
Good afternoon, everyone. We are doing the graveyard slot. Even the questions, there are a few of them. And to energize you from time to time, I will throw in just a dry joke there and there. Hopefully, Prof. Masuku won't mind because time is of the essence. Just for housekeeping, we have got one last session after this. I think the first speaker in the last session is Itumeleng Mkabela, and then the last speaker is supposed to be Nevdalu, and I'm asking that all those speakers, there's a lot of them, they upload their presentations as we're speaking, as we in this session with the computer guys there. Right, so please make sure that you have done that. And for smooth sailing as well, what I will then just do is to ask to see if all our presenters are here. So I'll just go through name by name. This is to avoid cases where we then see that the presenter is not around. So we have got Madilonga. Is Madilonga around? Okay. Langa. Okay. Mopai. Okay, good. Mnisi? Faith Mnisi? Okay, it means that on our program, Faith Mnisi is not in, so we'll jump Faith Mnisi. Shlope, is Shlope around? Sorry. Is Shlope? Okay, thank you, Shlope. Gurubeni, is Gurubeni around? Okay, thank you. Makatsela? Makatsela is in. Mayepa? Oh, okay. Good. And then Kosi, I think I saw you around. Okay. Whilst they are busy doing the setup, just a few changes to the house rules. I'm actually co-chairing this session with one other person, Dr. Mbanjwa here. To make things easy in terms of time, if Dr. Mbanjwa, you don't mind just standing a bit, people need to see you because, yes. The guy wearing some very agricultural clothes. So, by the way, that was the first dry joke. Okay, so, so if Dr. Mbanjwa then stands up, it means you've got about a minute. So you start wrapping up then. And then, if then I stand up, it means you need to start shifting from those bases. Okay. Can I then ask our first speaker to come around? Oh, you are, Matilong. Okay. Then it means our first speaker is here. Okay. Our first speaker is Rofiwa Madilonga. And Rofiwa is looking at hydrological models and understanding groundwater. Thank you.
Yes. Greetings, everyone. Good afternoon again. My name is Madonga Ropa Teresa, and I'm working under the supervision of Professor Matiwa, Professor Chikore, and Dr. Kesmat. The project that I'm here to present is titled Using the Advanced Integrated Hydrological Model to Understand the Groundwater and Surface Water Interaction. Yes, Some of us might have wondered, how does groundwater and surface water actually communicate? Well, I was once in their shoes. The groundwater and surface water interactions that are really important for sustaining ecosystems and our water availability. The climate variability and increase in the water demand intensify the need for integrated modeling approaches. The advanced integrated hydrological models allows simultaneous simulations for groundwater and surface water interactions. There is a limit of applications which globally exists. This highlights the needs for systematical evaluation and capacity building. We have, or there is, a limit of knowledge. Where there is an interaction between surface and groundwater is poorly understood globally, in Africa, and also in South Africa. There are gaps of which have been identified, where there's a lack of water, which hinders the effective management. And these knowledge gaps tend to threaten our water security and ecosystems health. The aim of this study is to review the advanced hydrological models so that we can be able to understand how does groundwater and surface water interact and improve the water resource management. And the following are the objectives. The methodology of which will be applied in the study, the approach will follow the systematic review. The data sources will be obtained from the Google Scholar Web Sciences corpus, where the keywords included groundwater and surface interactions, hydrological interactions, modeling. The selection criteria was rigorous, relevance, geographic diversity, and the analysis was identification of the patterns, gaps, findings, and the recommendations. So this is the Prisma flow diagram, which was used to identify the studies via database. The process followed the identification of the studies, screening them, and inclusion. At first, we identified a total of 203 studies, but in the end, we ended up having studies that were included into the assessment, which were 90. How did we come to that? We excluded 54 due to the duplicacy and 34 due to the the data not being relevant and the outcomes not being relevant. The review of the findings, the diagram that you see, it essentially shows the yearly distribution of the articles which are published between 2000 and 2025. The data actually highlights the critical and the growing role of the integrated hydrological models into understanding how does groundwater and surface interact, not only globally, but also in local scale. This diagram shows the number of studies which showed interest in the use of integrated hydrological models from 2000 and to 2025. So figure number 3, it shows the African countries, of which I'm very proud to say that South Africa showed to be with the leading number of studies which showed interest into integrated hydrological modeling for surface water and groundwater. However, provincially wise, the leading province was Gauteng, followed by Western Cape. The least is Limpopo and Free State. In the world regions, Africa has shown a really great interest into applying the integrated hydrological models for groundwater and surface water. So this shows the interest into improving our water management and security. The diagram that you see simply shows the study's distribution of the water models. So the fully coupled model, of which is Mike Shee, it contained 30%. So this is a widely applied and dominant, and studies have shown interest and preferred the fully coupled models into the groundwater and surface water because it provides accurate and reliable results. The SWOT and mode flow showed 25%, where is the second best model that is used to determine the land use and the agriculture. The conceptual or numerical model is the third applied model, of which is mostly used into the data cast. However, why is calibration and validation is important? The first diagram shows the different methods of which are used widely or globally for calibration. The study showed that there's a global trend towards the automatic calibration. However, the manual calibration remains essential for integrated hydrological modeling. The automatic is because it's effective and it doesn't require so much time. And the validation techniques, why? Why are we supposed to validate our models? This is to ensure that there is a robust and reliable model results. Many studies have showed that the performance matrix is the most preferred technique. What are some reviewed key findings of the integrated models? The MACSHI, which is the most preferred, the studies have determined that they tend to capture full hydrological cycles where there's groundwater and surface water interactions, recharge, and overflow. The SWOT mode flow that tends to provide a better base flow and recharge simulations compared to only just using a SWOT. Path flow, it tends to resolve distributed groundwater and surface water processes. GS flow, it is strong in the water budgets and it provides long-term water climate change impact. The cross-model impacts, the studies have shown that all integrated models, they tend to improve groundwater and surface water understanding. But we are here now. We need to identify that throughout the different models which have been reviewed, which one is the best? The fully coupled model has proven to be the best and the most recommended because it provides the most comprehensive process presentation and is mostly used globally in countries such as Europe, Africa, Australia, and also Asia. The second best model is SWOT mode flow because it can be able to simulate complex coupled hydrological processes accurately. In conclusion, all across reviewed models, the integrated approaches are found to be significant just because it improves the understanding for groundwater and surface water interaction. The key takeaway, there is no single model that is sufficient enough to give the best results. So we need to couple our models to have better results. So the recommendation of this study is that we need to invest into improving our data availability and promote wider applications. And the second recommendation is to improve collaborations and capacity building. These are some of the selected references. And last but not least, thank you. We're the champions here.
Oh, unfortunately, I can't see the projectors, but I can see what is being shared now. All right. With that being said, I think you gave us a lot of time for questions. I was judging you by Dr. Mbanjwa and there. And then I will then take questions, 3 if possible. Any questions? Hands? I know this is the graveyard slot, but give us some questions, please. There's one question there. Okay, there's a second question there noted.
Good day, ma'am. Can you kindly elaborate or what is your scientific contribution on your study?
Actually, this is a systematic review. So I'm reviewing different studies which have been conducted or published. So what we're trying to do, I'm trying to understand how does groundwater and surface water interact so that we can be able to improve our water management and water security. So it's a review. It's not my actions.
What is your scientific contribution? What does it contribute in this field of study?
Yes. So how it contributes. Actually, the aim is to shift more into our focus in the use of using coupled methods instead of only one model. Because if we're using only one model, we don't get accurate or best results. Yes. So if we're using coupled methods, then can be comprehensive or extensive, but the results are reliable. Yeah.
Hello. Okay. Thank you for the nice presentation that you have delivered to us. So I wanted to ask, have you ever considered, like, since you're dealing with models, have you ever considered or look along the WIP model? Even when you are choosing the model, like what drives you into choosing the models and come to a conclusion whereby you couple them?
Yes, I think the focus of a research or the theme is important because we have thousands of models, right? However, we can also be based on the methodology that have been done and check the gaps that are there to fill. And the gap which have been identified is that there is limited of knowledge and there is lack of studies where they tend to couple different models to get the results. So we only focus on the weak models to get results. And they're not accurate enough, they're not reliable. So coupling them, it tends to be more of a best option. Yet it needs a lot of computational data, but it is highly recommended, more especially in the research space.
Okay, we still have one more hand here. If you can just wrap up within 2 minutes, I'll be glad.
Hi.
Hi.
Good afternoon. It was a nice presentation, but I've got just one question for you. You talk about a database on your presentation. Where does it fit in this groundwater?
The database?
Yeah, there's somewhere where you spoke about the database. So I don't know where does the database come from because it's all about the models. So now there's a database in some of your presentation.
Okay.
Or maybe I missed.
Yeah, please stop me. I'll just go back and then you can tell me, right?
No, there was some slide where you mentioned some database, so that's why I recorded it down. Or maybe I just misread it.
Okay, I'm not sure. And however, if maybe I spoke about it, but I'm not sure. But maybe it's definitely the data that was obtained from the reviewed studies. But yeah.
Let us clap hands for Matilongi. Thank you so much. I still don't understand how this study related to Orlando Pirates, but oh well, maybe that's just me. Our next speaker is Lydia Langa, who is speaking on Moringa and agri-silviculture systems.
Okay, good afternoon, everyone. I'm Langa, RL. I'm going to take you through the title, The Performance of Vegetables under Moringa Oleifera Agri-Silviculture System and Fertilizer Application. I'm a PhD candidate, and this topic is part of my current study. Under the supervision of Prof. MP Upape Mabapa, Prof. P.E. Dlamini, and Prof. A.R. Nhlala. Ladies and gentlemen, this is my outline for the presentation. Ladies and gentlemen, we are facing an increase in population rise, which means that there is food that we need to produce to feed the increasing population. Therefore, we need to also adapt or change methods of cultivation or productivity, such as agri-silviculture. Whereas we know that normally here in South Africa, we are using monoculture every year as part of productivity, to increase productivity. But then this study focuses on agri-silviculture, which is the integration of trees with agricultural crops. The trees that we'll be focusing on here is Moringa oleifera, which is a fast-growing tree that it also has the benefits, the medicinal benefits, and also the ecological benefits. Moving on to Swiss chard. Swiss chard is a spinach. Normally we call it spinach. It's the leafy vegetable that forms part of our dietary benefits every day that we normally consume it. And also the Cucurbita moschata, which we normally call it butternut, is mostly recognized as a colorful morphological varied fruit or is a vegetable, which is produced in a broad variety forms worldwide. The aim of this study is to investigate the performance of vegetables under Moringa oleifera agri-silviculture system and fertilizer application. The objective of the study is to determine the effect of Moringa oleifera agri-silviculture system and fertilizer application on the performance of vegetables. The hypothesis align with the objective. Moving along, the study was conducted at University of Limpopo Green Biotechnologies Research Center with the coordinates stipulated, and then it was conducted under moringa trees which were planted in 2019, and the precipitation in Green Biotechnology Research Center is ranging from 450 to 750 millimeters with a temperature of 18 degrees to 24 degrees Celsius. Randomized complete block design was used in this current study with 3 treatments. Treatment number 1, soil vegetable; 2, fertilized vegetable; 3, vegetable planted under moringa. No pesticides were used during the experiment, and each plot will consist of 6 rows. Butternut and spinach were planted with an inter and intra-row spacing stipulated in table 1. And also, fertilizer that was used, it was potassium ammonium nitrate according to the recommendation rate per crop. Soil samples were collected before planting randomly in an experimental field so that physiochemical properties of soils can be measured. The physical and chemical properties that were measured are stipulated and in table 2. And also the gravimetric soil moisture was collected on bi-weekly basis. Ladies and gentlemen, the parameters collected here were chlorophyll content, plant vigor, plant height, biomass, leaf area, radiation use efficiency, number of branches. Yield parameters of butternut were failed to be collected due to accidental harvest and damage caused by monkeys. The analysis of variance was conducted using statistics 10.0, and list of significance was used to separate the means at a 5% probability level. The 2 figures stipulated here are the results for moisture content that was collected on bi-weekly basis. As you can see, according to the treatment effect, that the highest moisture conservation was obtained under moringa agri-silviculture. Therefore, the results are in line with De Cavallo et al. 2021, which reported the same results in response to moisture conservation. The soil samples that were collected They were collected before and after according to the treatment effect. Ladies and gentlemen, as you check here on table 3, that the pre-plant soil analysis according to the table, as the table is stipulating here, the highest nutrients are found in agri-silviculture system. This table was collected on soil properties under butternut plantation. Then in table 4, it shows the physiochemical properties before and after planting in butternut productivity. Also, the same is happening here with the nutrient content. As you can see, that more nutrients are found under moringa agri-silviculture. With response to the results, the performance of spinach in fertilizer application, as you can see, the highlighted results here, it simply means that there was high performance under fertilization application. And then the current study is in line with the study of Hassan, who reported better performance of baby spinach as influenced by NPK fertilizers. Table number 6 represents the performance of butternut in fertilizer application and moringa oleifera agriculture.
Just speak to the mic. You're speaking to the side of the mic.
Oh, sorry. Okay, table number 6, it represents the performance of butternuts as influenced by fertilization and agri-silviculture system. Same results here under butternut. You can see that the more performance we got it from the under fertilization application. Maluke et al. Also reported the high performance of butternut under organic and inorganic fertilization. In overall, fertilizer application played a role in vegetable production compared to agri-silviculture system. Therefore, this study rejects the null hypothesis. And also, agri-silviculture system can be recommended to smallholder farmers due to the ability to conserve water. And also, as you saw on previous results, that in agri-silviculture system, the nutrient contents, they tend to be high under moringa agri-silviculture system. I would like to acknowledge my supervisors, University of Limpopo, for giving me an opportunity to conduct this study, and also the funding from FoodBev. Thank you.
Thank you, Ms. Langa. You are very timely. Can we get hands for questions? It seems they are scared of you, Ms. Langa. Just one question. Did you say that? Oh, there's a question there, please.
Okay, thank you. What a good presentation you had there. I just want to, can you go back to your data collection?
Which slide?
Of your data collection.
Data collection. This one.
The previous one, under your methodology. This one. Okay. Looking at your data collection, I do not see your soil analysis data collection, but your result, you start by telling us the findings of the soil, nutrient, and the different plant that you have used, but under your data, it was not stated. And also, I was interested to see how did the spinach perform compared to the moshata and the under the moringa tree, because the moshata is a trailing vegetable, and then the spinach also will want to utilize a lot of sunlight, but pairing the three together, how is the biomass and the yield, the vigor? So I was interested to see that. Not the soil nutrient and also the other thing, but their productivity. Concerning the yield of the mochata, you said monkeys affected your yield, but what about the yield of the spinach and, I don't know, the moringa? So we were interested to see about the yield of those two other crops when you plant them together.
Okay, thank you for the question, Dr. Maila. With response to yield on spinach, normally biomass was collected bi-weekly to measure, like, the leaves were harvested, and then the biomass was recorded from the leaves on bi-weekly basis. So biomass, it serves as a yield, because on spinach we normally target the leaves. I don't know if I was able to answer your question.
What was the yield? The yield, was it high or low under the moringa of the spinach or the moshata?
The biomass was high under fertilized plots. As we know that nitrogen is as we know that spinach normally relies on fertilizer so that it can really grow vigorously. So these results were obtained. The highest biomass, which was 340.47, it was obtained from fertilized plots where spinach was planted. Thank you.
Thank you.
Sorry, Ms. Lang. Oh, my bad. There was one more question around soil analysis. I think still from Dr. Maila, just to respond to it before we close.
Can you please rephrase the question? Was it a question or a comment?
The question was, under your data collection, there's nothing of soil, but under your result, you give us the whole result of the soil nutrient under the three crops. So it should be included in your data collection.
All right, thank you for the comment, Doctor.
Okay, I will take it that that was you responding to that question.
No, I think Dr. Maila wanted to know that since the data collection is stipulated here, then I started with I should have started with soil results. Is it? Yes. Okay, thank you. I will consider it next time. Thank you, Doctor.
Okay. Thank you so much. I think Mopai is our next speaker. Thank you, Mr. Okay. Can Mopai come to the front? You're not Mopai. Okay. We're just having some issues with loading here. Whilst Mopai is loading, I will fire one more dry joke, and this one is academic in nature. That's just to keep you guys alive. So 2 weeks back, I was attending the NRF Next Emerging, Next Generation Researchers Symposium. I don't consider myself emerging anymore, but they called me. One of the things that I put there, up there that I like so much, there was a meme where a scientist was saying that, here is this perfect innovation that works all the time, does all the things that we found. Now we just have one problem. We need to now find a realistic problem that goes with it. So that's just to remind whoever is presenting here today that we are looking at the societal impact of your research as well. If you can lean towards also the economics of your research as well, please try and do so. Otherwise, that question remains elusive. One more thing that I realize is that the previous chair did not introduce themselves, so that was Dr. Ledwaba. I also did not introduce myself in the same vein. I'm Sandile Hadebe. I think the reason why I did not introduce myself was that because there was Prof. Masugu there, so I was trying to look timely. Otherwise, Mopai, are you ready? Okay. Just a reminder, if you are the following speaker, please go check if things have been loaded properly. And whilst the questions are starting, the following speaker, we're asking that they come closer. If possible, you can start even like moving towards the front benches if you're at the back. Thanks. Do you mind if we get another speaker? Do you mind if we get another speaker and then we follow? This one? Yeah. Good. Okay, good. Mr. Mpai will speak to us about drying method and plant extracts. Thank you.
Good day, everyone. As introduced, my name is Mpai Class, a PhD candidate here at the University of Limpopo, and I will be taking you through part of my research, our research projects, which goes by the title Phytochemical Screening, In Vitro Antioxidant, Anti-Diabetic and Anti-Obesity Activity of Viscum Compressed Cola Extracts, subjected to different drying methods. This is the table of content that I will be taking you through. I believe we can agree that since antiquity to present, plants, particularly medicinal plants, have been the basis for human health and well-being. And measle 2 species, which is Viscum compressed cola, is not an exception. So one might ask, what is Viscum compressed cola? Briefly, Viscum compressed cola is an evergreen, leafless, epiphytic, hemiparasitic plant that mostly is found in the tropical regions of Africa, and it belongs to the Viscaceae family. Its extracts have been used in the traditional medicine system for treatment of various ailments, such as, but not limited to, your epilepsy, mental illnesses, hypertension, and inflammation. That is probably because of it is rich in diverse bioactive compounds with strong antioxidant effects. And one of the problems is that there is insufficient research and documentation of this measle 2 species. Hence, its values and applications remain indigenized and common knowledge to few, particularly herbal practitioners and our sangomas. In numerous industries such as traditional medicine, ethnopharmacology, food and beverage industry, pre-treatment and preparation is one of the critical and fundamental stage. Hence, it determines the quality of the final product of the derived extracts from the medicinal plants. And one of the mostly widely used common pre-treatment and preparation method is drying process, which simply occurs by removing the moisture from the plant material so that we can inhibit microbial growth and continuous metabolic and enzymatic processes which might lead to undesirable chemical alteration or degradation of our extracts. And therefore, by inhibiting that, we might enhance the efficiency and efficacy of the extracts. This study envisioned to investigate the effects of different drying techniques on phytochemical compounds of Viscum combreticola, the antioxidant effects and potential ethnopharmacological applications for treatment of diabetes, type 2 diabetes and obesity. I'm sorry, I don't know what is happening with the pictures. Yeah, but let me continue. The stems or nodes of these plant species, Viscum combreticola, were harvested or collected at Tombisilica College of Agriculture here in Limpopo, and the voucher specimen for future reference was submitted at Larry Leach Herbarium. The harvested or collected plant material were then dried using 4 drying methods, which are sun, shade, solar, and oven, following the stipulated time period as well as temperature and relative humidity conditions. We then grounded the dried plant material into fine powder for biochemical analysis, whereby we looked for phytochemical compounds that might be present in this plant material, whereby we tested for alkaloids, flavonoids, glycosides, quinones, and anthraquinones, so forth and so on, following the cited methods by the authors that I have cited. We further did in vitro antioxidant assays to check the antioxidant properties or effects of this plant material, and then we followed the DPPH and ABTS assay as well as FREP assay as described by the cited authors and with minor modifications. Since we have a problem of diabetes and obesity, these are one of the public health concerns. So we thought of doing we thought of checking if this plant material or the extracts can be used in treatment of diabetes as well as obesity, whereby we followed the alpha amylase assay as well as alpha glucosidase assay to check the anti-diabetic activity of Viscum combreticola extracts. And then finally, we did the in vitro anti-obesity activity, whereby we followed the lipase assay as described by Kim et al. 2010 with minor modifications. This study adopted a one-way analysis of variance, and then Duncan multiple range test at 5% significance level was used to separate means using a computer statistical software, GenStat, and then GraphPad Prism was used to draw our graphs. Table 1 shows the phytochemical testing of Viscum combreticola extracts subjected to different drying methods, whereby we followed the stipulated chemical protocols in our second column. From the table, we can see that solar-dried extracts as well as sun-dried extracts were rich or showed abundance of alkaloids, flavonoids, phenolic compounds, steroids, as well as glycosides and quinones and anthraquinones. And in contrary, oven-dried and shade-dried extracts, they showed least presence of alkaloids as well as tannins. There was no significant difference between the terpenoids as well as saponins that were observed in all the treatments. The abundance of solar-dried and abundance of phytochemicals in solar-dried and sun-dried extracts may be due to a rapid inactivation of enzymatic processes as well as metabolic processes that might lead to phytochemical degradation as well as alteration. And in contrary, the least abundance of alkaloids and other phytochemical compounds in oven-dried might be due to exposure of plant material to high temperatures that lead to degradation of high-sensitive compounds. We further did an antioxidant effects to see the antioxidant capacity or activity of these extracts, whereby we did the DPPH, which is graph A, which shows that there was no significant difference between the DPPH content that was exhibited by oven-dried, shade-dried, as well as sun-dried. And going to FREP, ferric reducing power, there was no, the sun-dried extracts exhibited the highest FREP content compared to shade-dried, oven-dried, and solar-dried. Going to ABTS content, again, sun-dried extracts exhibited the highest ABTS content, but then there was no significant difference between the oven-dried, solar-dried, and sun-dried ABTS content. We did the, as I've mentioned, the alpha amylase inhibition activity to see if these extracts can be used to inhibit the breakdown of starch into smaller molecules. And then from the graph, we can see that at concentration 100 microliters, the extracts were able to inhibit the alpha amylase activity compared to the control. And the highest inhibition percentage activity was recorded in sun-dried extracts, followed by solar-dried extracts. In contrary, the oven-dried and shade-dried, instead, they were enhancing or accelerating the amylase activity. This corresponds with the phytochemical compounds results that I have shown prior here, whereby the alkaloids, flavonoids, as well as terpenoids are responsible for the inhibition of the alpha amylase activity. We further did the alpha glucosidase inhibition activity since the breakdown of starch into glucose occurs in two phases, and we found that sun-dried and solar-dried extracts showed the highest inhibition activity at 600 microliters. We also did these are some of the research gaps, and these are the future prospects. This research study aligned with the 3 SDG goals, reference list, and then I would like to acknowledge these entities, and I thank you.
Let me go to the mic. Okay, Mr. Mopai doesn't want to give me back the mic. I think he doesn't like the way that I'm chairing. Sorry, Mr. Mopai. It was strict instructions from Prof. Masugu in terms of time. Okay, I've reserved about 5 minutes then for us to be able to ask questions, including some of the missed sections. If you didn't see them, maybe we can use it for that. Can I see hands? There's a hand there at the back. Please just keep it up. Oh, okay, they've seen it. Okay, there's a second hand as well. I've noted it.
Afternoon, sir. I saw you presented SDG goals. So I want you to help me navigate which SDG goals are you mainly focusing on?
SDG goal number 3, 12, and 15.
Okay, so I'm not sure if, can I be able to also navigate it at SDG goal number 6?
which is?
Clean water and sanitation.
Sorry for interjecting. Can I just ask that you ask a direct question, especially with time limitations? Thank you.
With that one will take time, so, and on this one of SDG 3. Thank you, sir.
All right. Okay, and that's just to everyone. My apologies. If you're going to ask, I'm just asking that you go direct to the question. Again, it's on behalf of Prof. Masuku. These are not my rules. Yeah, thanks.
Thank you, sir, for the nice presentation. My question is on the moisture when the drying process. You said you were drying the plant. So my question is, what's the effect of the moisture on the plant? Why are you not using it as it is? You are drying it.
Drying is a technique that is used to preserve or prolong the shelf life of the plant material. Hence, I've said that moisture in the plant, it will result into microbial infestation or growth, as well as continuous enzymatic processes and metabolic processes that might lead to phytochemical degradation or alteration, and that is not what we want for the plant to be effective.
Thank you. I didn't get that point when you presented.
Thank you.
Mopa, you look like you want to fight me for the mic. When I come to the mic, you must move. I'm chair for a reason. Okay, it looks like we no longer have any hands, and that's good. Colleagues, the other reason why I'm being this prompt with time is because if you look at our program, we're supposed to finish at 6 p.m. And then we are 3 hours behind schedule. So any time we can save, I'm trying to make sure that we save it to your benefit and mine. The next person who is supposed to be presenting, I'm not sure where they are. Brilliant Lope. And I'm asking that whenever you know you are the next speaker, you start coming to the front as the questions are. Oh yes, please clap hands for Mr. Mopai. For speakers, please adjust the mic to your preference if you are about to speak. Thank you. Okay, Hlope will be speaking to us about wastewater use in agricultural systems. Please take it away.
Good afternoon, everyone. My name is Hlope Brilliant, and I'm currently busy with my second year of Masters in Agriculture, focusing on soil science. And what I'm about to present is part of my master's work, which focused on evaluating the quality of treated wastewater for irrigation. But for the purpose of this presentation, I'll just be focusing on evaluating measured cations and anions in treated wastewater for agricultural reuse, which was a case study of Capricorn District, co-authored by Dr. Khopa, Prof. Mpatlele Mahwane, as well as Dr. Masebe. Water scarcity has worsened throughout the years, more especially in arid and semi-arid regions, and this is driven by factors such as climate crisis, which led to the uneven distribution of rainfall, such that other areas receive more rainfall as compared to other areas, population growth, which increased the demand for water, as well as industrialization. In order to mitigate the effects of water scarcity in agriculture, treated wastewater then comes in as a strategy. However, treated wastewater also comes with its own constraints. For example, its quality varies seasonally, mainly due to the fact that different volumes of treated wastewater are produced in different seasons. which then creates operational issues, as well as capacity constraints. Additionally, the limited infrastructure as well as the high costs associated with maintaining the current infrastructure of treatment plants may also alter with the quality of treated wastewater. Hence, the study aimed at assessing the chemical suitability of treated wastewater for irrigation as compared with tap water, which was a case study of the Capricorn district. This study was conducted in the Capricorn district, which is characterized by an annual temperature that is between 5 and 29 degrees Celsius, and annual precipitation that is between 400 to 500 millimeters, as well as humidity that is 30% in winter and 40% in summer, classifying it as being a semi-arid environment. The study was conducted in a 5x3 factorial experiment where the first factor were the treatment plants, and the second factor were the sampling month, where water collection occurred on the 15th of each month in 3 replicates. The pH of the water as well as its electrical conductivity, the cation ions as well as anions were analyzed in the water. All data were then subjected to analysis of variance using Genstat 18th software, and to his honest, significant difference was employed to assess the mean difference at a significant level of 5%. The results revealed that the treatment plant, the sampling month, and their interaction had a highly significant effect on the pH of the water, its EC, the concentration of all measured cations, which consisted of calcium, magnesium, potassium, and sodium, concentration of chloride, sulfate, phosphate, and that of nitrate. However, the concentration of fluoride, the interaction between the treatment plant and the month of sampling had no significant effect, but the treatment plant itself had a highly significant effect, with the month of sampling also having a significant effect. The pH of the water fell within the FAO as well as the SA range, that is between 6.5 to 8.4, except for one occurrence for tap water that was collected in April, which exceeded both ranges. The pH of the water consistently remained higher in tap water as compared to treated wastewater across all treatment plants, and such an observation may be attributed to the fact that tap water and treated wastewater are treated differently. Tap water is treated for drinking, hence it involves processes such as pH adjustment, which involve basic disinfectants such as lime, which will then increase its pH. But when we now look at the treatment plants in question, they lack tertiary treatments that would then involve the use of such disinfectants, hence the pH remained lower in treated wastewater as compared to tap water. Coming to the EC of the water remained low in tap water as compared to treated wastewater, and it was about 15, 27, 27, and 57 times less than that of Mankweng, Lebuakgomo, Polokwane, and Bochum. Even though the pH was lower in tap water as compared to treated wastewater, all samples were within the standards recommended by FAO as well as SA. Such water were then classified as excellent, meaning that it poses no restriction for irrigation. However, such findings contradict with those found by Mosau in 2023, whose water fell in the highly saline category. Such contradictions may then be attributed to the fact that treated wastewater is not the same, and treatment facilities are also not the same. When we now look at the concentration of cations, starting with sodium and calcium, both of them were lower in tap water as compared to treated wastewater, and both of them were still within the thresholds recommended by the FAO, meaning that irrigation with such water would cause low risk of sodicity in receiving soils, and also this would be an advantage because calcium promotes soil aggregation. When we now come to the concentration of magnesium, all samples were within the 60.75 milligrams per liter threshold, except for Bochum treatment plant, which exceeded the threshold throughout the 3-month period. And irrigation with such water is a risk because it would degrade soil structure, reduce its permeability, and also lower crop productivity. When we now look at the concentration of potassium, only tap water remained within the 2 milligrams per liter threshold, with treated wastewater across all treatment plants during all the 3-month period exceeding the threshold. And this irrigation with such water would also be a risk because it would lead to a build-up of potassium in the soil, thus reducing its hydraulic conductivity. These findings align with those found by Mulizi et al. In 2015. Continuing with anions, the concentration of fluoride was well within the FAO and the SA range, which is 1 milligrams per liter and 1.5 milligrams per liter, respectively, meaning that this irrigation with such water would not pose any risk for the receiving source. When we now look at the concentration of chloride, all of them were within the SA range as well as the FAO range, except for one incidence in Bochum collected in May, where the concentration of chloride was slightly higher than the recommended threshold of South Africa. This irrigation with such water would then be a risk because chloride is only an essential nutrient at low concentrations, but if it happens that frequent irrigation occurs with such water, it would then accumulate in the soil, posing risks, especially to chloride-sensitive crops, leading to leaf burn as well as reduced yields. The concentration of phosphate in the water was only within the 2 milligrams per liter limit for tap water, while treated wastewater across all treatment plants for the 3-month period exceeded the threshold. This is attributed to the fact that the treatment plants in question are poorly managed because phosphate has a tendency of evading biological treatment processes which are not even carried out in the treatment plants in question. Coming to the concentration of nitrate, the concentration of nitrate was also within the thresholds only just for tap water, but for the treatment plant, for the treated wastewater collected across all treatment plants, it was exceeding the thresholds, meaning that irrigation with such water would then lead to leaching issues as well as over-fertilization. Even though tap water was the only one that remained within the threshold and it was the only one that was considered safe for irrigation, irrigation with such water would then need the supplements of fertilizers in order to meet the requirements of the crops. These findings contradict with those found by Mosau in 2023, where the nitrate levels in treated wastewater remained well within safe limits, emphasizing that with optimized treatment systems, the level of nitrate in treated wastewater can be brought back to meet the threshold required. Last but not least, the concentration of sulfate remained within the threshold suggested by the FAO as well as the SA threshold, meaning that the water is safe for irrigation and it can act as a source of sulfur for crops, more especially for the treated wastewater from the treatment plants because it was high as compared to tap water. It can therefore be concluded that the treated wastewater contains elevated levels of cations and anions as compared to tap water, and such a concentration varies both spatially and temporally. Treated wastewater can therefore be used for irrigation, provided that these management practices are taken into consideration, blending the water with water that has less concentration of these ions, continuous monitoring of soils that are irrigated with treated wastewater, as well as considering upgrading the treatment plants. I would like to thank the University of Limpopo for allowing me to conduct this study, as well as the National Research Foundation for founding it. These are the references that were used in compiling this presentation. Thank you.
I think, Ms. Lope, you should also thank me for giving you 1 extra minute. You were chowing into my time there. Okay. That means that we've got about 4 minutes for questions. There's a question there. And can I see hands for other questions, please? Maybe one more. Okay, one should be enough then in this case.
Can I talk? Oh, hey, I can. Hi, how are you? This was a very nice presentation. I just wanted to confirm, I heard you talking about the FAO standards and the SANS. Is that which one is the South African one?
The South African ones are the ones that are set by the Department of Water and in South Africa.
Yeah, like which one is it? Like, is it the national drinking water? Which one is it?
It's for irrigation.
It's for irrigation?
Yes.
Okay, that's perfect. I just wanted to confirm that.
No, it's for irrigation.
No, it's okay. Thank you.
It seems like Ms. Lope, you were very thorough in your presentation. So can we clap hands for Lope? There's a key change that's happening now. You will know that I had a co-chair to begin with, and he will not be a co-chair if he doesn't stand in front of you. So what that means is that you'll hear another voice now here at the front. For those who are now presenting, it means if I stand where he's sitting now, you've got 9 minutes, and we're asking that in the interest of time, you move on towards your conclusions whenever then you see someone standing on that. And I need to apologize to you for this hoarse voice, Shaka Ilembe type of voice. I'm a bit fluish. I don't have flu, I'm a bit fluish. I went to the coast, and then I came back here, so going to a wet environment, then coming back to a dry environment, there's a bit of an adjustment that I have to go through. Dr. Mbanjwa, please assist. Please clap hands for me. I don't think I did a bad job.
Documents. Yes, it's okay. Kuduveli. I know it's difficult to no, this one is the new version. This one is the old version. The old one is called SWAT, and then the second one is the upgraded version of the SWAT plus. It's called SWAT plus. Interesting. Let me see. Good way. I think you put it on documents. Put it on the USB. Okay, no, it's fine. We can put it. I can't find it. The presentation. Yeah, this is the one.
Good afternoon, ladies and gentlemen. I have been introduced, so I won't be repeating that. I'm only requesting the 3 other presenters, Khapane, Amokelani, and Mary, to be uploading their slides in the meantime if they haven't already. So our next presenter is Mutali Kuduveni who will be talking about advancement in the application. If you look at the title, there are two double SWOT. I think it means whatever he will present is a double problem. But let's hear from him.
Good afternoon, everyone. My name is Mutari Guduveni, and I'm going to be presenting on the topic of advancement in the application of SWOT and SWOT Plus in modeling wetland hydrology, implications for Southern Africa. So this presentation is based on a systematic review. So my supervisor is Prof. F.I. Matiba, co-supervisor Prof. H. Chikori. So wetlands provide essential ecosystem services, including water regulation, pollution filtration, erosion control, biodiversity support, and socio-economic benefits for surrounding communities. So these valuable ecosystems are increasingly threatened by land use and land cover changes, which include agricultural expansion, urban development, and infrastructure construction. So to gain more insight into the water systems, hydrological models should be employed as a tool. So these models help researchers obtain information on various aspects, such as hydrological fluxes, trends, and the impacts of climate change and land uses. However, in some cases, models are applied to estimate sediments, yields, nutrients, fluctuation, and land use and land cover changes, with little knowledge on the hydrological processes of the study area, which influences how well the system can be represented by the model. So in this review paper, or maybe a review study, we're going to be using SWOT and SWOT Plus for this review. So the methodology that we used include journal articles, identification, screening, and assessing. So the first thing that we did, we identified 84 articles, and then we ended up with 31 review articles. Due to the fact that we had to screen them, where we found that we had to remove 11 non-English journals, and then we also assessed for their eligibility. Then we found that we were left with number 39. So if you check on my figure 2, this is just showing you the yearly distribution of the extracted data for my systematic review. So the review findings for my review study shows that the geographical distribution studies they were in North America, Asia, Africa, and Europe, showing that in North America, that's where there was a huge distribution of these studies being conducted. And then if we check, so these reviews included the time frame from 2008 to 2025. And then if you check 2020 to 2025, there was a huge shift in using this model, showing their efficiency. So in terms of the calibration techniques that are normally used for this type of model, we see that SUFI2 is the one that is normally being used, and then there is also SWOT-CAP for this calibration. And then if you check distribution model types that are normally used, we see that the SWOT is the one that has been normally used because it's the first one, but now 22, it is showing that SWOT Plus is also emerging as one of the models that are being used. So the type of wetlands that are normally being studied or being modeled, it shows that the riparian wetlands are normally the ones that are being mostly studied. This is due to the fact that there's a good connectivity between the upland areas and riparian zones, made them particularly suitable for representing using SWOT Plus. So, and also we see that also depression wetlands, we can get references from prayer, potholes, vernal pools, but on this one we needed to modify the SWOT So there were other studies which were conducted in North America. So this is the, underneath is the reference where you can get the study for that. So we also got groundwater fed wetlands. These ones need require aquifer modification, which can also include swat mode flow. So we also had estuarine wetlands. This one was coupled with hydrodynamic models. So cold and plastoran wetlands also, as also the other presenter talking about modeling, she spoke about coupling of the models. So here we see that some of these estuarine wetlands needed coupling with hydrodynamics models. So implication for wetland management and modeling in Southern Africa. So the application of SWAT across Africa has shown strength in estimating freshwater availability at regional scales, but challenges remain in accurately representing wetlands and dam-regulated subbasins due to data scarcity. Lessons from global studies are highly relevant, such as Canadian case where a modified SWAT model captured groundwater dynamics and CO2 emission, offering approaches applicable to peatlands and high-altitude wetlands, which will be similar to the ones in Lesotho and eastern South Africa. So adaptations of SWAT to simulate base flow and groundwater upwelling in Alabama provide useful methods for modeling groundwater-fed wetlands, similar to the ones that are found in KwaZulu-Natal. So a key limitation for Southern Africa is the lack of reliable calibration data, which reduces model accuracy and effectiveness. So in my conclusion, so we managed to review 31 studies applying SWAT, SWAT Plus, and modified SWAT to wetland hydrology. SWAT remains widely used due to accessibility and long-standing application. Shift towards SWAT Plus due to its emphasis on the connectivity of special elements with watersheds such as hydraulic response units, aquifers, channels, reservoir, ponds, canals, and pumps. SWAT Plus can better simulate the riparian zones, floodplain dynamics, and wetland river interactions. Due to the impact of anthropogenic activity and climate change on water systems, modeling of the wetlands will assist in providing scientific base for future water management and wetland management. With the inclusion of landscape units, SWAT Plus offers an improved spatial heterogeneity, making it more adaptable than SWAT in depicting the interaction and processes occurring within a watershed. So these are the selected references. Thank you.
Thank you very much, Guruven. Can we get questions for the gentleman behind me regarding SWAT? I just hope whoever is asking is not from economics. We're not talking about SWOT analysis.
No,
SWOT.
no. Thank you for a wonderful presentation. On the results section, where you presented about, I think the continents there.
Oh, yes.
So I was asking, why couldn't you just, let's say, put the countries so that we get to know which country specifically, let's say, has much publications as much as SWAT and SWAT Plus is concerned?
Yes, I had it on my review paper, but I just decided to just put the continents. But one of the places, it was Canada. It is one of the places where they have a lot of studies that side.
Okay, thank you.
No, man, thank you for that.
Thank you, sir.
He gave us enough time for questions, so we can still accept 2 more questions for him.
Thank you so much for your presentation. You presented something very interesting around calibration.
Yes.
I think you had 5 calibration methods, and then you had other.
Yes.
Do you mind just going to that slide for me? I'm asking that you act like the president here and take us into your confidence to tell us what are those calibration methods, how do they differ? Because as part of your conclusions, you said to us that there's lack of calibration data. And we also know that when you're calibrating, sometimes there are data requirements for each model. So can you speak to those data requirements as part of taking us into your confidence? Thanks.
Okay, normally for SWOT and SWOT Plus, we normally use NSE and then also the R squared methods for calibration. Yes, so those are the others.
Sorry, just take me into the 5. I'm not really speaking to the others. What are the differences in terms of data requirements between those 4 and 5, and why are there 5 to begin with? Why isn't it just one calibration method? I'm asking as a layman here. I'm not into water modeling.
Oh. No, okay, maybe I will start by manual calibration, one. Manual calibration is the one that normally will assist us because you can easily manipulate the whole system where you can even change different kinds of things. So, and then we can have, so yeah, that is the one that people normally use. But the reason that we have to use different kinds of calibration is because we want to check if maybe the data that you got is giving you the same results. So these calibrations, they do the same thing, but it is always good for you to use different calibration methods. Yes. Yes.
I think I will have to bail him out in the absence of further questions. Thank you very much, Kuduveni, for sticking to time. Thanks.
Thank you.
Let's give him a round of applause.
There's also another one.
The next presenter is Mahatsela Kapan.
This is the presentation.
For her?
Yes, for her.
Okay, that's good.
I can take it off now. Did you copy it? I can take it off. Okay, no, it's good.
Which one? The silver one.
That one. Okay, she is going to speak to us about flood risk and vulnerability at Hamotiba. I've always thought I'm safe from floods because I'm a KZN boy until I see her title. Over to you.
Thank you very much for this opportunity. Good afternoon, everyone. I go by the name of Mahatsela Jessica, and today I'm going to be taking you through a review of flood risk and vulnerability in peri-urban settlements of South Africa. I'm supervised by Professor F.I. Matiba and co-supervised by Mrs. M.C. Motabo. Just to give you a bit of a background of my review, flood risk remains one of the most destructive natural hazards globally, with an increasing frequency and severity in observed recent years. In South Africa, peri-urban settlements faces heightened vulnerability due to informal housing, inadequate infrastructure, and limited access to resources. This attached video was taken from Ramutiba. It is a visual representation of the devastating impact of floods on communities with poor drainage systems. It serves as an important reminder of the urgency to address this flood risk in these areas. The aim was to review flood risk and vulnerability in South Africa with a particular focus on peri-urban settlements, and its objective was to analyze key factors influencing flood risk and vulnerability in South African peri-urban settlements to guide effective flood risk management. A systematic literature review was conducted using academic databases such as Google Scholar, Web of Science, and Science Direct. Peer-reviewed journals were selected based on such terms such as flood risk, flood vulnerability, peri-urban settlements, and South Africa. The Boolean operators were applied to refine the search, and references were reviewed to identify any further relevant additional sources. The publication date range was set from the year 2000 to 2025. This was to ensure recent relevant study selection. The included sources were the ones that focused on flood risk, flood vulnerability, peri-urban settlements, and South Africa with an empirical data. And excluded studies comprised of studies that were focusing on other hazards, studies that were not accessible, and publications that were lacking empirical data. Well, this was to ensure a comprehensive and most relevant selection of literature. From a pool of 111 sources that were identified, duplicates, irrelevant, and non-South African related sources were eliminated, which resulted in a final selection of 84 sources that were used in the literature. Titles, abstract, and full context were screened to ensure the alignment with the inclusion criteria. From the findings, it was found that a flood risk in peri-urban settlements, it is shaped by 3 core components, which are exposure, hazard, and vulnerability. These components, they do not work in isolation. Instead, they enforce one another. Figure 3 here represents, this figure here shows how these 3 components overlap with one another. Where there is high exposure, a severe hazard, and a deep vulnerability, risk is very critical. For example, a household that is located on a floodplain area with a severe hazard, with a poor drainage system, faces a greater level of risk. So to address, flood risk is not only about the accumulation of water, but it's also about who is affected and how severe is the hazard, and also how well can the communities cope or recover from the flood events. So to address flood risk, all these 3 components will have to be addressed simultaneously. Extreme rainfall, poor soil characteristics, and flood-prone topography were identified as key environmental drivers. The review revealed that in Limpopo, there often be intensive rainfall, intensive and unpredicted heavy rainfall, which increases this hazard. When this combines with a poor soil absorption and flood-prone topography, it heightened the hazard component of flood risk. Informal houses and high levels of poverty were identified as critical key socio-economic drivers. Many residents in South Africa live in flood-prone areas with a poorly constructed households and lack of access to resources. This decreases the ability of the communities to prepare for, respond to, and recover from floods. On the other hand, poor drainage systems was identified as a key institutional weakness. The review revealed that in many peri-urban settlements, the systems are either blocked, insufficient, or entirely absent. So during heavy rainfalls, water accumulate, and the communities faces a more severe impact of floods. Adaptive capacity and household resilience were identified as two indicators that significantly determine how communities prepare, respond to, and recover from floods. So this graph represents, so this graph shows compares these two indicators across 9 South African provinces. So looking at the graph, provinces such as Northern Cape, Western Cape, and Gauteng have a high percentage of both indicators, which means that the households in these regions are well equipped both socially and economically to prepare, respond to, and bounce back from floods. In contrast, Limpopo ranges within the lowest, which means that there is still, which means that the communities in these regions still suffer from informal housing, inadequate infrastructure, and also lack of institutional support. So from the findings, flood risk is not just does not come from a single source. Instead, it comes from several factors that works together. On the environmental side, we see intensive and unpredictable rainfall, which increases the hazard. And also the issue of informal housing and inadequate infrastructure makes the communities even more exposed. Weak governance and lack of proper planning makes it difficult to effectively manage this risk. So to reduce flood risk, we need to address all these dimensions together. So in conclusion, flood risk is a multifaceted issue that needs collaboration interventions. So we need to strengthen local institutions and improve infrastructure, and also promote inclusive planning as a key factors of building resilience in these peri-urban settlements. Education and community involvement are just as important as they help the communities prepare to respond to and recover from floods. Thank you.
Thank you, Ms. Hopani. You took all your time and still a few seconds from me. Can we please ask the flat lady, how safe are we to be at Bolivia after 4 o'clock? Any questions for the flat lady? She can give us an idea, how safe are we to be here after 4? Please ask her. Okay, yes, I can see one.
Good afternoon. I just have this one simple question for you. You mentioned poverty as one of the key drivers to flood risk, right? So in South Africa, there's this other slide you were showing a graph, Gauteng and Western Cape. They were the most infected, they're the areas with the high occurrence of such flood risks. But we know that Gauteng and Western Cape in the whole of South Africa, they are one of the most developed provinces in our country. So there's less poverty in those two countries, those two provinces. So I want to know, how is it that they're doing well? There's less chance of, there's less percentage of poverty in those provinces, but they have a higher flood risk, whereas poverty is one of the key drivers to flood risk.
No, please don't take the mic from him. Can you please repeat the question?
Okay, poverty is one of the key drivers to flood risk. That's what you said, right?
Yes.
Yes. So I'm saying there's this other graph, or a bar graph. On that bar graph, Gauteng and Western Cape, they are the ones who are having the most high chances of flood risk. The percentage are high.
No, this graph is talking about the adaptive capacity and households' resilience as indicators that determines how the communities can prepare for, recover, and bounce back from flood risk.
Okay, it makes sense. Hence the percentage are high.
Yes.
Okay, my bad, I'm sorry.
I can see another hand. Oh, okay. Timekeep, there is a question. This should be the last one.
Thank you so much for your presentation. I was looking at your title, and you were saying that you are assessing flood risks in Gamotiba and Tagalang peri-urban areas, and I think you shifted, then you gave us a review. But then you sold me a dream then from that point of view, so I still want to know, from your drivers that you picked up from the review, what could explain that one video that you showed us where there was a lot of floods and you said it was in Gamotiba. So from your drivers that you picked up from the review, which ones could be associated with what we saw in the video? Thanks.
The number one key driver that is associated with that video is a poor drainage system, which is a key driver of institutional weaknesses. If you look at this video, it shows
the accumulation of water in this area. I think if there were proper working drainage system, the water would have like flowed to, I don't know, somewhere else instead of accumulating on one place. Yeah. Can I have this?
Thank you, Ms. Hapan. Let's give her a big round of applause. I know we have been clapping hands since morning. I can see the tired in your faces. This one?
Yeah.
It's not yours.
They said I should take
Okay, our next presenter is Mayepa Amokelani. She will be talking about carbon isotopes. So let's give her a round of applause while we are preparing her slides. Thank you. Okay, over to you.
Good evening, ladies and gentlemen. Standing in front of you is Ms. Mayepa, AF, currently doing my PhD. So the presentation that you're seeing on the screen is part of my PhD study. So it is titled Evaluating Carbon Isotope as the Proxy for Water Use Efficiency in Winter Legumes Cover Crop under Different Irrigation Region. It is under the supervision of Prof. Aisi, Dr. Mjonji, and Dr. Mamabulo. So world population is growing so faster, thus making the food production a major challenge due to water scarcity. So water scarcity is declining the crop productivity. This is because inadequate water supply is coinciding with the critical crop staging, resulting in reduced crop yield or crop failure. So that makes the farmer to choose a crop to cultivate so that they can prioritize this irrigation. So incorporating of these winter legumes cover crop will mitigate the water stress due to the fact that winter legume has a lot of benefits such as regulating soil temperature, conserving soil moisture, and suppressing weeds. So understanding the relationship between the isotope and water use efficiency will help to find a way to solve these water scarcity problems. So the objective is to determine the effect of water use efficiency, carbon-13, and their relationship in winter legume and irrigation regime interaction. So the hypothesis was derived from the objective above. The study site was conducted at Safarikale Experimental Farm and of Kolako, which is having the following coordinates as shown in the screen. And the average annual rainfall for Safarikale ranged from 650 to 700, while for of Kolako ranged from 300 to 500 millimeters. However, the minimum and the maximum temperature at Safarikale ranged from 15 to 30 degrees, while at of Kolako ranged from 18 to 35. However, the soil texture for both locations are the same. So this experiment was laid out in a split plot design with 4 replications where each plot size was 9 meters squared. So the main factor was 4 winter legumes, which is the forage pea, lupine, clover, and veg. The sub-factor was irrigation regime, where I have water stress and well-watered, where well-watered was irrigated up to full field capacity, while the water stress was irrigated half of field water capacity. So the rain gauge were installed in the middle of each plot where the trial was conducted in winter season to avoid the rainfall interference. And all management practices were done when necessary. The biomass was collected in a quadrant of 50 by 50 in the middle of each plot, where the roots were separated from the shoot and oven dry at 65 degrees Celsius, where water that was collected from the rain gauge was added from planting up until harvesting, and the formula below was used to calculate the water use efficiency. Leaf samples were then collected in the middle of each plot at the height of 20 centimeters above ground level, and the samples were oven dried and grinded into powder, where they were sent to a lab and then the temperature conservational elementary analyzer, and the formula below was used to calculate the carbon-13 isotope. So all data collected was subjected to SAS 9.4 Vision 2014, and the mean was separated using LSD. So the table that you're seeing on the screen show the effect of water use efficiency on winter legume and irrigation, where both locations show non-significance or the interaction of winter legume irrigation. However, the lowest water use efficiency was observed, and the highest shown on bed at with the fact that the leaf area was more to make transportation where the hazard was the fact that the reported by 2015 et al. 2010, which also this result by Shatter et al. 21. So table 2 shows the isotope, carbon isotope on winter legume and irrigation region, where Safaricale didn't show any significant, but Afcolako showed a significant difference on the carbon-13. So water stress show the highest on the fact that when water then to limit the carbon dioxide to the use of the plant the plant doesn't have a choice but to choose the carbon dioxide that is entering within the plant. So if in case the carbon that is entering into the 2019 on Bali, and Rail et al. 2014 on Bali. So the figure 1 shows the relationship between water use efficiency and biomass. However, there's a positive relationship between water use efficiency and biomass, as you can see. Figure A and figure C and D, they show that the points are close to the line of best fit, which show that they have a strong correlation or they have a strong relationship, as compared to graph B, which the points are scattered away from the line of best fit. That shows that there's a weak relationship between the two variables. However, the positive correlation was in line with the finding by Cordons et al. 2004 on soybean. The relationship between water use efficiency and carbon-13 showed the positive correlation between water use efficiency and carbon-13, as you can see on figure number A and number C and number B. So as the carbon-13 increasing, the water use efficiency is also increasing. However, under well-watered at Ofcolako, there's a negative correlation between water use efficiency and carbon-13. So the study, these study results were similar to the finding by Wang et al. 2022 on rice and Minin et al. 2022 on beetroot and Zhu et al. 2019 on alfalfa. So lupin and P are discriminate more carbon under water stress when compared to well-watered when comparing them with those other winter legumes. However, lupin and pea are valuable winter cover crop Winter cover crop that the smallholder farmer can adapt to their cropping system, especially in winter. This is because in winter, most of the farmers are not planting anything, but they're leaving their soil bare. So this is the chance for them to plant this kind of cover crop so that there will be a nutrient recycling. So water use efficiency can be used as the proxy for carbon isotope, hence null hypothesis will be rejected. So winter legumes are recommended to the farmer who are cultivating under limited irrigation or under water stress, water scarcity area. Further studies can be done on recommended winter legumes under different irrigation application method on different agroecological zone, where further studies can also be done on different level of irrigation, where the irrigation or the amount of water can be measured. The scientific contribution for this study is that it's going to generate information to enhance the knowledge of the winter cover crop that the researchers and the farmer are having, and to also identify water sufficiency. It also identifies water use efficiency legumes that the farmer can incorporate into their farming system to ensure sustainable agriculture and food security. It's also going to provide opportunity for further research to address the climate change challenges. I would like to acknowledge the Center for Global Change, my supervisor, University of Limpopo, and NRF. These are references which were used in the presentation. Thank you.
Thank you, Ms. Maepa. We have about 5 minutes to ask her. I can see one hand for now. Yes. There is a second one.
Okay. Thank you for a very good presentation. I'm worried about the stress treatments. Looks like the results for carbon isotope and water productivity are not different. So I ask you the question, how did you execute or how did you induce stress on your legumes? Because I detected that the results are actually the same. Did you measure any soil water content? How did it differ between the well-watered and the water stress treatments?
Okay, thank you for the question. So what was happening is that I was irrigating 4 times in a week for well-watered and 2 times in a week for water stress. So during those 2 other days which I was not irrigating for water stress, that's when I was exposing the winter legumes to, I was stressing them, actually not irrigating. So I measured the temperature and moisture. So under water stressed, because the canopy was not completely covering the soil, the temperature were too high and the soil seems to be dry because the moisture content was very low. So what I can say is that since cover crop they conserve moisture, maybe during that stage where we collected data for carbon isotope, this winter legume managed to conserve water to use at that critical stage. That's why maybe the results are not having that huge difference.
Oh, okay, thanks. I think Dr. Mashilo, you stole that question right out of my under my nose. You were presenting some, I think it was regression analysis that you were presenting. You don't mind to just go back there because I think you made conclusions based on those. You've gone back there. Okay, I don't see them here, but it's fine. I still remember what was happening. Okay. So the relationship between what you call your water use efficiency, but let's call it water productivity for now, and your carbon isotope, your carbon-13 isotope, they were very weak. You hardly had an R square above 0.1 or 0.2. And that signifies a very weak relationship. but then you go on to conclude that you can use carbon-13 isotope as a proxy, even though there are weak relationships. Do you mind just maybe helping me out there? I'm a bit confused.
Okay, no. This is because the correlation between carbon-13 and water use efficiency was that whenever we find that the carbon-13 is high, whenever that we find that the carbon-13 is high under water stress, which means that the plant can be able to photosynthesize and give us the end product that we want, even though that the water is not irrigated up to the field capacity.
Okay, that's fine. Just as a take-home comment, I think you need to go back and re-look at the role of carbon-13 in photosynthesis. You're throwing it off there a bit from time to time. Thanks.
Okay, thank you.
Okay, on that note of weaker correlations, I will no longer accept more questions for her. She has already left the stage. Okay, so it's okay. Thank you, Ms. Maeb. I think also the correlation between our mines and Bolivia Lodge is getting weaker. So I have been requested to ask those presenters for the next session to be, in the meantime, uploading their slides so that we don't waste any more time. Also, I'm requesting that any member of the organizing committee advise us in terms of time. I can see we're supposed to be having a 15-minute break there. So can they advise us whether we will still have that 15-minute? That is the first advice. The second one is clarity in checking whether there was no mistake there. It says tea break. Is it not a wine break? Thank you. The next presenter is Mary Goss. I will come back with all the responses. Okay, she will be presenting to us about the hydropetrological impact of urbanization on water resources. Over to you.
Hi. Sorry, guys, please bear with me. I have flu. Hi, everyone. Standing before you is Mary Kosi. Yes, I had to make sure that you still had the tea break because I prepared like an opening line. So he said you still have, so it's still valid. So it seems like the person standing before you and the tea, it's me. I spent so much time preparing that. I hope you guys laugh because I prepared. Anyway, I'll be giving a presentation on evaluating the impact of urbanization on water resources using a hydropetrological approach, and our case study is X22J. This is the presentation outline, cover the background or to the conclusion, and why urbanization basically? So I'm sure we all know of an area when we were younger which was used for livestock grazing or agricultural crop plantation, or it was just an open field, but all of a sudden there's a mall there. In some extent, there's a city, a whole city standing there. So that shows that urbanization is inevitable. So basically, urbanization has been noted as one of the drivers of hydrological changes because of its direct interaction with the surface and the soil, which in turn affects the way water is partitioned on the soil and on the surface and the subsurface. And because of this, we have applied the hydropetrological approach, which gives us a window into the hydrological processes taking place within this medium. So basically, it gives us an opportunity to convert soil survey data into hydraulic information that can be used to conceptualize those flow dynamics. By that, I mean that can be used for, that we can use to show how water is behaving underneath the surface and on the surface. And that brings me to my aim for the study, which was basically looking at the interaction between soil, water, and urban development with the focus on understanding how urbanization will affect the subsurface hydrology and the catchment hydrological response of the Quaternary catchment X22J. These are my objectives, but in a nutshell, I had to sort of analyze land use for 1990, 2007, and 2024, and also digitize a soil map, which was completely new to me, and also basically use all that information to give you a full picture of what the hydropetrology was in that study area. So those are my outputs, sort of the products, you see, we in the thing, entrepreneur. So this is the catchment. It's located in Mpumalanga, and it's one of the 35 quaternary catchments of the Crocodile River catchment, and it covers an area of 240 kilometers squared. That beautiful map there, I made it, yes. So for this study, I needed primary data and secondary data, and the secondary data comprised of climate, hydrology, DEM, land type, and satellite images, and then the primary data was basically me going to site and actually collecting the cell samples, yeah. For analyzing my land use, those table 2 there, it shows you how we divided the land use classes. So for analyzing the existing land use analysis, like for 2007, 1918, 2024, we used the random forest algorithm in the Google Earth engine. And then for predicting, we used the Melus plugin in QGIS, which sort of uses the CA, ANN, and MLP. And our environmental variables were elevation, distance to road, population density, and DVI. Figure 5 there, it shows you the architectural network that is used in the study for the ANN, right? And this is us on site collecting the cell samples, and that's Dr. Eddie Smith there, who's also a former student of Prof. Van Tol, who's one of my supervisors, and we also got some of the sampling points from him, with legacy data, and that's my supervisor and a team from Sayon as collecting the samples. And then for digitizing the soil map, we had the core variables, which are land type, geomorphones, curvature, total water index, all of this information, we got it using the Saga. It's another GIS platform, and from a DEM, so we got all of that from a DEM, and then we created a set of rules that we used in QGIS to sort of create a beautiful map that you'll see shortly. And then for the with the quantification of fluxes, we use the SWOT model. And for the model performance, we used NSC, RSRP bias, and the outputs that were the focus of this study were lateral flow, which is interflow, percolation, represented recharge, surface runoff, which represented responsive, and then evapotranspiration. And for sensitivity analysis, model calibration, and validation, we use an RSWOT model, which is an open source RStudio-based software that is used for SWOT calibration, validation, and all of that thing. And then for table 4, those are the focus parameters for this study. In terms of results, and what we saw is that for our overall accuracy for our classified images was above 0.5, which I was happy about. I was happy, so if I'm happy, everyone has to be happy about it. And then the kappa coefficient, it was also about 0.5, which is acceptable. And then for the predicted land use was 0.52, the kappa, and then the overall accuracy was 0.75. So what we saw was that we're going to see an increase of 5% in urban areas between the period of 2024 to 2040. And we also saw that between 2024 to 2040, we're going to see that 20% of the natural vegetation will be transitioned into urban to built-up areas, as well as 10% of the cultivation areas. So I'm only focusing on the results that are more important to save time. So for our soil forms, we actually identify 7 soil forms, but then for this presentation, I'm only presenting 5. I would like to draw your attention to the champagne one, which made a lot of soil scientists happy. I was very clueless about it because I'm not a soil scientist, but everyone was like, oh, yay, peaty soil. Okay, and then we found that most of the soil samples that we collected, they fell within the sandy clay loam and sandy loam, which was good. It was also evident by the fact that we had a lot of agricultural activities in the area, so yay. And then, so we found that in terms of distribution of texture, most of the soil samples had high sand content, okay? And those are the beautiful maps I was telling you about. So, figure 14A, that's the soil form map that we sort of digitized using QGIS. And then those are the mapped soil forms. And then that was transitioned into the hydropedological types, which were responsive shallow, matching with MISPA, interflow, soil bedrock, responsive Houghtonnian, interflow shallow, Richards deep, and Richards shallow, and responsive wet. So the difference between the three Richards is that the other one is influenced by built-up areas. The other one is basically the type where if water falls in, it runs off immediately. This could be an open rock or anything, and then the responsive wet, it has to be saturated first before water runs off. I hope that makes sense. I hope it does. And then this is an example of what we did. We have a bunch of hill slopes, but for this presentation, I'm only presenting one of us to show you what the impact of urbanization would be in a hill slope. So let's pretend that you have a hill slope which is interflow dominated, meaning that the water moves laterally. And then all of a sudden, the surface is sealed. Now, the water will start to move in a Houghtonnian, which is that the responsive Houghtonnian, and then we get less water actually moving laterally. As you can see, that disrupts the whole hydrological region. But of course, interflow is a complicated, especially in urban areas, it's a complicated process because you have to take into account pipe leakages and all of those things. And this is how we calibrated our soil maps for the for the hydrological model, and this was a hydrological model, and our results were 0.58, 0.68, and this we got them from SWAT. And we also did the urbanization to sort of show you how which one is dominated by the land type. So this is how we did. We found that sub-basin 8 was the most dominated, and then from that we found that evapotranspiration decreases in highly urbanized areas, and we found that due to sealing of the surface and wide surface runoff increases as well, and then recharge decreases as well as lateral flow. And that's the conclusion. Thank you.
Thank you, Ms. Kosi. We needed that energy. And she deserves at least to be asked 3 questions. I don't see any hand yet. Oh, okay. The timekeeper.
Thank you, Ms. Kosi. I think I enjoyed your presentation. Very few words, very visual. I think that's how I like my presentations. But again, I'm not into hydroecological modeling. You are modeling water flows, and then you are modeling them against urbanization, if I'm to be correct. And the thing that your indicator is evapotranspiration. Fair and fine, I understand that. My main concern whenever there's urbanization that happens is that there's now urban water use. Each household has a quota now that they are given for water that they use, and I will assume that increases the amount of water that is used by a system, by the urban system. Wouldn't you agree? If you build an RDP, you must put a tap in there and people use a certain amount of water. Okay, that's basically what I'm saying. But then the question then becomes, did you consider the urban water usage that comes with urbanization as part of your modeling?
Okay, so that will fall under the scenario analysis, but remember my study was basically just looking at the hydrology and the hydropetrology. I was not looking at the water requirements that is needed for urban people as they grow. So I was basically just looking at the hydrology and how this system as a whole will be affected, not really what people, how the abstractions will look like. Yeah.
I don't see any other hand. On that note, let's give her a very big round of applause. Thank you, ma'am. Ladies and gentlemen, I think our session has come to the end, and I had promised that I will give you the response with regard to the break, and it goes as follows. You are requested not to take that 15-minute break because we are pressed time-wise. So on our way, when we are leaving after this next session, then you will grab something home. On that note, I would like to thank you for your time and patience, and I will hand over to Dr. Motsecha, Dr. Hopa, and Dr. Mbokane. I can only see two of them. Thank you very much.
Good afternoon or good evening. And let me take this opportunity to not really welcome you, but to awaken you by indicating that we are requesting our presenters to load their presentations. There was a request made, but I haven't seen any movement towards the brothers who are assisting us. In the meantime, let me take the opportunity to check the availability of our presenters before I introduce myself. I'll introduce myself while they are loading their presentation. We have Itumele Mkabela, all right, Makudu Matiba. Can you proceed to the far end there so that you load your presentations? Lidimo Makopa, all right. Lamuli Risimate. Lamuli Risimate. I don't see any response. Sophia Koga. All right. Mususumedi Malema. Mususumedi. All right. Have you loaded? Please proceed to load your presentation. And Yonwaba Matungu, Masilo Tebiani is absent. We got a request. Surprise Mutala. All right. Jabulani Ntuli. Jabulani Ntuli. Absent. Cedric Silwani. Jabulani. Oh, Cedric Silwani, and the last one, Tibulwa Nebutalo. All right. Okay. I believe they are proceeding to load their presentations. In the meantime, I can then take this time to introduce myself. Okay. All right. In the interest of time, I am getting an indication that if the presenter is not around, we will move. We will skip the presenter. All right, I'm Lesego Muteka, going to co-present this session with Dr. P.M. Khopa and Dr. E.M. Mbogane. Dr. Khopa will assist us with the time management.
She will wave to indicate that you are running out of time, and I will stand and come closer to you, and I will chair the 6 presenters. Then Dr. Mbogane will continue with the other 6. With that said, can we see our first presenter, Itumele Mkabela? And we are going to use the same protocol, 10 minutes presentation and 5 minutes question time. Can we give her a round of applause? All right, Itumele Mkabela will be presenting to us potato cultivar specific yield recovery to variable hail intensity levels in subtropical temperate climates. A round of applause to her to cheer her up.
Good afternoon. My name is Itumele Mkabela. I'm a master student. This is part of my current study. So today I'll be taking you through potato cultivar specific yield recovery to variable hail intensity levels in subtropical climates, along with my co-authors. Potato is the 4th most consumed crop globally, and it contributes to food security and also agricultural economies. However, since potato is planted in the field, it is particularly vulnerable to climatic factors such as the occurrence of hailstorms. And this is a problem because the occurrence of hailstorms usually coincides with the planting of potatoes. When hailstorms occurs during the growing season, it actually causes stem injury, canopy loss, and this results in yield losses, whereby a complete disruption to the potato canopy can result in about 70% yield loss. And these yield losses means that these 70% yield losses means that there will be less potatoes reaching the market, and that means that but the market might increase the prices of potatoes, meaning that some consumers will not be able to afford, which then can result in food insecurity. With climate change, the occurrence of hailstorms are expected to be more frequent and also more intense. In the picture here where there is a car and also a picture where there is a potato canopy, actually in 2022, during our potato trials, hail actually occurred and disrupted our field, and this actually resulted in severe yield losses. And to support this, there is a recent case study where a commercial potato farmer mentioned that last year they had 3 hailstorms in just 1 season. And this just proves to us how this occurrence of hailstorms actually are causing a problem to potato production. The current mitigation strategy that farmers are actually using to mitigate the hailstorms in potato production are usually expensive, and some small-scale farmers are not able to access them because they are expensive. So there is So one strategy, one affordable potential strategy that they can use is selecting cultivar that they can be able to recover and compensate for yield losses. Therefore, the study aims to develop a cultivar-driven strategy to minimize yield losses and maximize recovery. The study was conducted in 2023 to 2024 and 2024 to 2025 growing seasons at the University of Limpopo Experimental Farm located within the Capricorn district. The site experiences temperatures of about 5 to 28 degrees Celsius in winter, and the summer temperatures can escalate to about 36 degrees Celsius in summer. And these high temperatures in summer can contribute to the likelihood of hailstorms. So during our two study seasons, hail actually occurred 9 kilometers away from the farm. However, it did not reach our experimental field. So this actually proves to us how this region is actually becoming more vulnerable to the occurrence of hailstorms. The study was conducted in a 4x5 factorial arrangement in a randomized complete block design with replicates. The 4 potato cultivars were sourced from West Grow, which is a potato breeding company, and they differed in their time to maturity, whereby we had the early to late maturing types. So these cultivars also differed with the popularity within the South African potato growers and also its consumers, whereby we had the most popular cultivar, which is Mondal. I'm sure all of you know this one. And then we have 3 potato cultivars, which are recently developed. So it was important for us to check the recovery prospects of the popular cultivar in relative to the recently developed ones. Hail damage was induced using a hail hammer following the procedures outlined by Bucket, and we first calibrated by 400% defoliation, and then we used the proportion that we actually used the number of strikes for the 100% defoliation to calculate the lower levels. Data was collected on yield recovery, canopy cover, and time to maturity. And data was analyzed using GenStat version 21. And the mean separation were done using Ben-Foron test. And we also conducted regression analysis using Microsoft Excel. Here we have the regression analysis, which we use to model the relationship between the inflicted canopy disruption levels and also the parameters, whereby the regression analysis showed an inverse relationship, whereby increasing the hail disruption level actually resulted in decrease in all these parameters. However, when we looked at the cultivars, the late maturing cultivar Panamera actually had the gentlest decline in all these parameters. And this actually indicated that it had a showed a slower yield and also canopy loss, which also indicated faster recovery. Panamera actually produced superior high yields of about 39 tons per hectare, and also a high yield recovery of 64% at complete canopy disruption. And this means that with these high yields and high yield recovery, means that farmers can still, in hail-prone regions, they can still be able to produce more yields, which will then mean that the market will then be able to get more potatoes, which then will help secure food security. The result, the high yields and yield recovery in Panamera was actually attributed to its higher maximum canopy cover, whereby it actually achieved high maximum canopy cover and maximum canopy recovery compared to the other tested cultivars. So this high maximum canopy cover and canopy recovery in Panamera actually maximized light interception, which meant it maximized the photosynthesis, and this means that it produced more assembly that were then translated into the tubers, then resulted in high yield and high yield recovery. Another factor that was linked to these high yields and yield recovery in Panamera was actually time to maturity, whereby Panamera actually took longer time to mature, and this was because Panamera is actually bred to be a late maturing cultivar, and these longer maturation period actually allowed Panamera to recover its, to regenerate its canopy to its full potential. So Because it recovered its canopy to its full potential, it meant that it was able to maximize photosynthesis, light interception, meaning that it maximized photosynthesis, which then resulted in superior high yields and yield recovery. The result of the study indicated that cultivar selection can be indeed a solution in mitigating hail damage in potato production. So farmers in hail-prone regions, they can plant Panamera for better yield recovery and yield. So for breeders such as West Grow, since we're working with West Grow and it's a breeding company, they can focus on traits such as larger, longer-lasting canopies and also longer maturity. So by selecting and planting these cultivars will mean that there will be more yields produced under hail conditions, and this means that there will be more potatoes reaching the market, meaning that this will actually secure food security. So for future research, we recommend that the test of hail disruption at different growth stages, so that they can be able to check how these cultivars perform if hail occurs at different growth stages. I would like to thank the University of Limpopo, the Northwest University, and also the CRISP project under which this project falls under. Thank you.
Thank you very much, Ms. Kabela, for a very nice presentation. Can we get the questions from the floor? We had a few minutes to entertain the questions. Any questions from the floor? At least one. Okay, there's a hand.
Thank you for your presentation. My question is, do you think soil physiochemical properties had an impact?
Oh, okay. Since we conducted this study, like we didn't check the soil physiological properties. So we assumed that since we conducted the study at the same place, so we assumed that the soil properties were almost the same. So I don't think they had an effect.
Okay. Do we still have any other question? She utilized her time very well, so you can take a minute to ask another question, then we can release her. Okay, it seems like there isn't any question. Then the question from myself is, sorry about it. You mentioned that Mondial is the commonly used cultivar, and it seems like Panamera is the new cultivar that is doing well. So I'm interested maybe for one, what is this? For one recovery variable or one trade related to hail recovery, which percentage difference have you observed between Mondial and Panamera? Just one trade.
I think it was maybe the longer the canopy cover. So Panamera actually had like taller plants, which then and also okay. I think it's the canopy cover whereby Panamera had taller plants, whereby it was able to cover the surface area, so which in Mondial was not the case.
Okay, I'm satisfied with the answer. Can we give her a round of applause as she is ascending the stage? She is descending the stage. And can we get the next presenter, Makudu Matiba? Makudu Matiba. 1, 2, 3 minutes going. Makudu Matiba. Oh. Thank you. Let's give him the round of applause as he's ascending. Makudu will be presenting to us characterization of seeds. Can you assist us with loading in the meantime? Makudu will be presenting to us characterization of seed quality of indigenous and localized South African cannabis genotypes. They are loading the slides. A round of applause to welcome him.
Good afternoon, everyone. My name is Ephraim Matiwa. I'm currently doing my second year in Masters, and I'm happy and pleased to present to you a subset of my study titled The Characterization of Seed Quality of Indigenous and Localized Cannabis Genotypes of South Africa. So for all those of you who may never or have never seen a cannabis plant, this is basically how it looks. And please don't be confused with all these terms. Cannabis just basically means tipaje, you know, the very same ones jafelo, you see. So yeah, cannabis has been a part of South Africa's culture for many years, especially in rural areas. Therefore, it has a lot of economic value to these communities. Even when it was banned in the late 19th century, many farmers were able to keep on producing and selling this crop. Well, they did that illegally, by the way. So based on that, they were able to also store and keep their own germplasm or seeds and further passing them down from generation to generation. Recently, I think also you may have been aware that there is a rise in interest in cannabis. So this can also benefit South Africa, and it therefore becomes imperative for us to study this germplasm in order to provide a scientific based information that will further make sure that this valuable crop can be used in a more sustainable way, and that can benefit both the stakeholders. And by stakeholders, I also mean the farmers and all the communities where where this germplasm is concentrated. So even though we have a chance to, you know, venture into this cannabis thing, the South African cannabis industry faces multiple challenges in order to fully establish, one of which is the lack of registered, localized, and indigenous cannabis strains. Therefore, this means that a majority of registered cannabis farmers rely solely on, well, not solely, but mainly on imported strains. So, by indigenous or localized strains, I basically mean strains that, seeds or strains or cultivars that have adopted to the local or our environmental conditions. So these imported strains sometimes have a problem in terms of adapting to our environmental conditions. So it therefore becomes imperative for us to study these strains of ours. So I'm sure some of you may be wondering, well, how is this study relevant to the theme of this conference? Well, if you think of it, it's simple in a way. Seed quality is the foundation of crop production, meaning without good seeds, then farmers risk reducing their harvest potential, which directly affects their profit, which is a very important thing. Also, identifying seed batches that meet the quality standards allow for easy registration of these seeds and easy patenting, basically protecting this germplasm or intellectual property in order to prevent theft. This means that farmers will be able to sell their seeds in the mainstream cannabis market, further creating income and economic growth for our country. Therefore, the aim and objective of this study is to assess seed quality by looking at and determining their viability and vigor. A total of 19 strains were collected from 3 provinces, majority being Limpopo. And this is the table of all the strains that were collected. Tests, basically seed quality tests were conducted in the plant production lab using a complete randomized design with one factor replicated 3 times. The test was split into 2, namely the seed vigor test and viability test. Looking at seed viability test, this is basically a measure of whether the seeds are alive and are capable of germinating. The key test for this, for viability, was germination percentage. However, other tests were conducted, mainly the electrical conductivity test, which we used the EC meter from the PASCO science, and respiration dynamics, mainly focusing on carbon dioxide expulsion by the seeds, and imbibition test. So this is the germination test I was talking about. With seed vigor, indices such as germination velocity index, mean germination time, and seedling vigor index were measured, and along with seedling growth parameters, mainly focusing on shoot length and biomass. Data analysis, or all the data that was collected in this study, was subsequently analyzed using GenStat. This is the overall table of results that were conducted, that were found in the study. So with seed viability, only 5 strains managed to meet the quality standard as outlined by the International Seed Testing Association, that are set at 80% or above. This high viability is mainly, or corresponds highly with the seed's metabolic processes that are crucial for germination. For instance, if you look at the table, the imbibition test results for strains that are best performers were mainly moderate or higher, which means that the seeds were successfully able to initiate the seed's metabolic processes, which include the hydrolysis and the breakdown of stored food produce. Food produce or energy in the seeds in order to produce energy that is then used for radical protrusion. And that was the case for also viability. This is the graph for showing viability performance per strain. With seed vigor, the trend was that the seeds that had high vigor also, seeds that had high viability also yielded into high vigor. So generally, there are strains that 5 strains met the Easter standards, and then all the other strains didn't meet. But we have strains that were a few points shy from the Easter standards, which, in my view, they require just minor improvements in terms of viability and subsequently vigor. In conclusion, this study proves that we do have our own germplasm that meets the standard and can be further used as foundation seeds for developing a local cannabis strains that can be absorbed into the mainstream cannabis industry, therefore reducing the dependence on imported strains. And for further research, I think we can take all the strains that didn't meet the Easter standards into your micro propagation protocol in order to make sure that they do meet the standards. Thank you.
Thank you very much, Mr. Matiba. He has stolen a minute of my time. Can we entertain questions from the floor? Any questions for Mr. Matiba? Yes, there's a question over here.
Thank you for a very excellent presentation. Why did you not use the standard germination test using the paper towel method? It's one of the recommended methods, as you've said, by Easter.
Well, even the Petri dish method is one of the recommended method. It's a matter of semantics, which one is best. But in this case, the Petri dish method allows for easy monitoring of the seeds in terms of, because I had to go every day looking at whether the seeds germinated or not. So with the paper towel method, I have to disrupt the experiment in order to count the number of seeds. So with this one, I can easily visually look at the Petri dish and count the number of seeds that germinated daily. Yes.
Okay, thank you, Mr. Matiba. Do we have any question? In the interest of time, let me release him and welcome Ms. Makhopa. I saw Ms. Makhopa was ready. She was closer to the stage. I'm surprised why she went back. Let's give her a round of applause and request the next presenter to ensure that the presentation is loaded and she or he remains there closer to the guys who are assisting us. Please. Ms. Makhopa will be presenting to us the bibliometric review of research trends on hematological parameters. Over to you.
Good evening, everyone. My name is Ledimo Faith Makhopa, a PhD student at the University of Limpopo under the Department of Agricultural Economics and Animal Production. And my presentation today is on the bibliometric review of research trends on hematological parameters of sheep between the years 2000 and 2024. So hematological parameters are important in assessing the nutritional, metabolic, and health conditions of an animal. And then when they are combined with clinical findings, they can help in diagnosing diseases and also prognosis. So a bibliometrics is a method of indirect research on developing a certain field that has emerged in recent years. So with a bibliometric analysis, we're trying to expose research trends, prolific authors on a certain topic, and the most prolific countries in order to expose research gaps. And based on our knowledge, there is no research on hematological parameters of sheep bibliometric analysis results. Hence, our aim is to conduct such a bibliometric analysis. And what we did, we used the Web of Science and the Scopus databases in order to retrieve data, and these databases are the mostly used databases in this type of study. And we did that by using hematological parameters, sheep, and blood parameters as our keywords, and we retrieved data on the 1st of September 2025. And then on Web of Science, we exported full records and cited references, and on Scopus is citation information and bibliometric information using a bib text, and all this was combined using a bibliometric R package routine. And with the results that we found a total of 2,582 papers with a total of 12,529 authors, and average citation per document were 11.59%. So as we look at our results on table 1 there, the bar graph represents the number of papers that were published per year. As we see in the beginning of the decades, the the topics on hematological parameters were fluctuating, but then there was a certain peak in 2020, and then in 2021 it was the highest ranking year with a total of 236 papers, and also 2021 was the year with the most average citations per year, followed by 2009. And the most prolific authors in this study were Khan and Kim, and then there was a total of 23 papers, followed by 22 and 20 papers in that order. And then the most prolific journals, we have Animal and +1 journals that were focusing on these hematological parameters of sheep. And then we have Aquaculture Research, which when we were retrieving information was focused more on fish, as you will see as we move forward. And then the most prolific countries here were the Asian countries and the European countries. And then we also saw a low performance in the African countries when it comes to this topic of interest. So the recommendation here will be that African countries collaborate with other countries in order to produce more papers that are focused on these hematological parameters of sheep, as they are important in the health of animals and also precision farming and profiling of animals, and also testing on whether a feed is working or not. So we are looking at a 3-field plot there. If we refer previously to the other slides where we saw that China and Turkey were the most prolific countries, we can see by the yellow, by the gray lines that are moving to the right, this is the author's affiliation. And then we see that Turkey has the highest number of affiliation, which also contributed to the number of papers that this country managed to publish, followed by China, which also had a lot of affiliation that also contributed to the number of articles that they were able to produce in this topic of interest. And then the most trending keywords we see there, we have hematological parameters, we have sheep, we have blood, we have expression, anemia, in that order. So it means that the most focus was on these keywords. And then there we have the core occurrence network. So the circles represent the keywords that were used, and the lines in between shows how these words were used together, and the thickness of the line shows how frequently these words were used together. So for example, on the blue cluster there, we see hematology, we see growth, we see performance, we see fish, and then we see rainbow trout. And then we can say that with this certain cluster, they were mostly focused on the performance of this fish with certain feed products to see the growth and all that. And then when we look at the red cluster there, it's mostly on neutrophils and inflammation. And then what we know about neutrophils is that they are responsible for the immunity of an animal. So they are the first line of defense. They produce an inflammation, triggering the system to say, go and fight a certain infection with the system. So we can say that with this cluster, they were most focused on the health of the animal in relation to the white blood cells, which are responsible for the immunity. And then on the green there, we see that they were focused on hematological parameters and sheep. And then there are these lines that are also showing the frequency, which is not a lot in between the clusters, but then it shows that these words were used together. So with this, we can recommend that since the red cluster is a bit small, and then it's in terms of the health of the animal, so more focus should be given to the health of the animal. And also the blue is more focused on fish. So we can say that more information, more studies needs to be done in this area in terms of sheep, so that we can have more nutritional studies in sheep. And then here we see a co-collaboration network. So we have the brown and then the orange. So these ones show that these authors work together. The brown ones work together. And then we see Chu there leading this area. It means this as his area of specialization. And then he's working with people in his institution. And also the the lines that are going into the other orange cluster means that they are also working with the other authors within other collaborations. And then we see on the far left there that there's a red cluster with 3 authors that are working amongst themselves, but then there is a lack of bridge between these authors and the authors that are more into this topic. So with this one, we can recommend that authors collaborate with the other authors. And then as we look at the say names there, they are more of a Chinese or Asian descent. So it means that China is working more on this area, so they can try and work with this Chinese authors so that they can get more information. And then there is lack of African surnames, meaning that there is lack of studies and collaboration with African countries with these other highly productive authors. And then when we look at the final figure there, we have countries such as China, we have Germany, we have Turkey, we have America, and then this shows that these are working together and they are collaborating more, and it is indicated by the thickness of the lines between them. But then if you go to Africa, you see that the lines are a bit blurry, meaning that Africa is sort of failing to collaborate with the other authors, which also could be the result of the lack of publication in these areas, because when we were doing this analysis, we found that South Africa ranked at 32 in terms of this. So this might be due to lack of resources and also lack of affiliations with institutions that might provide them with funding to focus on this area. So there was a lack of citation references and science categories, and this might be due to how the data was retrieved from the databases, which is the limitation of this study. And also another limitation is that Africa is not doing enough in terms of this current topic. And then we conclude that out of 2,582 papers, the trending words were sheep and hematological parameters. And then we recommend that Africa does more work, finds more funding, meaning governments need to fund this research institution in South Africa so that we can improve the productivity and the livestock production of our African countries. Thank you, these stakeholders, and thank you, everyone.
Thank you very much, Ms. Makopa. Questions from the floor. She exhausted all the 10 minutes, so limited time for questions. But at least one, at least one, at least one. Thank you. There's a question over there.
Thank you very much. I just need to ask a question. When you displayed your graph of publications, it highlighted that from 2020 there was a peak in the number of studies that were done under that you've used, right? So according to you, and this is your field, what do you think might have been the cause to such mass production of papers that were produced from 2020? Because that was the highest as compared to the previous years. And two other things, COVID-19, which arose around that time, it posed of limitation to such studies being done because there was minimum movement. I would just like to know from you, what do you think might have been the cause of such an increase in number of papers produced? Thank you.
Based on my knowledge, I would say that the sudden increase could be, since the keywords that were there, some of them were not only in animals, but rather the effect of this COVID-19 into this livestock production. So COVID-19, yes, it did have an effect where people wanted to learn more about these hematological parameters. And then again, when we say that they were testing nutritional purposes of feed, so it can be that they wanted to fight this global lack of feed. So they were trying to test feed using these hematological parameters along those years in order to improve livestock production and also improve disease resistance so that at least there can be an increase in animal production at that time.
Thank you very much. If you are not happy, please take it outside the presentation hall. You'll have more time. Okay, thank you so much, Ms. Makhopa. A round of applause, thank you. As we are also welcoming Risimate. Risimate? All right. We have requested the presenters to come forth to save time. Please, please, guys. Okay, Risimate will be presenting, evaluating the physical-chemical and biological properties of Lephalale River. Round of applause.
Good day. My name is Risbaden Tlamulo, and I'm here to present my FSA presentation, which is titled Evaluating the Physicochemical and Biological Properties of Lephalale River Water through the Utilization of Pure Montmorillonite Clay and Copper Oxide Montmorillonite Clay Nanocomposite under the supervision of Dr. Matabula, Dr. Slep, and Mr. Sbayo. This is my presentation outline from the background of the study up to the references. So coming to the background of the study, well, water contamination is a global problem, and Lephalale River is not an exception. Lephalale River, which is located in the Limpopo province, South Africa, in the Lephalale region, is a vital water resource to the community of Lephalale. However, it faces significant contamination from various anthropogenic sectors, such as the industrial sectors, domestic sectors, and the agricultural sectors. However, the recent literature clearly revealed the elevated levels of turbidity, color, E. coli, and total coliforms in the Lephalale River water, which poses a significant threat to the health of the local community, which is Lephalale, who rely on the river water. And this calls for an urgent need for the effective, low-cost, and sustainable water treatment method to ensure safe and accessible water for this vulnerable community. Coming to why I chose Montmorillonite clay and copper oxide in nanoparticles in this study. The reason behind that is because Montmorillonite clay, it is of low cost, and it is a natural adsorbent, and it possesses high surface area and exceptional ion exchange capacities, which makes it easy for the MMT clay to remove impurities in the water. And the reason why I also chose the nanoparticles copper oxide is because it possesses the antimicrobial properties. So it makes it easy for the nanoparticles to remove the microbial contaminants in the Lephalale River water. Coming to the aims and objective, the aim of this study is to explore the physicochemical and biological properties of Lephalale River water following treatment with pure MMT clay and copper oxide MMT clay nanocomposite. And this study consists of 3 objectives. The first one is to evaluate the physicochemical and biological properties of Lephalale River water prior and post treatment with pure MMT clay. The second one is to fabricate the copper oxide nanoparticles and the copper oxide MMT clay nanocomposite and characterize them with SEM and FTIR. And the last one is to evaluate the changes in physicochemical and biological attributes of Lephalale River water after treatment with the copper oxide MMT clay nanocomposite. Coming to the sample collection and sample analysis, well, the water samples were collected in the Lephalale River, in Lephalale, and the sampling bottles were autoclaved prior sample collection to prevent microbial contamination, since in this study there will be a microbial analysis of the Lephalale River water. And the samples were collected in 5 sampling points, and upstream, midstream, and low stream in triplicates, and the sampling distance was 500 meters. Coming to the sampling analysis, well, there are parameters, volatile parameters, which were analyzed in situ, such as pH, temperature, dissolved oxygen, conductivity, and turbidity. And the rest of the parameters were analyzed at CDM. Coming to the results and discussion. The table here, it shows the water analysis prior treatment of the Lepalala River water, where these results corroborate with the claims that are made in literature, whereby we have elevated turbidity, color, and TC, which is total coliforms, and E. coli, from sample 1 up to sample 5. Coming to the statistical analysis technique, well, in this study, I've used the Pearson correlation plot to determine the relationship between the parameters, the baseline parameters of the Lepalala River water. And in this plot, it shows that there is a direct relationship between color and turbidity, since it is accompanied by the strong positive correlation. And there is a strong negative correlation between pH, EC, dissolved oxygen, total dissolved solids, BOD, and COD, and vice versa. Coming to the optimization of the MMT clay adsorbent. Well, the clay, it was optimized in order to get the optimum performance. So the factors which were investigated during the optimization process, it was pH, dosage, and the contact time. So under the optimization, we found that the optimum pH is 6, as you can see on the figure. On the figure, where there is a percentage removal versus the pH, whereby it is accompanied by 94.37% removal, and the adsorbent dosage is 2 grams, and the contact time for optimum performance is 180 minutes. Coming to the Lepalala River water was treated using the patch adsorption method, whereby the clay was added into the water and there was stirring at 200 RPM, and there was filtration, and the filtered sample was taken to CDM for physical, chemical, and microbial analysis. The results after treatment with the MMT clay, they've shown that the clay was very effective in terms of removing some of the dissolved solids in the water. Since turbidity was used as an indicator, from before treatment, the turbidity was high, and after treatment, the turbidity moved from 11 to 2.2, which is more than 80% removal. However, coming to the microbial analysis shows that the clay failed or was unable to remove the microbial contaminants in the water, and that substantiates the reason why the clay should be coupled with the copper oxide nanoparticles, because they possess the antimicrobial properties which are effective when it comes to the removal of the microbial contaminants. Coming to the synthesis and characterization of the copper oxide nanoparticles. Well, the copper oxide nanoparticles were synthesized using the aqueous precipitation method, and the method is depicted on the screen, where there is copper acetate, which is used as a precursor, and the sodium hydroxide is used as a reducing agent. And the formed nanocomposite is later taken for characterization for FTIR and SAM. Coming to the characterization of the copper oxide nanoparticles, the graph, which is the FTIR graph, it shows that there are existing, FTIR, which is responsible for showing the functional groups on the nanoparticles, showing the dominant functional group, such as the hydroxyl and the carbonyl. And on the other side, the SAM, which is responsible for showing the morphology of the structure. It has shown that the structure of the clay is spherical. Coming to the synthesis of the copper oxide MMT clay nanocomposite, which is basically the amalgamation of the clay and the nanocomposite to form the amalgamation of the clay and the nanoparticles to form a nanocomposite. So the steps which were followed or which were used when we were fabricating the copper oxide monomer clay nanocomposite, it was the sonication, which we used to make the clay, to increase the surface area of the clay prior adding the nanoparticles. And there was stirring between the clay, the stirring of the clay and the copper oxide nanoparticles under a reflex of 5 hours. And there was a centrifuge, the clay was centrifuged for 10 minutes and washed, and then the nanocomposite was dried in the oven overnight. These are the results for the Lepalala River water after treatment with the nanocomposite. And it have shown that from prior and post treatment with the copper oxide MMT clay, the level of the total coliforms and the E. coli in the water decreased to -1, which adheres to the permissible standards of sense. So coming to the future work, on the future work, we are planning to increase the loading of the copper oxide nanoparticles, since we varied during the experiment, but the results that we got were the same, and also further research is required on the potential health and environmental risk. And the development, we are planning to develop the low-cost and effective filter that will remove impurities from river water in rural areas. And coming to the conclusion, the physicochemical and biological attributes of Lepalala River water were evaluated using the MMT clay and copper oxide nanocomposite. The treated water was found to adhere to the Sense 241. The purified water will not, therefore the purified water will not deteriorate the public health of the people in Lepalala River, in the Lepalala region, and it further addresses the SDG 6, which is all about improving the water quality and the universal access to safe and affordable drinking water. My acknowledgement goes to my supervisors, University of Limpopo, CDM, and EWCITA. Thank you.
Thank you very much, Mr. Risimante. He consumed all the 10 minutes. We have a minute for questions. Any questions from the floor? At least one. There is a hand there.
Thank you, Risimati. Please allow me to off-ramp a bit. So I want to know if there are any mitigating strategies that are put in place, to measure the contamination of Lepalala River water.
Come again.
Are there any measures in place that are being employed or implemented to reduce the contamination of Lepalala River, maybe by the municipality or government officials?
Well, on that one, they are using them. It's whereby they go and educate the people, because one of the anthropogenic activities which is proximal to the river, it's illegal waste discharge. So they are going, the municipality is going around educating the people about the harmfulness of throwing a debris next to the river. But it seems like the people continue to throw even though they are being educated. So I'm not sure if I answered you.
It's enough for now. Thank you very much. A round of applause to him. If you are still more interested, you can engage him after the presentations. The next presenter, Sophia Koka. Sophia will be presenting on optimization of biostimulant Kelpac using Topsis Shannon entropy for reducing soluble sugars in cold stored potatoes. A round of applause to her.
Greetings, everyone. My name is Sophia Koka, and I'm a second-year master's student, and I'll be taking you through a presentation titled Optimization of Biostimulant Kelpac using Topsis Shannon entropy for reducing soluble sugars in cold stored potatoes, a risk management strategy for acrylamide forming factor, under the supervision of Dr. T.K. Sateche, Prof. T.P. Mafio, and Prof. Mataba N. Potatoes during storage, they are susceptible to physiological defects such as sprouting, decay, and mass loss. To mitigate these physiological defects, cold storage it is mainly utilized. However, the prolonged exposure to cold storage leads to accumulation of reducing sugars. This is an unwanted phenomenon. The reducing sugars, they react with asparagine, which is an amino acid, during a Maillard reaction when the potatoes are cooked at or fried at temperatures that are higher than 120 degrees Celsius, and they lead to a chemical compound that is known as acrylamide. Acrylamide was classified as a potential carcinogen, and it is harmful and can cause cancer. So furthermore, the potatoes that have high reducing sugars, they also turn brown when they are cooked. The accumulation of reducing sugars negatively impacts both the potato and the consumer acceptance. Therefore, it is essential to develop post-harvest technologies that can minimize sweetening and consequently acrylamide formation. The biostimulant is an eco-friendly and cost-effective technology which is currently sustainable in agriculture to mitigate cold stress. Potatoes being a cold-sensitive crop, they accumulate sugar as a response toward cold stress. The biostimulant kelpec was found to be rich in phytohormones such as abscisic acid, gibberellins, brassinosteroids, and these are hormones that are known to mitigate cold stress or any other abiotic stress. Thus, we hypothesize that the biostimulant kelpec can protect the tubers from cold stress due to the phytohormones that it contains. TOPSIS, it is a widely recognized multi-criteria decision-making that is used in various disciplines. It identifies an ideal solution. In our case, the ideal solution will be the one that has low reducing sugars, and also it also identifies the negative ideal solution, which in our case it is the one that has high reducing sugars. Shannon entropy was also integrated in this study so that we can minimize subjectivity in the weighing process. Unlike just utilizing TOPSIS alone, the entropy assigns weights based on the variability and the information contribution of each parameter. Therefore, the aim of the study was to reduce acrylamide forming factor, soluble sugars in cold stored potatoes using the biostimulant kelpec. And the objective was to determine the optimum kelpec concentration for reducing soluble sugars accumulation in TOPSIS Shannon using TOPSIS Shannon entropy. For the materials and method, we obtained the fresh medium-sized potatoes. We used the cultivar Cifra from the commercial market, and the experimental design, we used a completely randomized design, and it was a 7x4 factorial experiment. The treatment and the procedures, the potatoes were washed inside a fungicide, and then they were then immersed inside a kelpec, concentrated using various concentrations for 5 minutes, and they were left to air dry, and then they were then transported into the refrigerator, stored for 60 days at 4 degrees Celsius. For data collection, we collected the glucose, fructose, and sucrose using the high-performance liquid chromatography, as shown on figure 4. And we also collected the starch granules size distribution utilizing the scanning electron microscope. For data analysis, we used the GenStat, and for analyzing the pictures from the SEM, we utilized ImageJ software, and we separated the means using Duncan's multiple range test. For calculating the TOPSIS model, we utilized the Microsoft Excel. These are the following steps that were utilized for calculating TOPSIS. However, as I've mentioned that we have also integrated a Shannon entropy, the steps for Shannon entropy start from step 3. Then the last step, it is where we rank the treatment according to the closeness to the ideal value. So the one that is further from the ideal, it is regarded as the worst performing treatment, and the one that is closer to the ideal point, it is regarded as the best performing treatment. For the results and discussion, glucose, as it is one of the main reducing sugars. At day 60 in control tubers, it was found to be high as compared to the Kelpac-treated tubers. Analysis of variance revealed that there were significant differences between the Kelpac-treated tubers and the glucose. However, it is worth noting that the lower concentrations, which is 0.5, 1, and 1.5, they have high reducing sugars, but not higher than control, and the 2% to 3%, they have lower reducing sugars. In this case, it is the glucose. This effect could be attributed to the fact that, as I've mentioned, Kelpac, it contains the phytohormones which are known to mitigate cold stress. This means that the phytohormones that are inside the Kelpac were able to mitigate the conversion of the starch to glucose. Furthermore, fructose, it is also one of the main reducing sugar. The analysis of variance revealed that there were significant differences between the Kelpac-treated tubers and the control. The same trend was observed whereby the higher concentrations, they have lower reducing sugars as compared to the lower concentrations, but both, they were not higher than the control. Sucrose, it is not a reducing sugar. However, it is one of the primary soluble sugar that is found in the potatoes. The same trend was observed whereby the control, it had highest sucrose content, and the Kelpac-treated tubers, they had low sucrose content. So this effect could be attributed also to the fact that it seems as if Kelpac was able to mitigate the hydrolysis of the starch from starch degradation to sucrose. The results, they show that the effect of Kelpac was indeed concentration dependent, highlighting the need for optimizing the treatment. The parameters and the weight of each response by Shannon entropy are shown below on the graph below, and this way they show us the influence of each parameter in the final rankings. As you can see, the glucose, it has the highest influence into the ranking process, followed by fructose and sucrose. The final rankings of the treatment that was evaluated by the TOPSIS model, it revealed that the 2.5 concentration was the best performing. This means that the 2.5 treated tubers, they had the least accumulation of reducing sugars as compared to the others. So it is also worth noting that the 2.5, 3, 2%, which are higher concentrations, they are ranked as 1, 2, 3, and the 1.5 up until 0.5, they are ranked as the 4, 5, 6. This shows that the higher concentrations of Kelpac can actually mitigate the accumulation of reducing sugars. And as we expected, the control is ranked the last, which shows that it had high reducing sugars, which leads to the formation of this acrylamide. So during starch degradation, the starch granules, they are the ones that influence how quickly or how slowly the process of the conversion of starch to reducing sugars takes place. So if they are smaller starch granules, it means that the process will take place quickly, and if they are large, it means that the process will take by slowly. As you can see, the control, it had smaller granules, which means that it has a more surface area towards the hydrolytic process of the starch degradation, whereas the Kelpac was able to mitigate that. So in conclusion, Kelpac seaweed extract effectively delays accumulation of soluble sugars in potatoes during storage as compared to the control. The 2.5 was identified as the optimum treatment using the TOPSIS entropy. Therefore, this means that Kelpac, it is a promising risk management strategy for mitigating acrylamide formation. However, further research is needed to actually validate the concentration dependency of Kelpac across various cultivars, storage durations, and storage temperature. I would like to acknowledge UL and also the NRF. Thank you.
Thank you very much, Ms. Goga. Any questions? Can we have a question at least? Questions from the floor? Okay, a question from myself. Which cultivar have you used in your experiment?
We used the sulfur cultivar.
Is it commonly used or is it a new cultivar?
Yes, it is a common cultivar, especially in the processing industry. As I've mentioned, this is the acrylamide forms when the potatoes are fried, so it is common in the processing industry.
Okay, a round of applause to Ms. Koka. There's a follow-up question.
Hi, mine is a simple question. It's because I've been hearing this word a lot and it's starting to bug me. What's a cultivar?
Okay, these are different, the different types, I can just say different types of the potatoes. We can actually refer it as a variety. There are different varieties. There's Panamera, there's Cifra, there's Mondial, and we used Cifra.
All right, a round of applause to Ms. Koka. Let's take it out. There's another question, but due to the time constraint, let's not entertain it. Please see her after, before she goes home. Okay, thank you very much. A round of applause to her. And let's welcome our next presenter, Malema. Malema. She will be presenting on validation of short to medium range rainfall and temperature forecasts. A round of applause to welcome her.
Good afternoon, everyone. My name is Mususimedi Rose Malema, and I am a first-year master's student under the Department of Geography and Environmental Sciences. I'll be presenting the paper under the topic of validation of short to medium range rainfall and temperature forecast. It's a systematic review under the supervision of Prof. Hector Chikore, Dr. Sarah Rofe, Prof. Fumurani Mativa, and Dr. Mary Jane Bopape. Let us begin by understanding why this research is so important. So short to medium range forecasts are the backbone of decision making for climate sensitive sectors like agriculture and disaster management. For farmers, they inform important choices on planting and irrigation. For our disaster management agencies, they provide the lead time needed to save lives and property. And then ultimately, they are the key tool for building climate resilience. However, the true value of any forecast is 100% dependent on its accuracy. This makes the process of validation, scientific checking of forecast against actual observed weather, an operational necessity. Despite this necessity, our field is confronted with several persistent and interconnected challenges. Firstly, while powerful models like the WRF, the ECMWF and the GFS are widely used, the methods to evaluate the performance are fragmented and lack consistency. Secondly, we know that forecast skill, particularly for a variable as crucial and chaotic as rainfall, declines significantly at about 72 hours mark. And mostly it is that there is a substantial underutilization of techniques like the sample forecasting and a pronounced gap in the user forecast validation. The process remains largely academic, often disconnected from the decision-making realities of the end users. To address those complex challenges, we defined a clear and focused aim for this review. And then the overreaching aim was to synthesize global evidence on validation practices for short to medium range rainfall and temperature forecast in order to establish a robust framework for improving regional forecast systems in climate vulnerable regions like Southern Africa. To ensure that our findings are comprehensive, unbiased, and reproducible, we employed a systematic review methodology aligned with the PRISMA guidelines. Our search was across 3 major academic databases, which was the Web of Science, the Google Scholar, and the Springer Link, which retained about 8,836 papers. And then after a two-stage screening process of the titles, the abstract, and the full text, 461 studies met all our inclusion criteria. And of this 461 studies, they constitute the foundational evidence based on our entire analysis. This structured, transparent process is designed to minimize the bias and ensure our conclusions are built upon a thorough and representative sample. Now, let us dive into the results. We first look at the bigger picture of how has this field of research evolved over time. This graph here shows a significant and steady growth in the publications, particularly after 2010. This surge is driven by an increased computational power, the growing agency of climate change, and a heightened global awareness of the value of a reliable forecast. And this trend confirms that forecast validation is a rapidly expanding and critical important field. Next, we asked, what are the tools that researchers are using? This chart here provides a very clear answer for that question. The weather research and forecasting model is a dominant tool that we see on the graph that is shown here. However, our crucial finding is that most studies use this model in a very simple way, only producing one forecast outcome. The use of assembled forecasting, which runs models multiple times to quantify uncertainty and essential for risk-biased decision-making, was underutilized. And this is a major gap that was identified. But how are these models being evaluated? Our analysis revealed a heavy resilience on a narrow set of metrics like the root mean square error and the mean absolute error. These are useful for models developers, but they provide little insight for users. In construct probabilistic metrics like the probability of detection, which tells a farmer how likely the forecasted rain event is to actually happen, were rarely used. And this highlights a methodological gap between the developer forecast and the user forecast validation. We also analyzed where this research is being produced, and the results revealed a stark and concerning global imbalance. The vast majority of our research output is concentrated in the global north, while the South African
ecological statuses of these grasses now. This is the comparison of the frequency of ecological statuses under and outside the Vachellia nilotica and Dicrostachys cinerea canopies. So outside the canopies of both Vachellia nilotica and Dicrostachys cinerea, increaser 2 grasses dominated. Now, increaser 2 grasses are grasses that are stress tolerant, are less palatable, and usually dominate in areas or in conditions that are moderately to highly overgrazed. So, yeah. These increaser 2 grasses dominated outside the canopies of both Vachellia nilotica and Dicrostachys cinerea, as well as under the canopies of Dicrostachys cinerea. However, decreaser grasses were dominant under the canopies of Vachellia nilotica. Decreaser grasses are the grasses that are then more palatable and increase with good conditions. So there was differences in terms of the effects of these 2 encroaching species on the ecological statuses of grasses beneath them, or below them, sorry. Moving forward to the biomass. So grass biomass was significantly affected by tree canopy, where higher grass biomass was found outside the canopies of Vachellia nilotica. This was the same with the total herbaceous biomass, where Vachellia nilotica recorded higher total herbaceous biomass outside its canopies. Dicrostachys cinerea did not have any significant effect on either grass biomass or for biomass, so it remained neutral. This then means that outside canopies or open areas provide more favorable conditions for more herbaceous growth and productivity, basically. Moving forward to the Mann-Hennigs richness index. So richness was also significantly affected by tree canopy, where higher richness was recorded outside Vachellia nilotica canopies. Dicrostachys cinerea still remained neutral. The Shannon-Wiener index, although the results here were not significantly, diversity was not significantly affected by either tree canopy or tree species, the mean differences showed a higher diversity recorded outside Vachellia nilotica canopies compared to under. Same story with Dicrostachys cinerea, it still remained neutral. And last but not least, the Pilus evenness index showed it was significantly, evenness was significantly affected by tree canopy as well, where a higher evenness was recorded under the canopies of both these two encroaching trees now, meaning that as much as lower richness and lower richness was recorded. The species that existed or were found under these canopies were evenly distributed. Now, in conclusion, we say that Vachellia nilotica has strong suppressive effects on herbaceous vegetation, while Dicrostachys cinerea remains neutral. And although outside canopy supported a higher productivity and richness, they are frequently dominated by less palatable and disturbance-tolerant species, which reduces forage quality. So the fact that outside canopies had more grasses doesn't necessarily mean that they are of good quality, which then leads to...
People are hungry. Any question for Yonwaba? One question at least. Oh, there is a hand over there.
Hi. Hello. It's because they said one question at least. Okay. So it's probably a simple question, but I wanted to know what motivated you to choose the size of the quadrant, the 0.5 by 0.5. Yes. It's the most common ones we have on campus or in our department, basically. Yeah. And a lot of the studies I was looking at for my literature review used that size of a quadrant. Yeah.
Okay. Okay, it's from the senior academy.
I couldn't go get to that, I'm so sorry, because of time. But recommendations would be one, in areas where these trees are encroaching in very high densities to a point that they are reducing herbaceous vegetation detrimentally, then it would be recommended that we at least practice bush thinning to reduce the number of trees that are available so that we allow for herbaceous vegetation to grow.
Thank you very much. Thank you for that. I hope you know the tradition by now, so I'm not going to be reminding you every now and then. The next student is Masilo. Is Masilo here? No, he's absent. Okay, Masilo is not here, so we'll skip that presentation. Can you find Mohlala, please? Is Surprise here? Surprise, you can come. While he is walking over, the title of his presentation is Growth and Yield Comparison of Commercial Land Races and Newly Developed University of Limpopo Copier Varieties. I hope I read it well.
Okay, thank you very much, Chair. Greetings, everyone. My name is Motlala Diaz, and I will be presenting to you the current study of my master's. I'm doing second-year master's. The study is titled Growth and Yield Comparison of Commercial Land Race and Newly Developed University of Limpopo Cowpea Varieties, supervised by Prof. Mabapa, co-supervisors, which is Dr. Vaku and Mr. Lutzol. Ladies and gentlemen, cowpea is a leguminous crop, which is scientifically known as Vigna inculata. It belongs to Fabaceae family, and it is highly valued for its versatility as well as high nutritional profile. The annual production of cowpea is about 4.5 million tons, and with Nigeria and Niger, as well as Burkina Faso, accounting for 74% of all African production of cowpea. Cowpea is domesticated from Africa and is now widely spread through Southern Asia. The production of cowpea in arid and semi-arid region faces a significant challenge due to the shortage of improved variety that are resistant for abiotic and biotic stresses. And these challenges poses a significant threat to cowpea optimal production, which ultimately affect food security. And with the world population that is increasing steadily and limited available arable land, there is a need and there is a paramount concern to increase food security. Effort has been made, and the University of Limpopo recently bred or developed 6 medium or early maturing cowpea varieties. However, there is a lack of growth and yield characterization of these cowpeas. And this poses, and this lack of information gives out uncertainty about their true potential. Hence, there is a need to characterize them. The objective of the study was to assess growth and yield performance of newly developed University of Limpopo cowpea varieties when we compare them with land races and commercial varieties. The study was conducted at the University of Limpopo Aquaculture Research Unit during 2024 and 2025 growing season, situated at the following coordinates in Limpopo province of South Africa. The site, ladies and gentlemen, was characterized by sandy loam soil, and it receives summer rainfall of between 450 to 650 mm, with the average high temperature of 28 degrees Celsius and 32 degrees Celsius, while the mean low temperature ranges from 16 to 19 degrees Celsius. The experiment was conducted, the experiment was laid out in a randomized completely block design, which consisted of 3 replications. Each replication had 10 plots with an area of 4 meters squared, with the spacing between the plant being 0.3, as well as the spacing between the row being 0.6. The experiment had 6 treatments of cowpea varieties, and the treatment was 6 newly developed University of Limpopo cowpea varieties, and 2 land races, as well as 1 commercial variety. The experimental plot had 3 rows of plants, and each row had 6 plants, and it was separated by 1 meter between. The following growth and yield parameters were collected. All measured parameters was then subjected to analysis of variance using Ginster's version 20, and Dunkel multiple range test was performed for treated means separation at 0.05 probability level. Table 1 shows growth and yield parameters of cowpea varieties, and as you can see, L1, which is land race number 1, took more days to flower after planting, and UL13, which is UL variety 13, that's how they were named. They took lesser days to flowering, and this variation, of course, suggests that there is a genetic makeup and photoperiodic sensitivity, and possibly adaptation to specific environmental conditions. And fortunately, this study is supported by Gennaro et al. 2015, who reported that there is a significant genetic variation that exists among cowpea varieties in terms of flowering time, suggesting that the observed differences were likely due to inherent genetic diversity among the cowpea varieties. Table 2 show yield parameters of cowpea varieties, and the highest yield was obtained from UL12, which was 490.7 kg per hectare, while Dr. Sanders, which serves as our commercial variety in this case, and landrace 2 produced the lowest of 190.5 kg per hectare and 253.7 per hectare. This observation, ladies and gentlemen, can be attributed to the fact that UL12 variety exhibited a semi-erect growth rate, which allowed for better light interception and air circulation within the plant canopy, which in turn promote healthier plant development. And again, is caused by inherent genetic variability among cowpea varieties. And our findings are in line with Sine et al. 2019, who reported that different cowpea variety can exhibit varying yield potential. Ladies and gentlemen, there was a significant difference among cowpea, among growth and yield parameters of cowpea. And newly released UL cowpea varieties, particularly UL12 to UL16, have a superior growth and yield traits compared to the traditional landrace and Dr. Sanders. And therefore, the study rejects the null hypothesis. And again, it recommends that this variety, newly developed cowpea varieties, be adopted for wider cultivation, and especially in a region where cowpea is a major food and income crop. And again, we recommend that further studies be done in different ecological regions to ensure validity of this cowpea, and as well as we recommend that this cowpea be compared with other existing commercial variety and land races. I would like to acknowledge the following stakeholders and Almighty God. Thank you. This is the references.
Thank you, sir. You finished within 10 minutes. Any questions for him, 1 or 2? There is a hand over there.
Hi, brilliant presentation, and I like that you acknowledge God is great all the time. So I wanted to just check, how would you describe your study to let's say a villager somewhere, like how would you describe your how would you explain your findings to some old grandmother somewhere who's involved? I'm not really big on agriculture, so some of the terms, they escape me.
Okay, thank you very much. Though I'm not sure if I got your question, but in terms of describing, I will say my study is simply on the characterization of the cowpea varieties that were only developed for certain characteristics. And as you know, as a farmer, we cultivate for different purposes. And recently, the cowpea remain, allow me to say, they are not really significant to a farmer who prioritize yield. Hence, I jumped in to say no, because these varieties were then bred for adaptation. Let me check the yield potential of this cowpea, so that when I introduce them to the farmer, I will be able to tell, or this variety, how are they important to the existing commercial and land races. I'm not sure if I answered you. Thank you.
I said one or two, one last one, if any. If there is no question, I will ask myself. What do you mean when you say newly developed University of Limpopo varieties?
Okay. What I'm trying to say, another term for develop is bred, meaning this cowpea was, if I can use normal English, I will say they were made, or yes, they were bred by one of our late professor in the university, meaning there was a breeding that occurred, and then they came up with these seeds for the cultivar then.
Okay. Yes, I'm not sure. I think we have answered. Okay, thank you. Thank you very much. You can clap for him. Okay, the next presenter is Jablan Doli. The title of his presentation is Disentangling and Closing the Nutrient-Based Potato Yield Gap Using Integrated Nutrient Management Under Temperate Environments of Sub-Saharan Africa. Over to you, sir. But why don't you use this?
It's more simple with this.
You can start.
I'm not sure if the laser is visible. I'm not sure if this laser is visible.
Is it what?
This laser.
That is your mouse, it won't show there. Yes,
okay. I want to know if this thing is available on the...
Okay, good evening everyone. My name is Chabulani Ntule, PhD student at the University of Limpopo, and it's with great pleasure to stand in front of you and present to you a recent paper on disentangling and closing the nutrient-based potato yield gap using integrated nutrient management in a sub-Saharan African region. Currently, Asian and European countries are the biggest potato producing areas in the world, largely driven by high intensive input systems. In contrast, African countries still have the lowest potato production, largely constrained by limited input systems. There are various interlinked factors that contribute into this yield gap. Among these includes chronic mismanagement and underutilization of fertilizers, where in the sub-Saharan Africa, there is low usage of fertilizers, largely constrained by high prices of fertilizers, limited access to quality fertilizers, and also lack of soil testing facilities, and also the general expensive cost that is associated with doing soil nutritional analysis for fertilizer application. Further exacerbating these issues, it's the climatic trends that shows a marked increase in warmer days, more infrequent prolonged duration of heat waves, and also increase in the overall maximum temperatures. Such factors, they contribute into increasing the potato nutrient-inspired yield gap, and this limits on-farm productivity and also makes it difficult for many resource-constrained farmers to compete in the local and regional market and potato industries. So we then conducted field experiments over 2 summer growing seasons in order to investigate the underlying causes of nutrient-inspired yield gap and also quantify these yield gaps and further develop an integrated nutrient management strategies that can assist in narrowing this yield gap. Our integrated nutrient management approach consisted of 3 practices. The first one, it was fertilizer application rate that consisted of 100% of the recommended rate to represent farmers who can afford to apply these expensive fertilizers. Then we also had 50% of the recommended fertilizer application rate to represent those farmers who can apply the fertilizers but in suboptimal amounts. Then you had control plots where we were not applying any fertilizers to represent farmers who do not apply fertilizers largely due to financial constraints. The second practice, it was soil surface cover, where we use a grass mulch to form a layer that can moderate the soil temperatures, therefore reducing the soil evaporation and nutrient losses that occur through volatilization. Then the last practice, it was cultivar selection, where we had 4 potato cultivars that differed in terms of time to maturity. Here we're testing if this, the time to maturity can have a nutrient uptake window that can be an advantage for closing these yield gaps. Now, our investigation indicated that when farmers do not apply fertilizers, yields are significantly very low, ranging between 5 and 8 tons per hectare. And this is the yield that most of our farmers currently are getting. Now, the difference between these low yields and the potential yield of 50 tons per hectare revealed a substantial yield gap of about 42 to 45 tons per hectare, of which is the yield gap that is caused by chronic mismanagement and underutilization of fertilizers. Now, when we applied a full fertilizer application, we were able to close this yield gap by about 21 tons per hectare. This indicates that about half of the nutrient-inspired yield gap is caused by not applying fertilizers. And this is also in agreement with other researchers in many other communities that indicate that there is an urgent need to improve farmers' access to fertilizers in order to improve their productivity and also give them a better chance to compete in the local and regional market and potato industries. Now, despite now the improvement in the yield, there was still a remaining yield gap here of about 21 tons per hectare, of which is the yield gap that is caused by nutrient losses, where we apply fertilizers, but we still get the yield gap that is caused by things like volatilization. Now, when we applied a soil surface cover, of which is grass mulch, together with full fertilizer application rate, we were able to close this nutrient loss yield gap by about 7 tons per hectare, of which is 33% of the nutrient loss yield gap. This indicates that applying grass mulch can be it's effective in moderating the soil temperatures, therefore reducing the outward losses of nutrients through volatilization. Now, the problem here is that grass mulch does not completely eliminate all the losses, because we still get a significant portion of the nutrients that remains in the soil after harvest, especially if farmers plant early maturing cultivars such as Suburban. Now, we used a late, and this is also shown by a persisting yield gap here of about 14 tons per hectare. Now, when we used a late maturing cultivar, of which is the Panamera, together with the full fertilizer application and the grass mulch, we were able to further close the nutrient uptake yield gap by about 7 tons per hectare. Now, the late maturing cultivars, they have a nutrient uptake advantage because they stay longer in the soil, therefore having a prolonged nutrient uptake. They also have traits such as early robust canopy cover development that also assist in efficiently taking up nutrients. Now, with this full INM approach, we were able to close about 83 to 84% of the nutrient-inspired yield gap. Then we also had a remaining gap here of about 7 tons per hectare, of which is the yield gap that unfortunately we could not close. And this can be caused by other various underlying nutrient losses that our INM could not account for. Now, what happens when farmers cannot apply full fertilizer application rates? Our investigation indicated that farmers, they can actually reduce the fertilizer application rate by about 50%. But so long as they integrate that with grass mulch, they can still obtain acceptable yields of about 38 tons per hectare, of which is enough for these farmers to compete in the local markets and potato industries. And also, with this integration, mulch also indicated to be more beneficial under a reduced fertilizer application. And together with the late maturing cultivars, they can obtain yields of about 41 tons per hectare. Here we emphasize that fertilizers alone is not sufficient to close the yield gap. These fertilizers, they must be complemented with nutrient loss mitigation strategies such as applying grass mulch. Now, with our study, we were able to identify and quantify various nutrient-inspired yield gap, such as fertilizer application yield gap, nutrient loss yield gap, and nutrient uptake yield gap, of which farmers can reduce or narrow this yield gap by applying fertilizers and integrating this with various nutrient loss mitigation strategies. We urge our researchers to also look into addressing the residual yield gap in order to completely close the nutrient-inspired yield gap by looking at other underlying causes of nutrient losses. We're also in need of a comprehensive cost-benefit analysis to indicate if can farmers adopt this INM approach and still maximize their profits. I would like to acknowledge the University of Limpopo, Northwest University, and the CRISP project. For more scientific discussion, you can please kindly read this article, and thank you very much.
Thank you, Chablan. I think you finished within 10 minutes, that's fine. One question? Okay. I see two there.
Thank you, Jabulani, and you're not the first one to present on application of fertilizers. I'm sorry if you are going to catch this stray bullet. So it's more of a comment. I mean, the world is trying to shift from the use of these, synthetic fertilizers to organic fertilizers. Hence, you know that these synthetic fertilizers are, to some extent, the cause of your health gaps that you have identified. Hence, they cause global warming and some of the climate change effects that ultimately will cause a reduction in production of potatoes and other crops. So how do you justify the use of these synthetics while we are trying to move away from the use of those fertilizers?
It's a comment, but still, it's a discussion.
Yeah, I mean, how will you persuade farmers to use this synthetic while we are trying to move away? I know that they are more concerned about
I get the question.
Maximizing profit.
Yes, thank you very much. And I agree with you, okay? The problem is most of the fertilizer recommendation are one size fits all, where we often oversaturate our soils with fertilizers. Now, our investigation indicates that we can actually reduce the fertilizer application rates so long as we integrate with other nutrient loss mitigation strategies such as grass mulch. So if we can reduce that, then meaning we can reduce those negative effects of chemical fertilizers. But the fact still remains that we need these chemicals in order to improve productivity. As much as they are bad, at least if ever we can find ways in which we can reduce these negative effects by reducing the fertilizer application rate and complement it with other maybe organic maybe fertilizers such as manure or other nutrient loss mitigation strategies. But the fact still remains, we need these fertilizers. Yes.
You wanted to ask, sir? Quick one, please.
Yes.
Yeah, just be quick.
Thank you for the opportunity. Well, I only have one question. So, given that potatoes have been studied a lot recently, I want to know what makes your study stand out in a way that it adds value or it has practical impact for farmers or, to be precise, the potato industry.
Thank you very much. Yes, there are a lot of studies that are on potatoes, but the yields for potatoes are still low, ranging between 6 and 10 tons per hectare in sub-Saharan Africa, meaning there's still a need to do more research. And we haven't had a paper or researchers that look into quantifying, doing a field quantification of nutrient-based yield gap. And also, there's also a need to do this because the yield gaps are quite huge between Africa and other leading countries such as China, India, hence I mentioned Asian and European countries. Now coming to practicality for farmers, grass mulch is more accessible and practical practice that farmers can use. You can simply go to the nearest bush, cut grass, and apply as a mulch. So, and also with maybe reducing fertilizers, we are trying to accommodate those farmers who, especially resource-constrained farmers. Did I answer the question?
Yes. You have answered. Whether you answered correctly, take your stuff. Okay. Thank you very much. You can clap for him. The next presenter is Cedric. And the title of his presentation is, Cedric is there, and the title of his presentation is The Health Status of The health status of drinking water contaminated by tailing dust in subtropical regions. So I'm going to take a mark for putting that.
Okay, thank you so very much, Chairperson. Good evening, ladies and gentlemen. My name is Cedric Selwani, as already stated. So, with the topic of my study, yes, it's true, it's not a mistake. It's actually the health status of DAOs, drinking water contaminated by tailing dust in subtropical region. So the word DAS simply means adult female goats, those who are not in animal production side. So it's not a mistake. I heard the chairperson when he was reading, he thought maybe there was a mistake. So yes, it's true. Health status of DAS, drinking water contaminated by tailing dust in subtropical regions. So this is the outline of my presentation. I'll be taking you through, which start from introduction until the references. So tailing dusts or tailings are mine waste generated after the iron is extracted from the ore. So the very same tailings, these waste products, we are talking about, they present a major health challenge for goats drinking contaminated water in subtropical regions. So we all know that the policy with regard to mining, it says at least the mines must make sure that there are dams where they deposit these waste products. They are always properly managed. However, that is not always the case, because we know that at some point, they always experience some overflows, which as a result, they subject this animal or livestock to a danger. So if this policy can actually be followed, where the mines, dams are properly managed against leakages and evapotranspirations of these very same tailings we are talking about today, this will of course allow an improved goat reproduction performance and health status promoted by the water, which will always be clean and available to animals for drinking. So hence, and then understanding the effect of these heavy metals, which are the tailing dust, the very same waste products, in drinking water from tailing dust could actually improve the aquatic life, not only the livestock. We also looking at the crops, they could improve, and people in general. So this will further increase awareness on the possible health consequences associated with these tailings dust contamination for goats or for water used by goats for drinking. So the aim of the study was to assess the effect of tailing dust contamination, contaminated drinking water on the health status of non-lactating goats. So specifically, we were examining their health status, looking at the body condition score, the live body weight, the corneal opacity, as well as the pharmacia scores. So the objectives was to assess the effect of this tailing dust contamination on water used by goats for drinking, looking at those health parameters I've just mentioned. So it was hypothesized that body condition score or all these 4 health parameters will not be affected by water contaminated by tailing dust. So the site or the study site, we actually involved 2 communities in this study, which are located in Palaborwa, which is in this Limpopo province. And then one community being Ramakushani, and then the other one being Ramashishimal. The Ramakushani area was the one which their goats were exposed to these mining waste products. And then Ramashishimali was a community area which was far from this mine. So in our experimental unit, we used 12 goat farmers, each farmer contributing 8 goats, and then giving us a total of 96 non-lactating ducks which were involved in this study. And then the selection of these goats, the criteria, we were looking at the household proximity to the Bushveld mine, which is this very same mine located in Palabora. So, and then the number of goats in Ramakushani, which was the area contaminated by these tailings dust, we only involved 40 animals or 40 ducks, which are these female goats. And then in Ramashishimal, we checked 56 goats. So the difference in number, it also tells you that these are the number of goats, female goats, which are non-lactating, which we could manage to access. So the difference, the other one being 40 and the other one being 56, simply tells us that in those contaminated area, which means these farmers, they were actually losing their livestock due to these tailings. That's why we only managed to get only 40 animals, whereas in the other place, we managed to get up to 56 non-lactating goats, female goats. So these goats were managed on free grazing communal rangelands, and then goats, they source their drinking water from either contaminated rivers or uncontaminated, depending on the community, whether it's in Rama Shishimale or Rama Kushali. So the selected goats were marked and monitored during their foraging journeys to the water sources, and then they were observed by herdsmen which were heading these goats which had over 10 years experience of heading these goats, of which they watched them from a distance of approximately 50 meters to minimize disturbance to these animals while they are busy drinking or foraging. So the experimental design, we involved a cross-sectional design where we focused on the household's demographics, and then it was guided by, alongside the availability of these non-lactating, or we can refer to them as dry does female goats, and the willingness of the owners to participate in the study. And then the parameters which was evaluated, or the factor, it was the health status of these animals. So we had the factors and the variables. So under factors for treatment, either the area was contaminated or not contaminated. And then the levels we had from the contaminated area, it was only 5 households, which they gave us, each household was contributing 8 to give us 40 animals. So from the not contaminated area, we had 7 households, each giving us 8 goats to make it 56. And then in total, we had 96 goats. So the variables which we measured are the ones I mentioned, the health status, which is the body condition, live weight, and the Famacha score, as well as the corneal opacity. So we also tested the water. For water treatment, we used 6 samples of water, and then collecting 6 samples from each area. Each area gave us 6 samples, and then which we had a total of 12 samples from both areas. And then in the water, we tested for heavy metals such as copper, chromium, magnesium, and lead. So the site, or where we tested this water, it was at the University of Limpopo Agro Food Technology Station, also known as LATS. And then under data analysis, we used SAS 9.5 2023 software. The general linear model was employed, and then where we had mean separation techniques that was used was Fisher's LSD. The significant level alpha was set as 0.5%. And then this was the following statistical formula we used. So under the results, we had 2 areas, remember, contaminated and non-contaminated. So this first table, table 1, just shows those heavy metals which we tested. And then the highlighted ones in yellow, they are those which were present in the water samples, in both contaminated and non-contaminated. So table 2 is the scorecard we use to score those goats, depending on or focusing on their body condition, with 1 being the animal being emaciated, and then if it scores 5, it's too obese, but if it's around 3, then it's in good condition. So table 3 is also showing the scorecard, but this time is for corneal opacity. If the scorecard is very high, it means the animal, it has some swelling lids, which indicates that it has some difficulties in eyesight. Whereas if it scores a low score of 0, then there's no opacity. The animal appears to be crystal, normal, or clear. And then these are some of the ratings which we use for Famacha score. And then for our table, last table for table 5 results, if you can check the 2 areas where we had contaminated or non-contaminated, I will focus on body condition. You can see that animals from the contaminated area, their body condition was very low as compared to uncontaminated. It was 2, which shows that they were lean as compared to those in uncontaminated area. Checking their body weight, of course, it correlates to the body condition. The body weight was low in contaminated. It was 20, uncontaminated, it was 27. The corneal opacity, it also says the same because it creates the links between these 2 parameters. So checking on the correlation, we had a... Okay. On the correlation, we had positive correlation between corneal opacity and body condition score, as you can see. So in our conclusion and recommendation, we saw that tailing dust contamination do significantly affect the health status of these animals, as it was characterized by low body condition score, and then as well as high corneal opacity. So these indications suggest poor health goes among those exposed to tailing dust. So in conclusion, we can say integrating body condition and corneal opacity assessment into goat management program is recommended for monitoring the health status, particularly in those regions where goats are exposed to tailing dust. So these are the references.
You don't have to read them. We know you.
Thank you for lending me an ear. Thank you.
Yeah, thank you. One short question, please, for him. One short question. Yes, you over there. You can. But did you hear the question?
Yeah. Okay, thank you for your question. The main reason why the study was gender-based specific is simply because, remember, with farming, most farmers, they prefer to keep female animals as compared to male animals, simply because they focus on the production side. They can bear the offspring for them. So you'll find the reason is because those farmers which we approached, they actually had many females animals as compared to males, because they sell males anyway, since they are not involved in their production cycle. So that's why we actually focused on the female more than the males. Thank you for that question.
Okay, thank you. If there are further questions, you can engage him after this. We still have a long week, so yeah. Thank you. Is the last presenter here? How does it feel? Oh, it's you. I don't want to butcher your name. Okay, our last presenter is Tibulawi, and the title of her presentation is Strengthening Traceability in Organic Agriculture Through Digital Payments and Automation, Pathways to Building a Climate Resilient Supply Chain in South Africa. Over to you.
Thank you, Chair. Good evening, ladies and gentlemen. Good evening. I know I am the one between you and going home. So he was telling me that I have 20 minutes because I'm the last one. So if you are nice to me, I will take 5 minutes. My name is Tibulawi Nebutaru, and I will be presenting my study on strengthening traceability in organic agriculture through digital payments and automation, pathway to building a climate resilient supply chain in South Africa. So this is one of the offsprings from my main study, my PhD study in digital payment awareness, perception, and willingness to adopt, checking the nexus between adoption and credit access by smallholder farmers in Vembe and Mupani districts. So while I was doing a literature review, synthesizing literature, I came across one of the benefits of digital payment, which is traceability. On the main study, we focus on financial inclusion of smallholder farmers using digital payment. So organic agriculture is increasingly recognized worldwide as a driver of sustainable food systems, environmental stewardship, and inclusive economic growth and human health. So traceability in this case is the capacity of being able to track back and authenticate the tracking history of a product from its origin to its to the final consumer, what we normally say from farm to fork. So automation improves supply chain efficiency by minimizing human error, enhancing real-time data captures, and enabling quick responses in times where there are disruptions. And also organic farming in the South African setting has a huge untapped potential to drive inclusive rural development and also export opportunity reinforcement. So the problem is that advances in these digital technologies such as mobile payment, blockchain, and automation have been shown to improve supply chain transparency and reduce risk and fraud and inefficiencies. But South Africa, however, when it comes to our organic agriculture, we are still facing unique challenges that undermine the potential for us to contribute to food security and rural livelihoods, and also for climate adaptation using these digital payments. So the sector is also constrained by weak traceability infrastructure, limited integration of digital innovations into farming, certification system, and also the big threat which is called the climate change. And then, however, there is empirical research on how digital tools can be tailored to the realities of South Africa's organic value chain. But this leaves our policy makers without sufficient evidence, because every analysis on this digital payment tool, it is done on I can say on a third basis level than on the primary level, where it involved the small-scale farmers. So this study was done so that it can set out effective models of digital adoption and also analyze transferability. So the aim of the study, the aim of this review is to evaluate the potential of digital payments and automation in enhancing traceability and climate resilience within the South African organic agriculture. So the objective, the first one was to assess how this digital payment and automation can improve traceability and climate resilience in South Africa, and also to identify the barriers and opportunities that exist in adopting these technologies. So the boundaries of this review, the geographical focus was South Africa, but with lessons which are drawn from other neighboring countries. And then the thematic focus, it was digital payments, automation, traceability, climate resilient, and organic agriculture. So systematic literature review approach was used and organized with the preferred reporting item for systematic reviews and meta-analysis, commonly known as the PRISMA framework. So the review was centered at the nexus of digital technology. So we used the Google Scholar, Scopus, Art Discovery in order to source our data. So theories like the technology acceptance model and the diffusion of innovation, they were also applied in order to draw or for guiding the thematic interpretation of the result. So the term, that's where we got the perceived usefulness, the ease, and the attitude with diffusion of innovation, together with the diffusion of innovation about innovation, attribute, and attitude. So this is just the PRISMA flow diagram. The first synthesis resulted in 152 studies, and then after duplicates were removed, we still remained with 152 studies, and then the records which were excluded based on the title or on the abstract, they resulted to 84 studies, and then we also had full articles which were assessed for eligibility, so it resulted in 68 articles. So the full articles which were excluded were 31, and the reason for this exclusion was that they were not agricultural related, they had low quality, or they were urban focused. So the studies which were included in the final analysis were 37 studies. So each paper was assessed using a quality appraisal score, and then the coverage ratio also was greater than 50%. So the literature review robustness index, which was calculated using the formula that you can see there on the slide, it resulted in 1.85, which indicated a high level of methodological integrity and strong thematic saturation across all the studies which were included. So this is some of the selected studies on digital innovation in agricultural traceability. For example, you can see that the first one there, we have a study by Liv et al., which was conducted in 2013, and the country was China. The digital tool or the focus were blockchain traceability. The methodology which was used, it was a case study, and then the main contribution of the study showed that the effectiveness of blockchain in organic supply chains. So from the synthesis, the global experiences, they revealed that countries like China, they use this blockchain adaptation, which they combine with this with QR codes. So what the literature revealed is that in China, what they basically do is that they combine, they attach QR codes to their perishable product, where when it reaches the final consumer, this product is scanned at each and every point of the value chain. So when it reaches the final consumer, you are able to scan using your phone to see the life, all the processes that your product has went through. Even countries like India, they have their digital infrastructure where they try to motivate farmers to adopt to these technologies and innovation. We have their ENAM and their AgriStack, and together with European unions, they are using organic regulation in order to enforce this digital record keeping. So in European unions, in order for a farmer to get a good agricultural practice certificate or water rights certificate, they need to comply with a digital record keeping, labeling of their product, and their supply chain transparency. Even in USA, big retailers like Walmart, they are using their artificial intelligence and Internet of Things, especially in their agricultural produce. So the findings of the review indicated that in South Africa, we still have a clear gap between the potential of these technologies and their actual usage, especially by our small-scale farmers in rural areas. So we still have issues like low infrastructure, high certification cost of your water rights certificates, and then we still have fragmented markets where we have many buyers and sellers, but no one has control over the market. There is still network unreliability, especially in rural areas, unreliable electricity, and unaffordable devices, because in order for you to adopt to this digital payment, you need to be able to have a smartphone, and which for some farmers it might be expensive. Although there are initiatives by our MTN Momo and Vodacom in Pesa, informal trading and inconsistent labeling, they are still persistent in these rural areas because our farmers are unable to use this digital platform because they still perceive it as risky and as a way of them being scammed of their monies. So the recommendation, I strongly recommend that we revise our financial and technological support to these rural areas, that when we have identified these technologies, we should not just only deploy it at a higher level, but we should also include our farmers. And also in regard of education and extension, the manuals and the instruction of these digital tools, they should be translated to the native languages so that our farmers can understand better. So in conclusion, again, we must also revise the digital economic plan together with the digital roadmap in order to include our small-scale farmers. So in conclusion, I will say deployment doesn't measure success. If you want to go fast, go alone. If you want to go far, go together. So this shows that there is a need for collaboration. The best time to plant a tree was 10 years ago. The second best time is now. So this is to indicate that let's not only have this FSA where we come and share ideas, we come and do all these policy recommendations. There should be a follow-up conference to say, what have you done with your ideas? Where did you take your recommendation? How are you far with your strategies? Ladies and gentlemen, acknowledgement, references, thank you.
Hey, yeah, man. Well, thank you for that. Thank you for the energy. Any question? One question, please. One question. Thank you for saving us. Okay.
Hi. Hello. That was a very nice presentation, and I liked how you touched on the literacy part because that's actually a challenge in most rural areas. Some people don't even have, so I like how you covered everything. So I like that. I don't have a question, I just had a comment. Nice.
Thank you. Thank you so much for that.
You don't have any question? Okay, if there's no question, I will release her. You know, but thank you for a nice presentation. The energy was was super. Ladies and gentlemen, that's it from this last session. Thank you very much for that. And on behalf of the organizing committee, we would like to thank everybody for participating. We really appreciate the students, the enthusiasm that you have shown, the quality of your presentations was really brilliant. And we would also like to thank the chairs and the co-chairs, the adjudicators for their work and time. We really appreciate what you have done. I know you have been sitting here throughout.